Good morning, everyone. Happy Friday, and welcome to the second day of this symposium. We were treated yesterday, I think, to a series of fabulous and diverse talks that gave complementary views on brain patterning, from comparative anatomical insights into brain evolution, to molecular studies into early brain patterning and experience-dependent plasticity, based largely on work in rodent model systems. Today's talks are going to build on these foundations and shift our focus to work on human and non-human primate brain development. So the speakers in the first session use in vitro, in vivo, and theoretical models to describe aspects of primate brain patterning and identify axes of this patterning that, if extrapolated, might be particularly relevant for human disorders and faculties. The speakers in the second session more directly consider the interface between these features of brain development and predominantly genetic risk for psychiatric disorders. Our keynote speaker today, Dan Geschwin, has published seminal studies that span both of today's session's topics, and we're delighted to have Dan here, and I'm really honored to introduce him. So Dan holds the Gordon and Virginia McDonald Distinguished Chair in Human Genetics and is Professor of Neurology and Psychiatry at UCLA. At UCLA, he also directs the Center for Autism Research and Treatment and the Neurogenetics Program. He also serves as Senior Associate Dean and Associate Vice Chancellor for Precision Medicine at UCLA. Dan received his MD PhD at Yale, where he trained in neuroanatomy and developmental biology, and then pursued postgraduate training in genetics at UCLA, where he's been since. He and his lab have played key roles in the science and consortia building that's underpinned the recent surge in discovery of bona fide risk variants for neurodevelopmental disorders. And he complements his work on neurodevelopmental disorders by also studying neurodegenerative alterations of the human brain at the other end of the lifespan. His lab's really been at the forefront of applying bioinformatic and molecular methods to understand brain patterning in typical development and to use these models as a context for functional characterization of autism spectrum disorder, predominantly ASD risk genes. He's published prolifically. He's a member of the National Academy of Sciences, has been awarded the Ruane Prize in Child Psychiatry, and is on advisory boards for the Allen Institute and NIMH. His papers often become, as I'm sure many of you would agree, kind of key landmarks in the field, and major publications of note have been characterizing some of the first to characterize post-mortem gene expression in autism and gene regulation and cell patterning in typical development. And it always strikes me that his papers kind of seem to magically, consistently ask the right question at the right time with extreme methodological rigor and creativity combined. And finally, I just want to note that Dan's creativity and his lab's creativity isn't just at the level of theoretical, empirical, and technical advances in developmental neurobiology, but also at the level of training the next generation of people working in this field. He's a really genuinely kind of caring and thoughtful mentor, and many of the people who've worked with him are now starting their own programs at kind of key institutions. So really, I'm going to stop and let the magic start. So I hope you join me in welcoming Dan.
Slightly embarrassing, just slightly. Thank you, Armin, for the very kind introduction. I also want to thank Armin and the other organizers for really pulling together and really congratulate them. This is, so far, we're only halfway through, and it's already been an extraordinary meeting. I just enjoyed listening to every single talk and look forward to the rest of the day. So it's really, really a privilege and an honor to be here speaking among all these other people whose work I admire and leverage in what we do. So I'll just jump right in. I have to, my one, here's some conflict of interest slide. I'm not talking about any of this stuff today, but I always put it up there just for just being complete. I'm going to have some word-finding difficulties because I think it's about 5:40 a.m. in California. And, hopefully it will get better as the talk goes along and the wheels and the oil kind of starts flowing. So, I was kind of thinking last night, how does all the molecular stuff that I do tie together to the really brilliant kind of neuroanatomical connectivity, developmental biology, and stuff that we heard about yesterday? And I was thinking about, kind of what we do and what I've been doing for the last 20 years. And in some ways, I can think about what we've been trying to do is look ahead, and there are territories. And this term, no maps for these territories. We've been map making, and what I'm going to talk about today is really making molecular maps and thinking about how genes connect to each other and gene regulation works in a kind of map-like form. This is not my term. This is actually a friend of mine, Mark Neal. This is early on in moving to Los Angeles. My wife and I was really interested in William Gibson, and this friend of mine who was a director made this documentary of William Gibson, the science fiction writer who wrote Neuromancer. And the terminology that came from this interview with William Gibson was there are no maps for these territories. William Gibson is very famous for coining the term cyberspace. And really, a lot of the modern movies like Matrix and other things are based on his short stories. So, and in this movie, there's not only music by Bono and The Edge, but there you will see Jonah Scooch Geschwin, Stunt Baby. I'll just leave it at that. So today, with the map idea, genetic risk for autism is complex, involving many loci. So I'm just going to basically give you the talk now, and then if I lose you, you can just come back to these 3 or 4 things that I'm going to mention. So it's very complex and involves many loci. And I'll talk about that briefly. Autism risk genes, however, despite extraordinary heterogeneity, converge during early brain development. It doesn't mean there aren't some that act later, but if you ask where they converge, it's during early brain development. And I'll talk a little bit about the time, cell type, and biological pathways. Then I'm going to, because of that, it really gave us a sense that early development is critical. And this gets to the map making. It became clear there are these amazing projects like ENCODE, that there just wasn't the appropriate maps for human neurodevelopment. And since gene regulation occurs in a cell type and stage-specific manner, it was critical to make specific maps of gene regulation in developing human brain. And so about 6 years ago, we started to think about this and eventually have been contributing to this effort. So I'm going to talk a little bit about 3D chromatin structure and open chromatin using 2 different techniques, the notion of expression quantitative trait loci, as well as, which is essentially how does human allelic variation, how do SNPs influence gene expression, how do SNPs influence splicing, which would be a splice QTL. And then, I'm going to touch on, I decided to pull out the single-cell transcriptomes because Damon Poliadakis is going to talk about that in one of the Data Blitz talks, but I will touch on it briefly. So autism is a heterogeneous neurodevelopmental syndrome. We all know this. We've hopefully all heard this. And one of the kind of complexities in thinking about autism, like other neurodevelopmental disorders, is that it shares not just comorbidity but risk with intellectual disability. About a third of kids have intellectual disability. Maybe 10 or 15% have epilepsy at different epochs in their lives as well. So if you think of those 2 major neurodevelopmental disorders, you could begin to think, in one framing. I'm not saying this is the appropriate framing, but one framing is that, essentially you put a crowbar, either a genetic crowbar or environmental crowbar into the brain, some kind of damage, during development, and you can end up with a range of neurodevelopmental disorders. And so, this kind of fits back to this notion of canalization that we've, kind of stolen from Waddington, which is the notion that what these really share is abnormal circuit dysfunction. There aren't genes for autism per se. The large effect size mutations that are mutated that affect genes are in core developmental genes. So what's happening is development is being disrupted, and that increases the risk for a panoply of neurodevelopmental disorders. It may not be nonspecific, totally nonspecific. In fact, we think the evidence that we have and that I'm going to share with you shows that there is some specificity to this, but it's also broad risk as well. So there's, it's not really a paradox in that every gene that causes intellectual disability doesn't increase risk for autism and vice versa. But there is a kind of overlap. And if you think about this as a disruption of normal neurodevelopmental processes, and then a failure of the organism to be able to compensate appropriately, then I think that's a kind of probably an appropriate model. But what autism shares distinctly are defects or abnormalities or atypicalities in social cognition and what I kind of call mental flexibility, or the presence of repetitive and restrictive behaviors. And another piece of this is that one also has to think, and I don't have time to go into it in gory detail, but I just always need to mention it, is that a part of the genetic risk for autism, a huge component, is polygenic risk. That means common genetic variation that we all share. And then the question is, how does that cause autism? Well, it's likely affecting a kind of, let's call it, this is an older slide, 3 major components: the language, the sociability, and the mental flexibility components. You can imagine that because these have distinct brain circuits, they have some overlap and some distinctions between them, that there'll be shared and distinct risk for each of these. And think of a normal distribution or contour of genetic risk, and folks with autism are just at one end of that normal distribution. So in that way, you can think of it as a continuum with what we call normal. And we just call people over one side of it abnormal because of the adaptive behavior, what it does in society. But it's just part of the human condition from that perspective. So, the last 15 years of genetics has been extraordinary in this field, giving us a foothold to begin to do neurobiological studies, and you heard about many of them yesterday. Genetic testing can currently identify about 20% of mutations contributing to autism via clinical microarray, Fragile X testing, some other single gene testing, and now exome sequencing. And so, kind of what have we learned? This is a few years old, and it's a review that was written by several postdocs in the lab, led by Luis de la Torre-Ubieda at the time. And so, what you can see here on the left are a lot of the findings from this exome sequencing. In the red there, then the copy number variants below it all the way on the left, and then the kind of syndromic forms of autism. It's been known for 30 years. Mary Coleman and Chris Gilberg had published several papers and actually a book, that autism was strongly connected to a large number of medical genetic disorders. It was thought previously that those were kind of separate, distinct. And it's now clear that a portion of autism is a collection of rare disorders, none of which account for more than 1% of autism. In fact, if you just took the top 10 exome sequencing causes, I think they might add up to just a couple percent totally. What we can see all the way on the right is that there's still quite a bit unaccounted for. There's still the predicted common inherited portion that still largely remains to be defined. And that what we've identified so far is around 10%. And likely, as we continue to discover, maybe the rare de novo stuff will account for 15 or 20%, leaving about 80% due to common variation and inherited rare variation. So where we are right now, and this was articulated by Morganka yesterday, is we can predict that there are going to be many genes, hundreds if not thousands. None account for more than 1% of cases. There are highly additive effects, and there's strong pleiotropy. That is, especially some of the large effect mutations that affect brain development, increase risk for other neurodevelopmental disorders like schizophrenia and ADHD or epilepsy. And what I'm not going to show here is also that even the common genetic risk shows quite a bit of overlap. If we look at the correlation in common genetic risk between autism and schizophrenia, it's about 0.2. So there's shared and distinct risk. And so one of our jobs is going to be to kind of use that genetic risk to ferret out the different neurobiological pathways. So one question that we've been asking almost since the beginning is there, we know it's going to be incredibly complex, is there convergence? And you can think about convergence at the level all the way to the right, at circuits, and as a clinical neurologist, that's how I think, neuroanatomically. And so clearly, certain circuits that are involved in social cognition have to be disrupted. But then the real question at a treatment level, if we kind of frame this like cancer or other biomedical diseases, if we're going to target specific mutations or proteins, is there convergence at the molecular or kind of, biological pathway level? And that's been a major question that we've been asking and that people are beginning to answer. And there is evidence that, there is convergence. I'm going to share some of that with you very, very briefly and then go on to the map making. The way that I think about this convergence is in this framing, that you have genetic variation and it's acting through mRNA and protein, through synapses and circuits all the way to behavior. And our challenge is to actually connect these various levels. And it's going to be necessary to use model systems likely to do this, as people discussed yesterday in detail. And I think that's going to be incredibly important. But one of the things we have to understand is how similar and different are the model systems that we're using to our in vivo actual normal human brain development. And that's why the evolutionary perspective is absolutely so critical. What I'm going to be talking about is measurements at the mRNA level. It's not because I think mRNA is the be-all and end-all, it's because I can do it. It's feasible. It's inexpensive. I can do it in high throughput. I can do it with high dynamic range, from one copy to cell to millions in a tissue. And I can do it in hundreds or thousands of samples, which makes it a very feasible and powerful technology. The other piece of this is that genes don't act alone. They act, they're co-regulated, and they act in pathways. And so what I'm going to be talking about is, is trying to use these kind of pathways and these patterns of co-regulation to get some biological insight. Think of it as making maps of how genes are related to each other. And the first insight is that RNA, unlike proteins, isn't interacting, they're not interacting with each other. What we're looking at is co-regulation. So if you look across samples all the way on the left, it has to do with how these RNA molecules go up and down together. We can measure that, take that data, and identify clusters or modules that are highly co-expressed genes from which we infer but don't know some form of co-regulation. And then from those modules, we find that those modules correspond to basic elements of biology. So shown in this cartoon here, if you take cortex, and we took these, Mike Oldham originally did this in the lab, you take hundreds of microarrays from human cerebral cortex and you ask, what is the major source of variation? It's cell type proportion. And so these modules turn out to correspond to cell types. It's a kind of in silico dissection. And so, one of the critical secondary insights was that gene expression obeys scale-free topology, as does protein, as does kind of all forms of life and every molecular measurement we can make. And so the map looks like the map on the right, an airline map, not a road map. And this gets to the maps for these territories in some ways. Scale-free networks are properties of self-organizing systems, and basically the rules are a small number of nodes have a large number of connections, most nodes have very few connections, and the few highly connected nodes are essential for the integrity of the network. Now, these networks are also, in addition to being scale-free, hierarchical and modular, and very similar to the networks that you heard about, in terms of brain connectivity networks yesterday. Very, very similar kind of graph theoretical constructs underlie both of these kind of maps. So we've used this notion so we can identify hubs, we can identify the modules of kind of co-expressed genes and the driver genes within them. And so one of the first questions we asked was, when and where do disease risk genes act? We had to wait for risk genes to be identified, and in 2012 there were a series of major papers collaboratively assessing the Simons simplex collection, mostly identifying de novo variants. But prior to that, there had been some gene discovery identifying genes at various statistical thresholds. So the question is, where and when over human brain development? We were fortunate to be able to use the BrainSpan data that had been produced as a collaboration between Alan and Sestan and Jim Knowles at USC. And so what Neil Parikshak did is he took from 8 weeks gestation to 3 years of age, took cerebral cortical data from RNA sequencing, and we focused on the cortex not because we were going to only study the cortex, but at that time it was the only data source for which there were other data sources that we could use to replicate the modules that we found in BrainSpan. We wanted to identify things that we could identify in multiple data sets. So we started with the BrainSpan cortical data and created normal neurotypical brain networks. And so basically asked, if we have large effect genes shown on the left with the large arrows that are acting de novo, these are genetic non-inherited mutations like Down syndrome type mutations where it occurs in the germline, isn't present in the somatic cells. That, again, is this 10 to 15% of autism or so. Then you have smaller effect rare variants, and then you have common variation. And at this time, those two were not defined very well. So what you basically do, you have hundreds of associated genetic variants across the genome, and we've organized, Neil has organized the developing brain into co-expression modules. And then he asks, are the genes that cause autism randomly distributed across these modules, or are they enriched in some particular process? And it turned out they were. That if we looked at, they were very enriched in, and I'm going to show you some of that data in a bit, but I wanted just to summarize it. The two major molecular pathways hit were chromatin modification, transcriptional regulation during cortical neurogenesis. And so, you know, the issue is you identify these modules, but you don't know what they are initially. So I'm going to show you some of that. So the, you know, the way to think about it is we had protein disrupting de novo mutations. There were missense mutations, and those two list together. At this time, in the genetics papers, there was no statistical signal for the missense mutations. But what I'm going to show you, again, shows the power of using a network framework, because Neil had identified these modules that were reproducible across multiple data sets and that corresponded to very distinct temporal epochs and biological processes. And rather than being randomly distributed across all of these modules, the autism genes were substantially enriched in M2 and M3 and a little bit in M16. And again, what's interesting is that he was able to see a signal for missense mutations alone. So if you use the statistical power of having de novos and then ask, okay, now if we look around those de novos, is there a signal for missense mutations? The answer is yes. Then if we look in siblings, because there's a very high mutation rate, background mutation rate, and so we all harbor loss of function genes in our genome. And there was a lot of garbage being published at that time where people were finding disease genes that just ended up being rare population-specific variants. We could do the same thing in the siblings, and this is what it looked like. We also did the same thing in intellectual disability. There were 400 Mendelian intellectual disability genes that Neil curated. And those also, they showed a trend in M3, but weren't quite significant. So we're seeing a more specific and stronger enrichment pattern for autism than for intellectual disability. So what do these modules do? I had already mentioned to you, but a little bit. But you know what you can see is that's the first principal component of the. I guess there are two screens here, so I'll try to do it on my screen. This is the first principal component of gene expression in that module called M2, and this is M3. You can see they're expressed very early. Red is birth, and both of them are enriched in transcriptional regulation. When we look at the protein-protein, we want to validate that these actually correspond to protein-protein interactions, and they do quite significantly. And so this implicated early chromatin regulation during neurogenesis and transcription factors, including TBR1, which you heard a lot about yesterday. If we look at the non-de novo risk, this is a much fuzzier gene list. We got it from Simon's website, and these genes have various levels of evidence, and it includes some inherited risk variants, again, much lower evidence. But you can see these genes are also expressed in M13, 16, and 17 during early development, but slightly later, some of them, like M13, but M16 is up actually quite early. And these turn out to be involved in various aspects of neural function, so synaptic transmission at the top, and then homophilic cell adhesion, so you can, again, begin to think about brain development unfolding and these gene expression patterns turning on related to first getting to the right place, sending out processes, and then neural transmission. What was interesting is if we actually, this pattern strongly overlapped, and that's a figure from a previous paper, with neuronal genes that were down-regulated in postmortem autism brain that we had since, subsequent to this, we've validated and replicated in another paper in 2016. So this shows some convergence of these neuronal synaptic kind of risk onto this developmental gene expression pattern that later is borne out in postmortem brain. So in terms of circuits, I rarely actually show this slide, but I thought with this, it's part of the paper. We took data from Jeremy Miller's paper, two Allen Institute papers, one fetal brain on the left, a laminar fetal brain, and the other Amy Bernard from adult primate cortex. And what you can see is that there's an enrichment of these modules in, number one, during human brain development in the kind of cortical plate and subplate, mostly postmitotic cells, but not exclusively. M3 highly enriched in the germinal zones. But when we looked in kind of the later adult, and this is primate cortex, we didn't have adult data at that time, one can see strong enrichment for the superficial layers. And if we looked at intellectual disability genes, they really showed an opposite pattern, an enrichment for layer 5, 6. It's not exclusive. It's about a twofold enrichment. And there's certainly some genes that cause autism that are expressed in layer 5 and 6, as we know. But this just showed a proclivity. And I thought this was super interesting. And this is just statistics to show that this is very different pattern when you compare this to intellectual disability. So we're able to kind of distinguish what might be a little bit more specific about autism than intellectual disability. And one of those things would be cortical-cortical disconnection. If you disrupt layers 2 and 3 and their connectivity, both inter and cross-hemisphere, that would be the prediction of these kinds of data. So going forward, we ask, do these networks do they replicate and have good predictive power? And the answer is yes. This is from several newer papers, including one of ours that's led by Elizabeth Russo and Laura Perez-Cano, that's on bioRxiv. It's been there for several months. And again, we can see M2 and M3 being enriched, not only in autism risk genes, but in genes that were identified in broad developmental disability and in this new iHeart cohort, which is a cohort of multiplex families called the Autism Genetic Resource Exchange. And what's interesting is in that cohort, M16 pops up again, which had the lower effect size variants in it before. It didn't have the de novo mutations. And it turns out that signal is due to a signal from lower effect size inherited mutations that we can pick up in our family cohort that you can't pick up when you're studying a simplex collection that's depleted for inherited variation. And so when we look in that iHeart cohort for the inherited variation, again, there's evidence for a very strong protein-protein interaction network that actually is around previously identified autism genes, including the BAF complex, which is really important in neurogenesis. And I made all those members blue up in the top right. You can see all those BAF complex members. It's totally non-random. It's a huge enrichment of these genes hit. So to begin to study this kind of cell type and laminar specificity in more detail, Damon Pouliadakis and Luis de la Torre-Ubieda engaged in a molecular taxonomy of cell types in human brain, sequencing using DropSeq 40,000 cells, again from 2 lamina, basically separating the germinal zones from postmitotic zones. And Damon's going to talk about this later. But needless to say, it does at least replicate what I've described to you before. So what we found, just to summarize, is that autism risk genes that have been identified to date show a proclivity, not exclusivity, for acting during early brain development, during the first and second trimesters primarily, and include different categories. There's transcriptional regulation, which occurs earlier, and then the genes involved in synaptic development, which are kind of subsequent. What I'm not going to show here is we're also able to identify genes like MEF2C and SATB1 that are transcription factors that are enriched and that are likely regulating these modules. And then in terms of the where, it kind of implicated the cortical-cortical connectivity. But this work really set out to us that we were really, we were really missing something, in that if we identify, and that this molecular architecture fits with a transcriptional regulatory architecture. So if we think of kind of natural selection and the large-scale, high-impact de novo genes that are all the way to the right, that are new mutations that are occurring, and they aren't things that are shared in the population, versus common genetic risk, which is older and shared in the population, one can see that the high-effect mutations are affecting transcriptional regulators, things that are very high drivers of development. And the bottom-tier regulatory targets are more likely to be those involved in synaptic development and function and carry the smaller effect size inherited burden. And this makes total sense, that there's a regulatory hierarchy that really fits with the evolutionary hierarchies. But it really highlights the need, it highlighted to us at the time, the need for refined understanding of regulatory networks during neural cell lineage specification and cortical development. So all these things that have subsequently started, the brain single cell initiative, PsychEncode, are really essential. But it really, this just points out how little we knew and how we really have to make these maps and understand what the parts list is and how they relate to each other and how cell fate is regulated. Many of you in the room have been doing seminal work in this. The work in autism is really pointing out how essential this knowledge is, this very basic knowledge. But how do we move from genetic variants to genes? I told you that most of the risk is polygenic. What I didn't tell you was that most genetic variation, the exome, the protein coding genome, only accounts for about 4% of the genome. And so most genetic variation that predisposes to disease sits in non-coding regions of the genome, actually even outside of gene borders. And so it really raises a critical question. Not only, we need to understand how genes are regulated and what these non-coding regions actually do. And so this is just to remind me to tell you that most genetic variation that predisposes to disease sits outside of coding regions in the orange regions. And so it's essential to have a map. You need this two-dimensional map of chromatin accessibility, histone modification, there's DNase, there's transcription factor binding, evolutionary conservation, etc. And this occurs in a tissue and stage-specific manner. However, what's even more interesting is over the last 5 or 6 years, it's become just very, very clear that gene regulation is not occurring in a linear fashion, that there's a 3- and 4-dimensional structure to chromatin that is critical for gene regulation. So that just because a variant is close to a gene, in no way should that mean that it's actually regulating that gene. And that's what's shown at the bottom. If you look at EQTLs, even outside of the brain, tissue-wide, in other words, genetic variation that affects expression, it's only about 50% on the closest gene. If it's not in the first 10 kb or in the promoter, it's highly likely to be acting on a very distal gene, sometimes 500-600 kb away. And so that's a kind of functional analysis, the EQTL, but you can measure chromatin conformation. You can actually measure these loops using techniques like Hi-C. And so what we started to do a number of years ago is generate a reference map of gene regulation during human neurogenesis based on dynamic chromatin accessibility. So we want to know what genes are expressed using RNA-seq, what regions of the genome are active using chromatin accessibility, ATAC-seq, which is, looks at open chromatin. It's like DNase-1, but uses a transposase that integrates into open regions, and then you sequence them. What genes they regulate. This is Hi-C, which is a technique that we used here, but there are others where you're basically pulling down regions, you cross-link them, regions that are acting, where there's a distal interaction. We can also do transcription factor binding site mapping using the ATAC-seq or DNase data. And there's some references below, and this work was led by Jason and Haejung and Luis de la Torre-Ubieta in the lab, who were an extraordinary collaborative group who each led different parts of this work. What we did is we just separated the cortex. We didn't, these experiments were expensive at the time. there's quite a bit of biology known about post-mitotic neurons versus the mitotic progenitor zones. So we just simply separated them into these two regions, which we call the CP, cortical plate, and GZ, germinal zone. And this is, we're enriching for a progenitor population and post-mitotic cells. And we reason that this will give us some reality check because we should identify known processes that are occurring. And if we don't, there's really a problem. So the first map we made was the high C map. And basically, again, just to remind you that gene regulation, what we're basically doing is finding gene loops or enhancer, promoter, actual physical interactions. Not all of these are going to be functional, but a lot of them are. And again, divided between CP and GZ so we can compare differential looping. And we validated these high C loops with using other orthogonal data. We used EQTLs and showed that the high C data predicted EQTLs and enhancer RNA, mRNA from cage sequencing from the Phantom Consortium. And we're able to show that they were highly related to each other. So this was not, we weren't picking up some biochemical noise, we were picking up things that could predict other functional relationships. Only half of the GWAS loci, when we looked at schizophrenia, which was the highest powered GWAS at this time, are acting on the closest gene. Many were acting more than several 100,000 base pairs away. And so I'm just going to show you what that kind of looks like. You have the genome browser at the top. Then here is the high C interaction map. This is the negative log of the p-value. And this is really significant. It's like 10 to the minus 60. That would be the false discovery rate of 0.01. There's the credible SNPs identified. Most gene regulation occurs within these regions called topologic association domains, which are identified there. And we can see we have a CP enriched locus there. And that turns out, you could see if you map that to the closest gene, you might miss it. It turns out, this looks like it's interacting with that cholinergic receptor promoter. And in fact, when we did this, we were able to annotate the 108 loci that had been identified by the PGC, identifying dozens of new risk genes and implicating new pathways, including cortical neurogenesis, which hadn't been identified before, or acetylcholine receptor activity. And those are just some of the genes identified by this functional analysis of the GWAS hits. We could also show that this was very tissue-specific, and we have a lot of data on that, but that if you looked at the credible SNPs, either identified by PGC or by us using a program called Caviar, and then we compared to different genes predicted by different methods. So we have the closest with high C on the right, or the closest without high C support. And you can see that with high C support, there's a really substantial enrichment in those genes that are dysregulated in postmortem brain. And of course, looking at fetal brain, those that are CP or GZ, we had the most enrichment. And we look at IMR90 cells, which I think is a fibroblast cell. If we looked at those that are just in linkage disequilibrium with the SNP, which is what the PGC, most geneticists would have used at the time, it really shows no enrichment. So there's a ton of biology here, and it really shows that by identifying the genes more accurately, we're able to find things that are likely more disease-related. We're also able to validate several of the loci using CRISPR. This is just showing first, if we just did a luciferase reporter with the risk allele, showing that it reduces the reporter expression, the T is the risk allele. And this is at a locus called FOXG1. This locus was 760 kb away from the credible SNP, but we had evidence from high C that this was likely to be regulating. So we have the SNP over there on the right, the FOXG1 locus all the way on the left with the high C peak below. And again, Luis CRISPR'd out a few of these in Heijong. And when we CRISPR out the region around this enhancer, which is 760 kb away from the gene, it diminishes gene expression, but not of the closest genes. We did a few of the closest genes as well. Well, going forward, Luis reasoned that by adding two orthogonal methods like EQTL or maybe now using open chromatin using ATAC-seq, we would even get stronger maps. So Luis did ATAC-seq in the same samples. And the idea is the same kind of design, CP versus GZ. We identify peaks at transcription start sites, ATAC-seq peaks, and find peaks that had significant correlations across all the samples. So here with ATAC-seq, it's just looking at open chromatin. It's not actually finding the direct binding. So here we're just using correlation. We take the promoter and ask, what within a megabase is it most correlating with? And then we overlay these peaks with the high C peaks. And we subset it to those that are either more associated in GZ or more in CP. So those more associated with progenitor proliferation versus post-mitotic zones. And so if we look at these distal enhancers, they regulate genes that are enriched in the expected cortical lamina, and we can see that here, that the GZ greater than CP peaks are in the germinal zones, and the genes that they're targeting are enriched in these zones if we use the Allen Brain Atlas data, and the same thing with the with the genes enriched in CP. So how can we use this? Of course, we can use this refined map to refine schizophrenia and other disease loci, but we're also very interested in this evolutionary question. So we first looked at human-gained enhancers, which were from Jim Noonan's lab, and they're basically enhancer marks that are quantitatively much stronger during fetal brain development in humans compared with macaque and mouse, as shown here. So these would be active enhancers based on these marks. And so now if we use, in the Noonan paper, they essentially assign them to the closest genes, and they use some non-tissue-specific TAD boundaries, high C, but didn't use gene loops. So it's essentially a closest gene type approach. But so when we reassign these, we can see, again, high expression of the genes targeted by, in the GZ, in progenitors, and then later during gliogenesis, which is really interesting, and then the cortical plate later. I mean, this is a trajectory you'd expect. What's super interesting is if we look at the enrichment of genes that are targeted in the fetal brain lamina from the Miller et al. Paper, we can see, again, this kind of remarkable enrichment, the GZ greater than CP. And if you look all the way to the right, these would be asking enrichment of genes regulated by human-gained enhancers within fetal brain cell types using the polyneural data that generated in Kriegstein lab. We're able to show that if we look at those that are unique to a specific cell type, that the ORG stands out above all others. That's in blue there on the right. If we look at those that are just shared or expressed, we can see it's targeting progenitor populations hugely and not post-mitotic populations. So these human-gained enhancers are acting in progenitor zones, and specifically in the ORG cells, the genes that they're targeting, if we're looking at specificity. And of course, maybe Arnold is going to talk about this, but adenoradioglea are greatly expanded in primates, and there are several labs here who have worked on this and really demonstrated this. So this was remarkable in that we were finding these genetically defined regulatory elements. We're targeting genes expressed in this development, in this human and primate expanded population that is purportedly involved in cortical expansion. So, on that theme, we decided to validate a few of the enhancer targets. This is one that was several hundred kb from the FGFR2, and there's the enhancer on the left, and there's the high C peak. And so, again, this was removing the enhancer in vitro in primary neural progenitors, significantly reduced the FGFR2, and Luis used 7 different guide RNAs, so different deletions of this region overlapping. And it reduced the progenitor number by, not conclusively, but likely by altering progenitor self-renewal. We saw that it increased kind of early differentiation of 2G1 positive cells. So here we find one of these human-gained enhancers is actually acting on a gene known to be involved in progenitor proliferation, and this provides a potential mechanism, one molecular mechanism for cortical expansion. Another utility for these kinds of findings is in mapping disease loci. So there was a disorder that had been described several years before that Luis found in the literature, where patients had microcephaly, polymicrogyria, and agenesis of the corpus callosum. And it was due to a genomic rearrangement. And what we could see, the genomic rearrangement is there in dotted lines. And we could see that 6 predicted enhancers to the EOMS gene would be removed to a different chromosome. And so we reasoned that perhaps one of these enhancers was driving it. So we took the enhancer that overlapped between the high C and ATAC-seq, the highest confidence, and Luis again CRISPR'd that out. And what's remarkable is there's about a 70 to 80% reduction with all 3 constructs, again in these primary neural progenitors. So this kind of demonstrates that there's a severe disorder. It's not disrupting the gene itself, but disrupting the relationship of these distal regulatory elements, which are quite far away, to the gene itself. And this gets to one interesting issue about therapy. You can imagine if you have haploinsufficiency in many genomic disorders where there's an enhancer issue, even in autism where you have one copy of the gene knocked out in many cases with de novo mutations, if you can identify these enhancers, it provides a therapeutic approach whereby you could use CRISPR activation to turn on the gene during the appropriate window of development. Now going back to kind of broader issues. So I've shown you how we can study specific genomic disorders. How do I take GWAS and begin to understand where GWAS is acting? So what Luis did and Jason Stein is we took a whole bunch of GWAS and we partitioned the GWAS across the genome into its where the genetic variants lie. And one could see a huge enrichment in fetal brain peaks and in adult brain peaks for schizophrenia and for educational attainment. And ADHD looked more fetal than adult. Intracranial volume, again, driven by fetal, not surprisingly. But some other disorders like major depression, depressive symptoms, GWAS, also loaded heavily on kind of this neurogenesis, which is really remarkable. So then we looked and asked, is it in the progenitors or is it in the postmitotic neurons, the fetal signal? And when we look, we see that the vast majority of the signal that we're seeing in fetal brain is due to the very early proliferation, let's call it the germinal zones, rather than the cortical plate. So kind of finally and lastly, I've shown you how we've used these biochemical techniques for looking at chromatin structure to begin to divvy up genomic risk. Oh, and one other point I wanted to make is that autism is at this point is a fairly weak GWAS. There's a new autism GWAS, and I'm going to show you some of that in just a little bit as I end. But one of the reasons, the size of the peaks here is really related to the strength and power of the GWAS. But you can see the distribution is really, it's really what's important. So the last thing that we've been working on now is defining a different kind of functional map that, of course, should overlap strongly with these ATAC-seq and high-seq chromatin maps. And that's the expression quantitative trait loci, or EQTL in brain. And this has been driven by our graduate student, Rebecca Walker. So we have over 200 high-quality RNA sequencing and SNP chips from fetal human brain. And we do the RNA sequencing, we do the genotyping, we discover eQTLs and splice QTLs, and then use that to ask, how does that overlap? Where does GWAS partition? There are a lot of questions we can ask here. We can even do something called a transcription-wide association study, or TWAS, which I don't have time to talk about today, but the paper is on the web and you can take a look at it. Not surprisingly, when we look at the fetal E genes, that is, genes that have an EQTL, they are distinct from GTEx, adult cortex EQTL, but there's an overlap, and it's about a 40% overlap or so, which is what's been seen in other fetal and adult tissues. So again, this notion of stage specificity and gene regulation, very critical. What I'm showing here is something very interesting. If we look at something, there's a measure called PLI, which is a measure of the constraint of the gene, the likelihood that a variant, a genetic variant identified that's protein disrupting, is going to disrupt that gene. It's a measure of the constraint. So the genes with high PLI, especially PLI over 0.9, are those that contribute to the autism de novo signal and are those that are protected or have much fewer mutations in human populations than expected by chance. So these are highly constrained genes. And again, not surprisingly, the fetal-specific EQTL are substantially enriched in genes at the very top of the PLI ladder. And again, this fits back to why we're identifying de novo mutations in fetal-expressed genes. So now we can ask what kind of enrichment for schizophrenia, for autism, GWAS are occurring in these EQTL across the genome. And so Rebecca took these mid-gestation cortical samples and ran WGCNA and network analysis and identified modules. And this is showing the relationship, the partitioning of the GWAS into various modules. And there's an enormous and highly significant enrichment in a yellow module, which turns out to be involved in chromatin regulation and transcription factor regulation. Again, a theme that we saw with the rare variants. This is telling us that the common variants are also acting early during this time. And when we look at cell type enrichment, again, using many of the human, our own data as well as the Kriegstein data, we can see that fetal upper layer neurons are enriched in this yellow module. Again, this speaks to the, this previous result from 2013 from Neil Parikshak, where he found this kind of upper layer predominant signal. With rare de novo mutations, this is with common genetic variation. And even more so, when we look again across GWAS enrichment, showing schizophrenia at the top and ADHD at the bottom, we can compare EQTL and splice QTL. And one of the messages I want to leave you with, there's tons of data here, is that schizophrenia is, again, highly significantly enriched in fetal EQTL, not in GTEx EQTL. I'm going to show you that in a second. And there's a trend for, for several others, ADHD a little bit, but not quite significant. Whereas the splicing QTL are even stronger. So if one partitions genetic variation across the genome, one's finding that it has a larger impact on splicing than on gene expression levels. And we can see now, finally, an autism enrichment in fetal splice QTL, as well as strong for educational attainment and schizophrenia, which are highly related to each other. And this is just showing the purple is fetal and the blue is adult. This is showing the schizophrenia EQTL enrichment across different arbitrary windows around the SNP, up to about 5,000. And you can see that there's a really large odds ratio of an enrichment over quite a distance there. In other words, the schizophrenia risk is partitioning more into fetal than in adult. So I'm going to end in 2 minutes with just saying this, that we have methods to efficiently interrogate interchromosomal interactions, open chromatin, and the effects of human allelic variation on gene expression and splicing, and this will help us inform tissue and stage-specific regulatory interactions. When combined with other data, these data are going to provide the maps for understanding the target of disease-related non-coding variation. And as whole genome sequencing is implemented over populations, these maps are going to be very useful. And this can be applied to understand the most vulnerable epochs and cell types across brain disorders. But we can also use this knowledge to elucidate disease mechanisms and elucidate the validity of in vitro systems. And I'm just going to leave you with this kind of teaser slide in a way, just to show. So one of the projects that we've been doing, Jason Stein and Luis developed a method in our lab that actually you can take RNA sequencing from in vivo brain and use that to assess how well am I modeling normal in vivo development in vitro. That's called context. We have a bunch of machine learning tools. And so we've been doing a project with Sergio Pasca, funded by CERM and Convergent Neuroscience. We're taking patients with multiple rare forms of neurodevelopmental disorders that have high risk for autism. And I'm just going to show you some of the data that Aaron Gordon produced here from kind of 3 of these variants that increase risk for autism and partially for schizophrenia as well. So now if we look at the transcriptome overlap with our previously published in vivo postmortem molecular pathology, we can see a highly significant overlap with the 16p11 and the 22q11 with autism, still significant with schizophrenia, but less so. And then if we look at kind of a modular, and this data is from this recent science paper published in February where we compared all the transcriptomes in these diseases, one again can see that if we look at the genes that are upregulated in schizophrenia and autism, they are more likely in the astrocytes, shown in red, whereas those that are downregulated are in neurons. And one of the things that's conspicuously missing here are microglia. But this tells us that at least what we've modeled so far in the dish, and we haven't added microglia, is that these in vitro systems that you heard Paulo Arlada really introduce very elegantly yesterday, that these are going to be really quite powerful in modeling at least some of the early factors that are occurring in disease, because we see this really substantial and non-random overlap with the patterns that we're seeing in vivo postmortem brain. So in conclusion, a fine-grained understanding of gene regulation during human brain development is essential to understanding the functional impact of human genetic variation in the nervous system. That integrative genomic and gene network approaches do provide an organizing principle for understanding molecular phenotypes in the brain and defining points of potential biological convergence in disease. Autism risk genes do converge on early fetal brain development during the peak of neurogenesis, involving transcriptional regulation and synaptic development. They impact glutamatergic neurons, and I would say upper layer, although Matt State's group and Anand did not find, we all converge on glutamatergic neurons, but I think the upper layer finding, especially with the new data that we have, is getting stronger. And this includes risk imparted by common variants, which also implicate the upper layer glutamatergic neurons. And that's really, really new. And so I hope that I've shown you that genomics provides a foundation for integrating and interrogating the relevance of in vitro systems as well. So I have a lot of people to thank. This work, some of the work that I mentioned was led by Neil Parikshak, Mike Gandel, Vivek Swarup, and Grant Belgard. All the people with asterisks are now out of my lab, more out than in, but they've all gone on to a better life outside, although maybe not a better state. I would say California is a top state, but and then Rebecca, Damon, and Chris Hartle are currently in the lab and led the well, you'll hear about Damon's work, but involved in the EQTL study, as well as Gokul Ramaswamy, and Aaron Gordon's been working on the convergent neuroscience. All the genetic sequencing work has been led by Elizabeth Russo and Laura Perez-Cano and Jenny Lowe. Then we have collaborators. Sergio Pasca I mentioned briefly. It's a very exciting collaboration, as well as funding from PsychEncode that's really allowing us to collaboratively create these maps, as well as the BrainSpan Atlas, which again, another collaborative project. So again, my hat's off to collaboration and to shared genomic resources. Thanks for your attention.
So this might be a naive extrapolation, but given your the gene effects in the germinal zone, the migration of neurons and the radioglia, would you expect to see, if you did a genetics-first stratification of patients, an alteration in an MRI structural variation that you could detect?
No.
Okay.
That's saying as a neurologist. I mean, we can, MRI has limited resolution. And we know that in autism, careful assessment of MRIs, there are people who have migration defects. And, if they have some of these mutations that are known to cause migration defects, one can see it. But I would say more often than not, we're not going to see that. We're talking about what are probably very subtle alterations. And I think Morganka and a number of other people talked about that yesterday. Very subtle perturbations in the patterning, not just these major hammer mutations is most of the population contribution. But if you did look at the rare variants, I think you will find an increase in kind of cortical dysplasias and other cortical malformations, absent corpus callosum, developmental abnormalities that are often seen.
Thanks.
Yeah, in one of your slides, it looked like I thought you were showing the upper and the lower zone thicknesses are associated with different intellectual abilities, or just that gene expression in adults or children related to the fetal brain differences?
I'm not sure I definitely wasn't saying that.
Okay.
Yeah,
all right.
so I'll have to explain it
All right.
afterwards.
So when you related, you're looking at.
Maybe I did say it. I didn't
Okay.
know.
All right.
But yeah, what was it?
Right. So most of these are then the genetic differences in autistic children that you're relating back to the gene expression profiles in fetal brain of normal people. Okay.
That's great. I'm just going to briefly introduce session 3 now. So yesterday we heard a number of stimulating talks about the organization, development, and plasticity of mammalian and sauropsid brains. In the final group session at the end, there was a lively discussion about drawing conclusions from one species and applying it to another, with the concern about overgeneralizing the results from a given species. And somebody pointed out and reminded everyone that there are very many different aspects of brain organization, with some being highly conserved across all mammals and across all vertebrates. The basic brain architecture, one could say, is highly conserved. The migration of GABAergic interneurons from the ventral telencephalon is highly, highly conserved. But there are also some features that are very clade-specific and specialized. Carefully distinguishing between these deep ancestral features and the more recently derived features of the brain is part of our task as neuroscientists, and I think one that doesn't generally receive enough attention in our field. So with that said, this session is intentionally looking at those aspects of neurodevelopment that are not broadly shared, but appear to be specific to humans, and in some cases, all primates. Again, we have a great lineup of speakers, starting with Arturo Alvarez-Boya from UCSF, Colette de Haye from University of Lyon, Arnold Kriegstein, also from UCSF, and Barbara Finley from Cornell University, and then the Data Blitz speakers, Aaron Gordon from UCLA, Ana Rita Ribeiro Gomez from University of Lyon, and Stefano Berto from Southwestern. So we'll begin with Arturo Alvarez-Boya from UCSF.
Well, thank you so much for the invitation to this fantastic meeting. It's really been a learning experience, really fantastic talks. And what I was planning to focus today is give you very briefly two very recent updates on the behavior of neural stem cells in the adult brain using the rodent brain as a model, which I think might be informative of what I'm going to focus most of my talk today is in some recent studies that we have done in the lab looking at the human brain. And we've been interested in the problem of adult neurogenesis for many, many years. It's a fascinating problem because of its possible relation to repair, but mostly because it's actually pointing to a totally new level of plasticity, and I'll tell you a little bit more about these. But actually, key to the problem of stem cell biology is how to understand a process that actually occurs over a very protracted period of time. So unlike development, where neurostem cells very rapidly divide and produce progeny in sequence to build up the tissue, after birth, there's a few progenitors that are maintained in most somatic tissues now, including the brain, with our new understanding. And it has been traditionally viewed that these cells actually are maintained by asymmetrical division, maintaining one cell as a pluripotent stem cell, while the progeny either generates through intermediate progenitors or directly the differentiated cells. So key to understand this process was actually the identification of these stem cells. And there's two regions in the adult brain where neurogenesis has been demonstrated. One is the hippocampus in the temporal lobe in humans, and I'll show you more data about that in a little bit. And the other is a very, very extensive area that covers actually most of the lateral walls of the lateral ventricle. And cells born here actually migrate in a very intriguing way a long distance to the olfactory bulb, where they actually integrate constantly and replace neurons throughout life. So this process has been the theme that my lab has studied over most detailed, and I'm going to tell you a few principles about it that I think might be informative in how we think and try to understand what happens in the human brain. So first, in terms of the identification of the stem cells, and this was an initially very surprising result, but now I think it's starting to make a lot of sense, these cells turn out to have astroglial properties and to be highly linked to radial glia. Initially, in songbirds, radial glia are retained, and they were identified as the progenitors of the new neurons. But later on in the studies in the hippocampus and in the subventricular zone, we also found cells with glial characteristics, or glial characteristics of stem cells. Cells that we call B cells. There's actually a B1 cell that touches the ventricle and has a primary cilia and is in contact with cerebrospinal fluid. And there's a very intriguing cell that we're going to study in much more detail that we believe is also part of the progenitor state that we call B2 cell that is not touching the ventricle. But I'm not going to be telling you about that today. In the hippocampus, Bettina Seri also identified a cell that we initially called radial cells or radial astrocytes. And these cells have been renamed, and they're now known by several names, including radial glia-like cells, even though they're not touching the ventricle. They're also called type 1 progenitors. But they're actually fascinating cells because they have a prominent radial process that crosses the granular cell layer, is in contact with many of the mature cells in the dentate gyrus, and then ramifies profusely, this is not shown here, in the inner molecular layer. So these cells generate new neurons also through intermediate progenitors, and also an intermediate progenitors was identified in the subventricular zone as key in the generation of the very, very large numbers of young neurons that are produced every day in a rodent brain that migrate to the olfactory bulb. A young mouse, a young adult mouse produces as many as 10,000 cells every day. So initially, we were all very excited about these cells because we thought this might be, key to brain repair and that they might maintain and retain many properties of early stem cells. So early models in the 90s and early 2000s suggested that these cells were pluripotent and were capable of generating all types of neurons for the central nervous system. But actually, it's become much more clear that, and especially studies in the subventricular zone, where actually there's a direct relationship with the early primordium in the brain, the ventricular zone, that actually neural stem cells in different locations are very different from each other. So I'm going just to show a couple of slides about this because I think this is actually very fundamental. So as I said, the cells collect from a very large area that includes all the way to the cold ends of the lateral ventricle. Actually, as we have learned now, there is also a medial component to this. So the medial wall facing the septum generates neurons that go towards the olfactory bulb and generate specific cell types. They all join in these very large domains where they migrate using chain migration for their movement to the olfactory bulb. So graduate student in the lab, Florian Merkel, was key to unravel these. And what he found by developing new methods to micro-label different domains, different stem cells in different subregions, is that different subregions are specialized for the production of different subtypes of neurons. And by doing this, he discovered that this very anterior tip actually produces 4 novel types of interneurons that were not known before, that are produced in very low numbers, but sit actually very close to the mitral cell layer, and probably play a very, very important role in the regulation. So this actually was key to understand why are these cells coming from so far, and the reason is that they are actually regionally specified. So now to understand what I'm going to show you in humans, this experiment that I can only show you one slide, but it involved a lot of work by Lucho Fuentealba, did barcoding from the embryo to the adult. And one of the main conclusions of this study is that this regional specialization that generates these different subtypes of neurons is already present in the embryo, including at ages very, very low. Look at this clone from E11.5. It's already highly regionally specified, generating only deep granular cells. So the idea that these stem cells are produced in the embryo, feed this area that becomes this germinal zone in the adult with stem cells that are pluripotent is highly incorrect. It actually suggests that these progenitors are highly restricted by their location from very early stages in development. Another very interesting observation about this, and something I had discussed at length with John Rubenstein, is that actually cells that come from pallial regions, from the roof, keep generating neurons in the adult and have a corresponding area in the adult subventricular zone, and they are specialized in the production of these tyrosine hydroxylase cells and a subpopulation of superficial granular cells. And these are GABAergic cells, even though earlier in development, these cells are lineage related to cells that produce excitatory cells for the forebrain. So there's two other major conclusions from this study, and I'm not going to be able to show you any data for that today. But one of them is that the generation of these cells around E15.5 is large, that these cells, after they're generated and specified, they become largely quiescent. So they retain this specificity, and then they don't divide very frequently. And this was very intriguing because we were working on the assumption that these cells were maintained by asymmetrical self-renewal throughout life. So this led to a question of how is it that we are able to maintain in a mouse adult neurogenesis for life. And again, there's no time to show you all the experiments, but this is one of the strategies that Kirsten Overnier used in the lab to label very small numbers of cells in the walls of the lateral ventricle and follow at very short times how the clones emerge in the subventricular zone. And what she found, surprisingly, is that the majority of these clones emerge by the formation of the transit amplifying cells and the disappearing of the mother cell. Then she also found that a smaller proportion of cells actually self-renews in a symmetrical manner. So the model that these cells are continuously maintained throughout life by asymmetrical self-renewal is wrong. And actually her work strongly indicates that most of these cells, about 80% of these stem cells, divide symmetrically and are consumed. A smaller proportion of them divide symmetrically and regenerate another B1 cells. And one of the very interesting questions is whether these cells are exactly equivalent. We've been able to follow a second round of cell renewal, but we do not believe that the cell renewal model is eternal in the way that it maintains for the life of the animal the population of stem cells intact. So when you look at the overall population of stem cells, there's actually a very high, rapid decrease in their number, and it is mainly due to the fact that the majority of these cells are consumed by the symmetrical division and the generation of transient amplifying cells. A smaller proportion self-renews and feeds back to the population of stem cells. As I said, we do not know if these cells are exactly equivalent to the mother cells or if there is something special about those cells that are derived from the embryo. So that's one of the things I wanted to tell you about the stem cells. There's two things I think to remember. One of them, cells are very regionally specified from very early stages in development. And the second is that these cells are not self-renewing continuously to maintain the population in the same state that it was in early stages of development, but the cells actually decrease with age. So this is actually much more recent, but years ago, through the effort of pathologists and neurosurgeons, we began getting in the lab human tissue. And I'm not going to review all of the early work, but it led to the identification of the rostral migratory stream in humans, in children. But then the surprising finding that in humans, unlike rodents, this process does not continue into adulthood, and even by 2 years of age, it's already highly broken, and the presence of these young neurons in later brains is extremely rare. There's still a few new neurons that you can identify, and I'm going to show you examples of these later on in the walls of the lateral ventricles. We do not know if those single cells are able to make the trip all the way from the subventricular zone, now a very long distance, all the way to the human olfactory bulb. But there's a few of these cells there. But what was very striking, and this was initiation of a number of studies that came later, is that Nader-Sanaid actually identified that in children, in infants, in addition to the migration to the olfactory bulb, here illustrated in blue, there is an additional migration to the medial prefrontal cortex that actually appears to deviate from that same trunk, that same rostral migratory stream in the ventral forebrain in humans. So how does the subventricular zone, where the stem cells are, looks in human? And I think this is highly informative. It looks very, very different to how it looks in mouse. So this is an 18-month-old children, and you can see that the subventricular zone here with the marker for young neurons, double cortin, is highly depleted, and the area where you find the young neurons early on becomes a gap area. And then separate to that, there is a region of high density of astrocytes Some of these cells still divide in the adult, very infrequently compared to the mouse. And some of those cells we've been able to isolate and have been made them to proliferate in vitro and make neurospheres. But this is a rare phenomenon, nothing like you would see in mouse. So occasionally, and I'm going to show you one of those examples, you see one or two of these double corn positive cells here in the subventricular zone, but nothing like that massive chain migration that you see in the mouse. But looking at this pattern of development, there's something very, very abundant and robust happening in these very early stages of infancy. And this attracted the attention of Mercedes Paredes, a neurologist that did a postdoc in the lab. And together with a group of collaborators at UCSF, we began collecting brains from children, and she found an amazing, very, very large and extensive migration in very young children. And this goes on very, very robustly up to 3 months of age, but still you can find some neurons going to the rostral pole of the brain, even in children of 1 year of age. So these cells come in several tiers. There's one tier that happens right next to the ventricular zone, where cells are mostly oriented tangentially, and I'm not going to show you all the data, but she plotted, made a vector for each one of these migrating cells, and wherever she could distinguish a leading process, put an arrow for that cell, and with that we infer the directionality of migration in these different tiers. Then there's very large collections of these cells around blood vessels in the subventricular zone, many, many cells migrating along blood vessels. Then there is another tier where the cells become very dispersed. Here it's very difficult to tell directionality, and we see that cells are oriented in many, many directions. They're not only oriented radially here. And then cells accumulate in these triangular kind of shaped structures, leading to many structures in the prefrontal cortex. And in particularly, Mercedes paid particular attention to this area of cortex where she quantified, and she saw that the migration of these cells into this part of cortex actually correlated with the appearance of multiple subtypes of interneurons. So we believe that this is a late migration of multiple subtypes of interneurons, likely arising from early stages in development where the ganglionic elements are incredibly active in the human brain. But this process, too, actually comes to an end. And by 6 months of age, there's very few cells, and as I told you before, by 1 year of age, there's maybe 1 or 2 cells that you still find with migratory morphology close to cortex, and in adults, we don't find any evidence that this activity of migration and recruitment of neurons continues. So we decided to turn our attention, and this is Mercedes Paredes, who was responsible for all the studies, together with Sean Sorrell, to look in the hippocampus, where there has been several studies suggesting that adult neurogenesis in humans continues robustly. And this was done in collaboration with a group of both neurosurgeons and pathologists, and with Arnold Krickstein at UCSF, and with Jose Manuel Garcia Verdugo, who collected very, very useful brains in Valencia, and also with Shengan Yan in Shanghai, in Fudan University, who also provided 3 key brains to this study. So we began at fetal stages, and again, there's no time to show you all of the data here, but we found, as Joseph Altman had described many years ago, the classical neuroepithelium that gives rise to the dentate gyrus, with migrating cells connecting it to the dentate gyrus. And the dentate gyrus in humans appears to be built sequentially. There's an area where there's a lot of addition. This is 14 weeks of gestation. This is 22 weeks of gestation. This is Ki67 SOX2, which is not a perfect marker, but a good marker for progenitor cells. And you can see that it's very high activity of progenitor cells. Many of them are dividing at this stage, adding cells to the dentate gyrus. The dentate gyrus grows like a worm and becomes actually very extensive in humans and becomes convoluted, that differs from the rodent. And then there's a population of hilar cells, and these cells continue generating cells in these early developmental stages and adding it to the granular cell layer. By birth, there's still many areas of the dentate gyrus that contain many young neurons here stained by double corneum, PSA and CAM, and these cells retain the morphology and the marker expression of young neurons, consistent with what has been seen in the rodent during development and in the adult. However, even at birth, this is not a continuous phenomenon. If you look longitudinally within the dentate gyrus, this comes in patches, and this is one of these hotspots patches where you see young neurons being recruited to the dentate gyrus. But if you look at different stages, by 1 year of age, these patches have become more isolated, but you still can find them there. there's clearly young neurons being recruited at this 1 year of age. By 7 years of age, these cells become a lot rarer, and there's about 3 to 4 of these cells per section. And by 13 years of age, we on average find 1 or 2 of these cells in every 2 sections. So we then looked at a large population, a large collection of samples from adult individuals, and there we cannot find any cells that have this morphology or marker expression. There's many PSN-CAM labeled cells. None of these cells, I think, are related to the process of neurogenesis. And then there's these round cells that we call HAT cells that are double cortin positives, but these cells are not unique to the dentate gyros. They're found in other areas, and these cells have been repeatedly counted as possible young neurons in several studies. So one very interesting thing is that by 13 years of age, there's still examples of cells that are being recruited there. And it would be very interesting to know when these cells were born. But one very interesting observation is that you look in detail into these cells and you can see how well preserved they are. These cells already begin to have clear characteristics of much more mature cells. So you can find an axon coming out of them, and you can have dendritic branching in them. So we think that these cells have been there for a while, but we do not know exactly for how long and where these cells were exactly born. So this is the evidence for young neurons in the dentate gyros that was really not consistent with what people had reported before. So then we went back and began looking at not only young neurons, and this is the pattern of young neuron developing and mapping that I had just told you in a more graphical map, but look at the progenitor cells. And again, we used, this is the quantification for the young neurons. We actually went and looked for Ki67 SOX2 cells later on in development. And there's something very striking happening here. One of the things is that you find in early stages in fetal development, as I was telling, a large population of cells that are proliferation are SOX2 positive, but you find these cells all along even into adulthood, but they're not unique to the dentate gyros. They're all over the brain, and I'll show you some maps of where they are in a second. But what you can see is that unlike species where neurogenesis continues robustly, you never see an accumulation of these cells in the so-called subgranular zone, that is the germinal niche where neurogenesis continues in the mouse. You also do not see an accumulation of blood vessels forming the base of the niche as it has been seen in the mouse. So even if we looked at all proliferating cells, not say perhaps in adult humans, the cells do not express SOX2 anymore, and SOX2 is not even a specific marker of stem cells because it's expressed in many astrocytes. Even if you look at proliferation, you can see that there's never a coalescence of a proliferative layer next to the of the dentate gyros in humans. And the proliferation is there. There's continuous proliferation even in adults, but there's nothing unique about the dentate gyros. There is no accumulation of these cells close to these areas where you see the young neurons. So perhaps some could say that these cells in here in the hylas are the progenitors in adult humans. But if that was the case, then we should find many examples of cell migrating radially towards the dentate gyros, connecting these progenitor cells to putative new neurons in this area. This is very different to what you see in the rodent, where there is a clear subventricular zone, and if you do a mapping of Ki67, there's an accumulation of cells right next to the dentate gyros where neurogenesis happens. So, as a summary, what we find is that in infants, there is a continued addition and recruitment of new neurons in the human. It's already patchy, as I said. In adults, we don't find it. There was no time, but we have also looked at monkeys. In monkeys, we do see a continuous production of neurons in early stages of life, but this also becomes highly patchy, and in old monkeys, very rare to have young neurons integrated into the dentate gyros. So this obviously, and as probably many of you have heard, has become very controversial, and some labs claim that they find robust evidence, even 700 neurons per day produced in the adult human brain. If that was the case, I think we would have seen it. But one criticism that has been raised repeatedly is that perhaps in our samples, because of the postmortem interval, perhaps of the pre-mortem condition of the patient, the neurogenesis was suppressed, or because of the postmortem interval, perhaps the tissue was not able to preserve the antigenicity for double cortin and PSN-CAM, and therefore we failed to see these new neurons. So I don't think that this is the case, because this is an example of one of the brains that was collected in China, beautifully collected brain only with 5 hours post-mortem interval. We don't find any cell that has the clear characteristics of a young neuron looking through rostral to caudal hippocampus. We found some of these round hat-like cells, and as you can see, those cells are not unique to the dentate gyrus. They're all over the place, and we believe that many of those cells are microglia or oligodendrocyte progenitors. But in the same brain, if you look at the walls of the lateral ventricle, where you sometimes see some migrating cells, you do find clear examples of cells stained with double cortin and having the characteristics of young migrating cells. So double cortin is preserved. So I don't think that the lack of antigenicity seen in the dentate gyrus is because these patients have some condition post-mortem that result in degradation of that antigen. But this is even more evident in what I'm going to show you in the last part of this study, because there are regions of the adult human brain that contain abundant number of double cortin positive cells, and these regions are incredibly interesting. So there's 2 regions that have caught our attention, and one of them is the so-called paralaminar nuclei next to the amygdala. And I'm going to tell you a lot about these. And the other region where you find abundant number of double cortin positive cells in the human brain is the entorhinal cortex, an area that Larry talked about yesterday as one of the hubs for connectivity in the brain. So we haven't done much with these cells yet, though they are abundant and they're very interesting. But we have studied now these cells in detail, and I'm going to tell you what we believe these cells are. So again, many of these samples, and I can show you in a slide if there's time later, many of these samples from where we did this staining were the very same samples where we could not find clear cells with this morphology and this marker expression in the dentate gyrus. So the paralaminar nuclei are very intriguing nuclei. They have been claimed to be prominent in primates, but cannot be found in rodents. They receive afferents from the hippocampus, from the uncus of the hippocampus, and they have efferents both to the amygdala nuclei, especially to basolateral amygdala, and to entorhinal cortex. Their function remains unknown. And this is a 3-dimensional map that Sean Sorrell has done in the lab. This is the basolateral amygdala. These groups of, in purple, are these granular cells that we call the paralaminar nuclei. And there's groups in the lateral area of the amygdala we call the lateral paralaminar. And then there's a very robust and large nuclei that we call the medial paralaminar. And we have mapped these separately. So I'm going to tell you briefly about the embryology of these. We have looked at their development, and initially we thought that these might be equivalent to the intercalated nuclei in the amygdala or GABAergic nuclei. So we were very excited initially that this paralaminar nuclei develops right next to the caudal ganglionic eminence. That's a big source of interneurons during development. It's one of the hottest spots of proliferation. And it has a tongue that projects right next to the lateral ventricle, right next where the paralaminar nuclei develops. So here's the basolateral amygdala, here is the PL, this is the medial PL, and this is the lateral PL. So it looked like these cells might be related, and more so, both of these express high levels of CUF-TF2. So initially we were excited about thinking that these 2 regions might be related because of their proximity and because of the CUF-TF2 expression. But then we showed this data to John Rubenstein, and he told us, be careful. And he actually suggested that we looked at TBR1. And it turned out that unlike the CG, these cells are actually highly expressing for TBR1. And actually, unlike the CGE, they do not express SP8. So they're a distinct group of cells. And now, again, there's no time to show you the data. We have found that later on, many of these cells convert into excitatory cells. So they develop next to the CG, but they don't seem to be derived in terms of marker expression for the CG. So what is intriguing about these nuclei is that if you see at birth, they're completely full with double cortin positive cells. It's a little pool of immature cells. And as development progresses, the number of these cells decreases, both with DCX and with PSA and CAM, and the number of cells expressing mature neuronal markers increases. And the time course of the formation of new neurons actually differs between the medial and the lateral paralaminar nuclei. And this is particularly accelerated during puberty. So TCX cells decrease in number, new and positive cells increase in number. The number of cells within the nuclei decreases also, especially during the first 2 years of life. So what is intriguing is that these cells have many of the characteristics of young neurons, including migratory morphologies. Not all of these cells have migratory morphologies, but a significant number of these cells retain migratory morphology, what looks like a living process and an elongated nuclei. And some of these cells with migratory morphology are retained even in patients at 77 years of age. So was it possible that perhaps there was a source of these cells outside of the paralaminar nuclei, and they kept feeding into the paralaminar nuclei, just like in the olfactory bulb in the mouse? So we decided to map, as we had done before from the frontal lobe, the position and the direction of all of these putative migrating cells. And this is actually a dense slide, but this shows the paralaminar nuclei again in purple, the lateral ventricle, and each one of these arrows indicates the presence of one of these young neurons. And the direction of the arrow indicates the direction of the living process. And one of the new things here is that the amygdala, like the frontal cortex, is not done before birth, continues with many migrating cells postnatally throughout the basolateral amygdala. It's not unique to the paralaminar nuclei. But even by 6 years of age, you can see that the cells have become highly restricted. The cells that have this migratory morphology are only in these regions where the PL are. And when you look, there is no connection both in sagittal sections or in frontal sections, suggesting that there is a migratory stream feeding these areas with young neurons throughout life. So we believe that this is resident cells that are there since birth. They retain migratory morphology. They might move a little bit around this area. We don't know that. But these are immature cells that are retained within this area throughout life. The other thing that we have looked is for the presence of possible progenitors close to these nuclei. So it is possible that perhaps there is some progenitor cells that have separated from the early ventricular or subventricular zone, have been seeding this area just like the subgranular zone of the dentate gyrus, and continue to generate cells throughout life. And you do find proliferating cells here labeled by Ki67, and some of these cells are labeled with SOX2, and these cells are sometimes very closely associated or in close proximity to some of these cells with migratory morphology, but they're not unique to the paralaminar nuclei. One of the first things to observe is that these cells decrease in number very rapidly within the first year of age, and then they're maintained at relatively constant numbers, and it's impossible to know whether some of these cells might in fact be progenitor cells, but their number and density is exactly the same or very, very similar to that you find in the adjacent basolateral amygdala, where we don't see the young neurons. So we believe that these SOX2 Ki67 cells are likely astrocytes that divide at a very slow rate, or perhaps oligoprogenitors, but it is unlikely that these cells are actually progenitors of the new neurons, though we cannot be absolutely sure in the absence of a lineage tracing experiment that that's not the case. So as a conclusion, we find no evidence of robust neurogenesis at adult human hippocampus. If it happens, it's a very rare phenomena. As you saw, there was a few cells at 13 years of age. I wouldn't be surprised that we might find some at 20 or 25 or 30, but this is the tail end, I think, of a process that's happening at higher rates during infancy and early childhood. But we do find evidence of many mature neurons in other parts of the brain, specifically in the ventral amygdala, in these paralaminar nuclei, and in the entorhinal cortex, a region that we have not studied in detail. We believe that these cells are immature cells that are maturing very, very slowly throughout life, and perhaps replacing the process of adult neurogenesis by having a pool of immature cells for plasticity reasons. So compared to the mouse, where we find very abundant neural stem cells, these cells are regionally specified, and I want to say just one word about these. These cells do not divide symmetrically, but actually are mostly consumed by symmetrical divisions, and some of them self-renew by symmetrical divisions. And there was no time to tell you, but there is now plenty of evidence, both from our lab and other labs, that neuronal replacement is not a repair mechanism, but it's actually a process related to plasticity and to the maturation of the cells. In humans, we do find a population of neural stem cells that can proliferate and can be isolated in vitro, but there's very few young migrating neurons, and they're very rare in the subventricular zone. They're almost non-existent, probably in the hippocampus. And there is evidence for recruitment of new neurons in infants, both frontal lobe and hippocampus, but not in adults. So what I want to leave you with is the idea that if you take into account how highly specified cells are from embryonic stages, how tiny the embryonic regions are at the stages that this specification happens, and how much the human brain grows afterwards, possibly one limitation why it's so difficult to maintain adult neurogenesis in larger brains is that there is a dislodge between the early specification of progenitor zone and possible regions where these cells could migrate. So that could be one reason. The other is that if you maintain the progenitors close to the walls of the lateral ventricle without distributing to locations, the migration becomes almost an impossible task because these cells would have to cross incredible vast regions of white matter. So the early specification, the way in which cells are maintained, the longevity and the size of the brain in humans actually suggests that there might be severe limitations to maintain this process. So with that, I'd like just to thank again all the collaborators at UCSF and outside of UCSF and the postdocs that did this work. Thank you so much.
Arturo, so looking at that schematic where you compared the mouse and the monkey and human, it looks like it's not necessarily that the adult, the human species is different in that it loses its hippocampal neurogenesis. It could be that it's simply a matter of time, that a young monkey is still just as old as your adult mouse, but even a young human is older than that infant monkey. So do you have any do you have any data on comparisons strictly by absolute time elapsed and what is the rate of neurogenesis in the different species over time?
Well, we cannot be sure, and I don't think anyone can be sure of the rate of neurogenesis in humans because our inference of neurogenesis is based on the presence of these immature cells, young neurons. We believe that they represent an excess of the process because this period during which a neuron remains immature in humans is very, very prolonged.
Right, but you could even look at just absolute numbers of neurons labeled with DCX at
Yes.
At the different ages, regardless at chronological age, regardless of. No, it would be the opposite. At biological age, regardless of chronological age. So 1 year, 2 years, 5 years, 10 years, regardless of whether that for that species means an infant or an adolescent or an adult.
But how would you tell the chronological age for the dentageros? It looks to me like different areas in the human brain have different time course of maturation.
Oh, it's possible. So I would just take absolute time points after birth. Birth does seem as a good point to a good equalizer for all species, as far as we can tell. Okay, you guys have to remember that when I say across species, I mean every single mammalian species. It's not just, okay,
But I think the data is there. We cannot see, there might be ways to reconstruct, and that's something we're trying to do, a whole dentageros from front to back to see if we find rare examples of double corn positive cells that have the morphology of young neurons in an 18-year-old, in a 35-year-old, in a 45-year-old. So for that, we need complete samples of the whole hippocampus and reconstructions. But we believe that they're going to be very rare, these cells. So I think part of the answer is already there. By adult stages, these cells are non-existent, or if they're there, they're very rare.
The amygdala and the prefrontal cortex results are quite interesting. Did you tell us in both cases whether you think those neurons are coming from the ependymal layer?
We don't know where those cells are coming from. There looks to be a massive tangential migration next to the ependymal zone in that area that becomes a gap area later. That looks like a super highway for migration in humans. And that area is full of young neurons at multiple rostral call levels. I just showed you the rostral part of the forebrain, but there's also cells farther back. We believe that many of these cells are probably coming from caudal ganglionic eminence. It's one region that remains very highly proliferative, even just at stages before birth. There's still many proliferating cells in the CG in humans, and some in the MG also, though those are rare. But we do not know. I mean, it's impossible to know what was your marker expression suggests that they have interneuron marker transcription factors while they're migrating.
So, and just quickly, I always thought the carbon dating experiments were a real tour de force. Why did you think they got the hippocampus wrong?
Well, I mean, it's a very creative idea. The problem is that carbon can be incorporated into a lot of things, not like thymidine that's only incorporated into DNA. So it has been claimed in discussion with Jonas that it cannot be, for example, methylation, because if you were to take all of the methyl groups, that would not be enough signal to see the few cases where they see evidence of adult neurogenesis. But if you look carefully at that Spalding paper, about half of the samples that they present are consistent with the idea that there's no continuous neurogenesis. They say that's highly variable, and they obviously pay attention to the ones that have more neurogenesis. Now, the problem with the methylation issue is that, as now many people have shown, when methyl groups get changed, there's also replacement of whole bases, so there might be many more carbons that are replaced. And there's something going on in the hippocampus that's very interesting, where there's increased SNPs, increased mutation rates in the dentate gyros that might relate to higher levels of repair that might also result in incorporation of carbon. So there might be other reasons what carbon might get incorporated into DNA that's not exactly related to the presence of young neurons.
There's some evidence I'm aware of from crustaceans that the new neurons incorporated in adulthood come from the vasculature. And I don't know if, I mean, I'm outside this field, so I don't know how to relate it, but is that just a mechanism that could never exist here, or is there something about the vascularization of the zones that give rise to new neurons in rodents that suggests that could be a source?
I have never heard of the vasculature as a source, but over the years in this crazy field that we are, the stem cell field, there has been all kinds of claims, like the hematopoietic system being able to give rise to neurons in the olfactory bulb. Over the years, I think it had become clear that all of this is part of the normal lineage from the ventricular zone, subventricular zone into the adult. There was no time to show you the data, but it's not only hypothetical, the link between the embryonic studies showing that the stem cells are radioglia, but there is direct lineage tracing of radioglia from embryonic stages to adult B1 cells. So I think there is very strong evidence there that that's the source of those stem cells.
Thank you.
Thank you so much. We have to move on. The next speaker is Colette Dehaye from University of Lyon.
I was still in the wrong computer.
So, good morning, everybody. I would like to start by warmly thank the organizers to inviting me to this meeting. And as Dan put it wonderfully, it's both an honor and a privilege. So I am from Lyon. It's a lovely city in the south of France. And our institute, the Stem Cell and Brain Research Institute, is mostly working on non-human primate. So today, what I'm going to do is to provide you an overview of published work which is related to the generation of cortical neurons. And in the second part of the talk, I will switch to unpublished results related to radial migration in the non-human primate cortex. So to start, and on this panel is just to remind you that there is, across these mammalian species, there is an increase in the tangential dimension of the cortex, increase in its surface area. Here you have the mouse, the cat, the macaque, which is animal species we are working on, and the human. So you can see that the macaque has a well-folded brain, and as Susanna demonstrated us yesterday, the human brain, in terms of neuron number, is linearly scale up of the primate brain. Besides this tangential expansion, there is also a radial expansion, which is to say an expansion in the thickness of the cortex. And this is shown on this cartoon here. And you can see that compared to rodent and carnivores, primates have enlarged supragranular layers, superficial layers compartment. And this is reaching its maximum here in humans. So supragranular layers are layer 4, layer 4 is a granular layer, and supragranular layers are layer 3 and 2. So this selective enlargement of supragranular layers in the primate is considered to contribute to the increased computational abilities, because these neurons from superficial layers are making the cortical network. And we have been interested for many years now in the developmental mechanism which are responsible for this increase in the supragranular layer compartment in the primate brain. And we have argued that one key feature of cortical development in primate is the outer subventricular zone. So back in 2002, we described the outer subventricular zone, which is a specialized germinal zone which is expanding in the primate brain. So just to compare mouse here and the monkey, so at comparable developmental stages throughout corticogenesis. So in the mouse you have two major germinal zones, the VZ here at the apical border and the SVZ, which is secondary ventricular zone here. In the primate, you see that the SVZ is increasing dramatically in size, and is later subdivided into inner SVZ and outer SVZ. And this pool of progenitors from the OSVZ correspond to the basal pool of progenitors. And you can see that I indicate on my cartoons like the time of generation of the infragranular layer, deep layer in the cortex versus the supragranular layers, superficial layers. And so you can see that at the time of the supragranular layer generation, the VZ is getting really small and reduced, and the OSVZ is a predominant germinal compartment. And so we have shown that this OSVZ was responsible for the generation of supragranular layer neurons in 2005. So you can see here a section with S-phase cells in the OSVZ at the time of generation of the superficial layer of the cortex. And we also noticed that the OSVZ included some progenitors with a basal and apical process. And more recently, we further investigated this issue of the progenitor types in the OSVZ. So what we are doing, we're just working on embryonic organotypic slices from the occipital cortex in the macaque brain. So this is the back of the brain, area 17, primary visual area, and surrounded by area 18. So we take this organotypic slice culture into a 2-photon time-lapse video microscope, and we can image the division, differentiation, migration for up to 2 weeks on this ex vivo assay. And at the end of the session, we do some immunolabeling to identify the cell we have been imaging. So what we have found is that there was a high morphological diversity in the OSVZ progenitors. And so in 2010, Arnold Kriegstein's lab and Wieland Hüttner's lab reported the the occurrence of basal radial glial cell in the OSVZ of the human fetal brain. So they mostly reported two types, the BRGs with a highly developed basal process attached to the basal lamina and the intermediate progenitor. So using unsampled, unbiased sampling method and combined with the time-lapse imaging, we then observed that in fact there was higher diversity, and on top of these two types we had three other morphotypes. So we had we found the IPs, we found some basal, sorry I can't see. We found some, these two types which were already described. We found some basal radial glia which have a process going up basally but not attached to the basal membrane. We had some bipolar BRG with both a basal and an apical process. We have BRGs with an apical process, and we have transient BRG which would have either an apical or basal process at a time during their lifetime. So when I mean these morphotypes are constant between during the lifetime of the progenitor. It is constant between two divisions. And each of these morphotypes is characterized by a proliferative signature, which is related to his self-renewal abilities, his neurogenic capacities, how much progeny is going to generate, and also some cell biology feature. And we did also some unsupervised clustering analysis based on machine learning, where we consider all these parameters without any a priori, and this returned at least 5 BRG categories in the OSVZ progenitor population. So when we do our imaging, we found that these the progenitor of the OSVZ have extensive proliferative abilities, and for instance, this is just one example of a clonal analysis, where you can see a progenitor dividing and showing these different morphologies. And here we have like multiple rounds of division, up to 6. So when you have many lineage trees like this, it's very difficult to extract the relevant information. So we implemented the statistical analysis, which is a state transition analysis, where we pull all our clones together. And these state transition analyses are made of nodes and edges. I'm not going into the detail, this is published. But what it shows is that the topology of the lineage relationship between these progenitors is different during the generation of infragranular neurons compared to supragranular layer neurons. And this topology appears to be more complex. One unexpected feature that we found is that we observe bidirectional transition between the different morphotypes. For instance, these bipolar cells here can divide and give rise to an apical process bearing cell, which in some instance can give rise to the BRG with both process. So again, this is at the level of the population, and such bidirectional transition have never been reported in the mouse, and also we have been looking at hundreds of lineage trees in the mouse, and we never observed them. So we think that this bidirectional transition introduces a certain level of stochasticity, which might be necessary to specify the large variety of cortical neurons in the primate brain. So if I summarize what I've been telling you so far, so if we start here with the mouse, so to produce the mouse cortex, which is shown here, we have a VZ and a SVZ, and we have hierarchical lineage with a radial glia giving rise to an intermediate progenitor giving rise to a neuron. Situation is quite different in the macaque brain with its enlarged supragranular layers. Here we have expanded OSVZ with at least 5 identified morphotypes of progenitors and with complex lineage relationship. We also showed that there was a very different regulation of cell cycle throughout corticogenesis in the primate compared to the mouse. So in the mouse, there is a progressive increase in cell cycle duration throughout corticogenesis, whereas in the primate, there is an increase in cell cycle duration, and then a decrease during the generation of the supragranular layers. And this is parallel by an increase and then a decrease in what we call cell cycle exit fraction. It means that when cells are proliferating fast, they are mostly doing proliferative symmetric division, and therefore they differentiate less. So this reduction in the cell cycle duration corresponds to an expansion of the progenitor pool of the supragranular layer neurons. And so we have been able to show that this reduction in cell cycle duration was due to a reduction in the G1 phase duration and also to a lesser extent to S phase duration, which led us think that there might be a higher complexity in cell cycle regulation in primates than in rodents. And indeed, this is published work as well. We have been joining forces with Ken Kozik at University in Santa Barbara at USC SB. And with Ken and Maria Arcila, a PhD student at the time, we have shown that there was a primate-specific microRNA in the germinal zone of the primate, and these primate-specific microRNA target mostly cell cycle regulators, in particular the G1S regulation. So, this primate-specific microRNA correspond to like an additional module of cell cycle control in the germinal zone of the primate. And so this, what I've been telling you with this reduction of cell cycle duration, which correspond to an amplification of the precursor pool, will ultimately give rise to an increase, be responsible for an increased production of the supragranular layer neurons. So then the question we asked next was whether this increased production of supragranular neurons would be paralleled by lamina specific rates of radial migration. And also we asked the question for area-specific rates of radial migration because, for instance, as you might know, there are clear cytoarchitectonic differences between different functional areas. And for instance, area 17, primary visual areas, has twice as many supragranular layer neurons in an arbitrary unit of column of the cortex compared to the adjacent area, area 18. So we looked both at these issues using the same methodology. We just imaged organotypic slices from the occipital cortex of embryonic primate. We infect the progenitors with retrovirus, GFP retrovirus, and then we look at the migration of the progeny of the infected progenitors. And this is what it looks like after like 12 days of survival. You have like migrating neurons going through the different compartments, and from their place of birth in the germinal zone, they cross the subplate to reach the cortical plate. And what I'm going to tell you now is what's the observation we have been making in the subplate. So first, what we observed is that a well-known phenomena in the mouse, well described, which is the fact that migrating neurons, that early post-mitotic neurons, go through a multipolar stage before initiated radial migration, and this has been so documented by many teams, is not found in the primate. So here is again a slice of what we image, and this is a proportion of multipolar early post-mitotic neuron, is the OSVZ of the primate, and we have like less than 10% of multipolar neurons in the primate, as compared to more than 80-90% in the mouse. And what we also find that the multipolar stage is not dispensable for proper radial migration, and it's therefore not evolutionary conserved. But then where we can say like where the primate brain diverge is that you will find in the literature numerous papers addressing this question of the multipolar to bipolar transition in the mouse and arguing that this is crucial to understand this phenomenon with respect to some neurodevelopmental disorder, whether it be autism, epilepsy, or whatsoever. So we have always to be very careful when we want to extrapolate data from the mouse to primate. And I think this is a very good example. So the next question is that I told you that the primate is characterized by the higher rates of neural production higher rate of supragranular layer neuron production. And so we looked at the migration rates for infragranular layers and supragranular layers. And this is just here an example of the tracks we manually reconstruct with the movies of the two-photon microscope. And again, I will show you what's going on here in the subplate. So we have been measuring the motility index of the infra and supragranular layer neurons, and we found that the motility index is migration speed minus the poses, so the intrinsic motility capacity of the neurons. So we find that there is an increased speed of migration in supragranular layer neurons compared to the infra neurons. And so this results in increased radial displacement by unit of time. So from this we can conclude that the supragranular layer neurons exhibit enhanced radial migration efficiency in the subplate compared to the infra neurons. In the next step, we looked at the trajectories of the migrating neurons, so in both areas, area 17 and area 18. So when we look at the trajectory of the infragranular layers, we found that the trajectory was almost strictly radial in both areas. By contrast, when we compared area 17 and area 18, we observed significant areal differences in the trajectory. You can see here that in area 17, the trajectory are radial, while in area 18, they are not radial, and they are meandering and a bit tortuous. And this is we compute the radiality, what we call the radiality index, which is a ratio between the radial distance measured with respect to the actual length of the trajectory, which is taken by the neuron. So from this, we can see that area 17 has a radial trajectory by contrast with area 18, which shows what we call abradial trajectories. So it has been shown by many teams that the interaction between the migrating neurons and the radial scaffold is important for proper radial migration. So then we wonder, maybe there is something different in the area 17 scaffold, or maybe there is something wrong with our slices. So we looked at this issue, and so these are immunostaining with vimentin, which is labeling the radial glia scaffold. And what this shows is that the area 18 radial glia scaffold is maintained, is preserved in the organotypic slices. I must also underline that we are doing this observation in prior to gyrification. And so these areal differences in trajectory do not appear to be linked in differences between the radial glia scaffold, which led us think that they would be at least partly cell autonomous. So having said that, we used an in vitro model to explore radial migration. We prepared neurospheres out of area 18 and area 17, and then what we found that when we look at the trajectory of neurons which would differentiate and migrate out of the neurospheres, we found like a radial trajectory from a post-mitotic neuron in 17 compared to a tortuous meandering trajectory in area 18. And this is illustrated here. This is a reconstruction of the tracks. and also the computation of the radiality index. So this points to the radial and abradial migration trajectory being determined in a cell-autonomous manner. So then we wanted to try to understand what was underlying these differences in migration trajectories. And so we turned to look at what was going on in the leading process of the cells. So the leading process is tipped by a structure which is similar to the growth cone, which exists at the top of migrating axon. And the growth cones is considered to be important for sensing the environment. So we looked at the carefully in detail at the morphology and the behavioral growth of the membrane at the top of the at the growing tip of the leading process. And what we saw that there were significant difference between area 17 and area 18 supragranular layer neurons. As it is shown here, the observation shows that the most frequent protrusion phenotype on the growing membrane of the leading process in area 17 neurons are philopodia. So philopodia has like thin finger-like protrusion, as opposed to area 18, which shows like a high percentage of lamellipodia. So lamellipodia has like broad sheet-like protrusion, and which are shown here. So these differences in these differences in protrusion phenotype are area specific, and we wanted to understand how they come about. And so we looked in order to try to understand. We look at the vesicular dynamics, at the membrane dynamics, and at the vesicular traffic of the leading process membrane. We did this on VAMP2 lipoaffected SP migrating neurons here in vitro, and so we image the movement of the vesicles, and we use we generate chemograph, which allows you to extract the direction of the vesicular traffic. So basically, you have the retrograde transport from the tip of the leading process back to the soma, the anterograde transport from the soma to the tip of the leading process. You also observe stationary vesicles and bidirectional vesicle, which will show both movements in both directions. And so when we compute the frequency of these different types of movement, we see that there is a significantly different vesicular signature between area 17 and area 18, with a significant reduction in retrograde transport in area 17. So as I told you before, that we think that these differences between area 17 and area 18 migrating behavior and protrusion phenotype are cell autonomous. We looked for a candidate which would be responsible for these differences in vesicular traffic. And because of our previous work, we knew that OSVC progenitor express different levels of P27 in area 17 and area 18. So P27 is a modular protein which has pleiotropic effect. It was mostly known for its cell cycle regulation effect. And when it is nuclear, when it is translocated to the cytoplasm, it can affect migration through its C-term domain. So we quantify the level of cytoplasmic expression between area 17 and area 18 supragranular layers, and we find an increased expression of P27 in area 17 neurons. So then we looked at the effect, whether these differences in expression level could be responsible for differences in the vesicular direction, vesicular transport direction. And so what we're doing, we overexpress P27 in progenitors, and we just look at the vesicular traffic in their post-mitotic progenies. And what we observe is that P27 gain of function, the increased level of P27, significantly reduces retrograde transport. This is a control, and this is a cell where P27 is overexpressed. And so we use also some mutants for P27 because I told you it's a pleiotropic molecule. So we have been able to show that this effect is not due to identified function, other identified function of P27, which is its activation, its inhibition of Rho A, and which is also its binding to the microtubule. Therefore, we wonder what is the mechanism by which P27 can influence vesicular transport directionality. So here it's a cartoon showing you that the molecular illustrating the molecular motor of retrograde transport. So the molecular motor is a dynein-dynactin complex. Dynactin has two subunits with P150 glued, which is a subunit of dynactin. So and this is so mediated the retrograde transport. So here this is immunoprecipitation from fresh tissue from area 17 SP, and it shows that P27 bind to P150 glued. So we wanted to get a bit deeper in this, and we did some computational docking simulation to try to better nail down the interaction between P150 glued and P27. And this approach shows that amongst the predicted interaction domain, the most plausible one is the CAP-GLI domain of P150, which is mediating the interaction of P150 to the microtubules. So if I summarize, we have identified a new function of P27, which is binding to P100 to the subunit of dynactin, and this is in addition to its map function here, which has been described in 2012 by the group of Laurent Nguyen in Liège. So far, I've shown you that radial migration mode is modulated in an area-specific manner, with radial migration, strictly radial migration in area 17 and abradial migration in area 18. I've shown you that P27 KEAP1 is mediating changing in the vesicular flow directionality. And we also, this is correlated with a different area-specific expression of RAB11, which is a small GTPase, and GTPase are known to be regulators of the vesicular traffic routes. So you can see that if you look at this immunostaining and this cartoon here, that area 17 and area 18 neurons show a different intracellular distribution of RAB11. In area 17, it's concentrated at the top of the leading process, whereas in area 18, it's the leading process. There is a continuous expression of RAB11. And if we do overexpression of P27 in area 18 neurons, we induce like area 17-like distribution of RAB11. And interestingly, we screened several candidates, and we find that beta-catenin expression in the leading process mirrors that of RAB11. I will come back to this later. So now we know how to modify retrograde transport. We have P27 as a tool. So what we have been doing is looking at the effect of retrograde transport reduction on the protrusion phenotype. So here, area 18, we overexpress P27 in area 18 neurons, and so we induce a reduction in retrograde transport, and this results in with an increase in the a reduction in the amount of lamellipodia in area 18, which overexpress P27. So this result shows that the reduction of retrograde transport induces modifies the protrusion phenotype. But now what we have been doing, we have been looking at whether the reduction of retrograde transport modifies the trajectory of the migrating cells. So this is a control migration pattern in area 18. This is two experiments in which we have been reducing the rate of retrograde transport either by micallolide B, which is a small molecule which specifically inhibits retrograde transport, or by overexpressing P27. So this shows that the reduction of retrograde transport induced straight trajectory in area 18 neurons. So we can summarize by saying that vesicular traffic impacts the radial migration trajectory via area-specific modulation of membrane growth directionality. And we have evidence that beta-catenin is a major player in this system, in this process, but I don't have time to show it to you. And sorry, something is popping up on the screen. I've got this coming. Oh. And so the last slide, because I'm going over time, the last slide is just a summary.
Wireless. Oh, that's your computer.
I couldn't, I did it, but I couldn't get rid of it. Okay, so the can we get out of the maybe I do this escape? Escape?
It's your escape right here.
Oh yeah, escape, yeah. And so this is the conclusion slide. Okay, so we have seen that there is differences in the tempo of production of cell cycle and also of neurogenesis of infra and supragranular layers, with difference in rate, speed of radial migration. And we have also seen that there are area-specific differences in radial migration trajectories, which are underlined by differences in membrane protrusion behavior. And this is determined by the levels of P27, which is differently expressed in these neurons in the two areas. And this is the last slide with the people in the team. In red, those who have contributed to the studies, and also in red, the people passed from the lab of collaborators. And our Henry Kennedy, which is our long-term collaborator. Thank you.
Is that related to the different species of outer radionuclei? I don't think you mentioned that. And also, do you think it could potentially even be initiated earlier on by the sort of direction to which the cells point when they're initially generated from the gelatin?
So question, several questions. First question, we explored two areas. And we also started to do it in prefrontal cortex and somatic cortex, but it's taking, it's very time consuming, so we don't have the answer yet. we looked in different areas in the mouse. In the subplate, there is no these meandering trajectories. In the ferret, Arnold has looked at it and published that you found tortuous trajectory, but within the germinal zone, not in the subplate, and you did it when on a gyrus. And this is what we have is different because we are looking prior the formation of any folding. And so this abradial trajectory in area 18 very likely means that the topology of the network will be different because there will be more lateral dispersion of the neurons born from one progenitor, or from the same colon of progenitors. So this will probably impact on the circuit organization.
So the interesting thing between Area 17 and 18 that's already known is that Area 17 is very, very specific to function. It's a large body and it's very tropically related, whereas the non
Absolutely, yeah. Yeah, so so area 17 is different in terms of what it receives from the thalamus, but as we have shown, is different in terms of its proliferation. And so the OSVZ is proliferating more in area 17 than in 18 because it has to produce, twice as many supragranular layer neurons. And we have evidence, it's not yet published, and we're doing this with Zoltan, that the thalamic afferents are promoting, which are targeting area 17, are promoting proliferation. And one specific feature, also I didn't have time to mention, is that in the primates, the thalamocortical embryonic thalamic axon, they just go to the germinal zone. It's not like in the mouse, in the rodent, they go above. So this is also an anatomical specificity of thalamocortical organization in the primate, which has, we think, important consequences.
Do you think that these remarkable differences in migration mechanics, such as the dynein or dynacting motor and their anchoring via P27, are these downstream of the genetic gene expression architecture that defines area 17 versus 18 through EMX, PAX, whatever, diffusible as well as membrane-bound gene expression patterns, and these sort of follow I mean, this is very deep cell biology.
Yeah, so this is
that is not necessarily predicted by our understanding of what makes cortical areas.
I think what's the interesting thing is that we can see for instance, the cytoarchitecture as a readout of the cell cycle, and the cell cycle regulation itself is a readout of all these early factors and gradients of gene expression. And what we see here, in fact, is that the proliferation rates and the migration in our understanding, are very much intertwined, because then you have this highly enlarged supragranular layer to form. Then you have to migrate faster, and also maybe you are more efficient if you migrate straight than if you wander about. So I cannot really answer your question, but I think this is another level of looking at things and thinking about things. But these differences in P27 level of expression, I mentioned we found them in the cycling precursors, but this will be in the nuclear P27. And what's important for migration is cytoplasmic. It has to be translocated. So it looks like all the machinery is really programmed to achieve at its best the final structure, if I may say so.
I have a very quick, follow-up, which is that, do you think that area 17 is really similar? And your difference between 17 and 18 is really similar, or you think that area 17 is really unique? Because there's a literature, as you know. Many people, Rakich and before that, Jones and Cahal and Jones and Powell have described how high the density of neurons is in 17, how highly stratified it is in the primate.
Yeah, so this
really is a non-typical cortex.
You could say it's a non-typical in that it has double the number of supragranular layer neurons, but this is also this peculiarity which has allowed us to go to the SVZ interrogate, because we have this nice system. Now, if we think like this enlargement of supragranular layers, it's reaching its maximum in the human cortex. So here we have a non-human primate, a monkey area, which is maybe closer to the cytoarchitecture of a human area because it has this enlarged supragranular layer compartment. And for again, for me, this is all related to the different programs of proliferation, because also these basal radial glial cells, they are providing the scaffold for migration. And again, this is very different from the mouse, because in the mouse, the radial glial scaffold is provided by the processes from the progenitor in the VZ, and it's not the case in the primate.
Let's thank Colette once again. All right, coffee break. Please be back at 11:10. So our next speaker is Arnold Kriegstein from UCSF.
Yes. Thanks so much. It's a distinct pleasure to be here. It's been a terrific meeting, as everybody has already agreed. And I will contribute my little part, which has to do with an explanation of neurogenesis in the human brain in particular, but with reference to other primates and to rodent models as well. And luckily, I'm not the first speaker, and so much of the background has already been described. As you heard from Suzanne, although the human brain is the largest among primates, it's certainly not the largest mammal brain or the most highly folded, but it does have the largest number of neurons. And that's the context of much of what I'll be talking about, namely the progenitors that generate the number and diversity of cell types, specifically in the human brain. And before I start with that, I wanted to summarize what we know about the mouse. Again, this is work that for all of you is redundant, but I want to emphasize that the radioglial cells along the ventricle are the primary source of all the neurons and glial cells in most mammal brains. So they're referred to often now as neural stem cells. In the rodent, throughout the life of embryo, they're really located at the ventricle only, where they produce intermediate progenitor cells, shown here in green, which then divide once in the mouse to produce a pair of neurons that migrate into the cortex. And that happens about 12 cell cycles to produce a radial array, a roughly radial array of neurons that span the 6 cortical layers. And then at the end of neurogenesis, these radioglia turn into astrocytes, again confirming their sort of multipotent nature and the fact that they're referred to as neural stem cells. Now, at early stages in human development, it looks very much like the mouse, which is shown here. I've shown an image here in a cross-section of the progenitor zones in the mouse, where the radioglial cells are stained red with an antibody to SOX2, and the intermediate progenitors, their daughter cells, are labeled with TBR2, and they form that subventricular zone adjacent to the ventricular zone. And those are the two progenitor regions throughout neurogenesis in the mouse. Now, early human stages look very much the same. But very quickly, the human outer subventricular zone develops, this very big expanded subventricular zone shown here. And on the right side, you can see the progenitors, which are stained in different colors because they're cycling. And the fact that the ventricular zone, as you heard from Colette, has become very small, and this outer subventricular zone is now by far the predominant progenitor region. And that's gestational week 17 in human, which is halfway through corticogenesis, when layer 4 is being developed. And so others, Rakich and Colette DeHay and Henry Kennedy, have speculated that those outer subventricular zone progenitors must be making largely these upper cortical layer neurons. but the cell types in that zone hadn't really been examined, and so a number of years ago, we and others started looking at those cells that are in the outer subventricular zone. And shown here on the left is a radioglia-like cell. Morphologically, it has that basal fiber in yellow that goes up to the cortical plate. The markers we had available at the time to stain ventricular radioglia in the mouse stain these cells, intermediate filament markers like vimentin and nestin, SOX2. Morphologically, though, they were very distinct. If you put a crystal of dye on the surface of the cortex at these later stages, it backfilled these fibers ending in cell bodies in the outer subventricular zone of this cell type, which hadn't been described before, so we called it an outer radioglia-like cell, and ORG cell for short. And more recently, Wieland has been calling these basal radioglia, but I'll stick with the term outer radioglia, ORG cells, for the rest of this talk. Now, we're lucky at UCSF that we can get donated tissue that we can treat very much like we do our mouse tissue, namely we can slice and culture some of these as well. And we've used a viral vector that only labels progenitors here. This is the outer subventricular zone over about 24 hours. And the progenitors, as you can see, are mostly these outer radioglial cells. They're migrating and they're also dividing. But they have a very particular mitotic behavior, which you may not have noticed. So I'll show it to you in these next two images, where we've only labeled outer radioglial cells. And when they're ready to go through M phase, the cell body translocates up along the basal fiber, and then the cell divides. And that happens with about a 100-micron jump in an hour. So these are rapid movements. And so we call this mitotic somal translocation. A little bit wordy, but it kind of tells you what's going on. And it's become a very characteristic feature of these cells. Not all ORGs do this, but most of them do in the human brain. And we've looked in other species and found the same behavior even in rare ORG-like cells in the mouse. So based on a number of other studies I haven't summarized, this is our working model of cortical neurogenesis in the human. And that is that at early stages, it looks very much like the mouse, shown here. But then the outer subventricular zone expands. The ORG cells derive from ventricular radioglia, and they go through a series mostly of these asymmetrical suffering Ewing divisions where they produce the daughter transit amplifying cells that go through at least, we think, 2 divisions or more to produce a clone of neurons that we think are all the same type of cell that then migrate into the same layer in the cortex. And this happens over time to generate multiple cortical layers. At the end of neurogenesis, the outer radioglial cells also transform into glia. We think they form a subset of astrocytes, in fact, which we won't I'm not going to discuss the non-neural cells in this talk. There just isn't enough time. And a number of years ago in my lab, we started doing single-cell nuclear sequencing in part to develop tools to identify these outer radioglial cells and to look at the diversity of progenitor types that we might not suspect in the developing human brain. And that's turned out to be a real gold mine in terms of not just coming up with new tools, but understanding some of the biology, which is what I'm going to spend most of my talk, I think, describing. So in our first studies, which wasn't that long ago, but in fact it seems remarkably primitive, because we started with our first publication of about 300 to 400 cells sequenced, the single-cell sequencing. And that's a very paltry number by today's standards, where people can do millions of cells. But this is only 7 or 8 years ago when the field began. But nonetheless, I just want to emphasize how much we learned from even just that small number of cells. So we enriched in progenitors by dividing the cortical slabs into ventricular and sort of non-ventricular regions. We dissociated the cells. We captured them on these microfluidic plates made by Fluidigm, and they were lysed and sequenced. And then we used bioinformatic techniques to try to cluster the cells that resembled each other together. And this is a Tisney plot where in yellow we've stained with canonical markers the neuroepithelial radioglia-like progenitors, and red are the intermediate progenitors, blue are the excitatory neurons, and the black cells are the inhibitory cells which had migrated into the cortex. And with this analysis of cell types, we were able to actually come up with the markers I was hoping for that distinguished, for instance, ventricular radioglia from outer radioglia. And what's shown on the left are genes that our data set were predicted to label ventricular radioglia, and these are in situ hybridizations to confirm that, in fact, all those genes, those new genes, were in fact labeling our old friends, the ventricular radioglia. On the right are the genes that our data set predicted would label the outer radioglia, and these were the ones I was most excited about. And again, the in situs show that they're labeling cells in the right region, in the outer subventricular zone. And then to confirm that they were staining the cells that we want, we correlated them with the morphology of the outer radioglial cells shown on the left, and in some cases actually did time-lapse imaging and saw the cells jump and divide, fixed them, and then stained them with these new markers to confirm that we were actually labeling outer radioglia. And after our paper was published, we were very pleased to see that the Allen Institute, using a different method of single-cell analysis, came up with many of the same markers for the outer radioglial cells. So this gave us now a set of tools we didn't have before to actually inquire about these cells in both the human brain and other species. But now I want to take a little bit of a detour to mention some insights we had in both normal development and also disease mechanisms based actually on these genes. So first, are these outer radioglial cells represented in the models you've been hearing about, for example, the in vitro organoid models that many people are now using to study human development and disease? And so in our lab, we've also been generating organoids from human pluripotent cell lines, and we've noticed that when we culture them over about 10 weeks, in this case a 15-week-old organoid, we start to see the gene signature, that is the network of genes that we've identified now as being an outer radioglial gene signature in the organoids, and suggesting that we might have ORG-like cells at that stage. And so we then sliced and stained our organoids the same way we did our fetal tissue, and this is an example of a time-lapse image from one of the organoids. And if you look at that cell with that long primary fiber, it's going through the mitotic somal translocation division that we find is characteristic of these ORG cells. So this is part of the evidence we have now that these organoids do in fact produce outer radioglial cells. And that turned out to be important when we studied this disease, lissencephaly, that many of you may be well aware of. It comes in several different disease states. The most severe form of lissencephaly is Miller-Dieker syndrome, shown here on the left and the bottom, where the cortex is entirely unfolded, and these brains are also smaller. They're microcephalic as well as being smooth-surfaced. And what we found is that the organoids made from multiple individuals with Miller-Dieker syndrome compared to normals had abnormalities in the behavior of these outer radioglial cells. Namely that when we looked at the Miller-Dieker outer radioglia, the cells were dividing by jumping further during that MST jump, but then they arrested and failed to divide or divided much more slowly. And what was interesting about that phenotype is that it didn't appear in the other progenitor types. The ventricular radioglia were dividing normally, intermediate progenitors divided normally. It was the outer radioglia that seemed to be selectively affected. And there's two take-home messages from that. One is that we wouldn't have seen this in the mouse. In fact, other people have reproduced these LIS1 mutations in mouse and not been able to see defects like this. And secondly, I like it because it links the outer radioglial cell to cortical expansion and maybe even folding, which is, of course, the main phenotype in this disease. It's also, of course, a cell-specific feature of a disease, which I think is just an example of other ways that we can start looking at human diseases in terms of cells that underlie the expression of the disease. And I'll get to that at the end of my talk. Another feature that I hinted at is that we not only have markers for these new cell types, but we also have insight into some of the gene networks and what they might be doing. And in the outer radioglial cells, there are genes expressed that are not found in other progenitor cells. And they fall into these general categories. Extracellular matrix production, epithelial to mesenchymal transition, which we think is actually how these outer radioglia arise from ventricular cells, and genes associated with stem cell maintenance, preservation of a stem cell niche. And every one of these processes that we went to the literature to look for had been described before, but not in developing brain, but rather in glioblastoma multiforme. As many of you know, this is a terrible, uncurable disease of adults. It's a tumor that arises in the brain. It's very difficult to treat because it's so highly invasive. It comes in several different flavors. The most invasive form is called mesenchymal GBM or glioblastoma. And we managed to get samples to do single-cell sequencing from our colleagues at UCSF. And what's shown in the heat map are gene expression patterns for these primary glioblastoma tumors. And the ORG gene signature, which I'm highlighting there, was found in the most aggressive and mesenchymal form of these tumors, suggesting to us that there might be ORG-like cells in the glioblastoma. So we managed to get some of this tissue. We sectioned it and stained it, as you're probably familiar with. I've mentioned it a couple of times already. And we time-lapse imaged these cells. And if you can look at this particular tumor cell, shown with the arrow, has the morphology of an outer radioglial-like cell with the primary process. And when it divides, it surprisingly jumped and divided just like the MSTs we saw in primary fetal tissue. This suggested to us that there might actually be a role for these outer radioglial cells in tumor formation or possibly invasion. So that led to a series of studies I just want to briefly summarize as unpublished work and actually in progress. But we followed multiple divisions of these ORG-like cells in the tumor, and as shown here, we found that daughter cells were themselves prolific, so that these cells are able to make progenitors in the tumor itself. They could be tumor-initiating cells. So we looked at the comparison, the correlation of the gene expressed in different cells in the fetus or the developing brain that could be sources of these tumors. And that included astrocytes, the protoplasmic astrocytes, fibrous astrocytes, OPCs, that is oligodendrocyte precursor cells, and of course radioglia. And the highest correlation of gene expression in these aggressive tumors was with the radioglial compartment shown here. And further, if we looked at the modules of genes expressed in these tumors, we found many of the outer radioglial cell signatures were some of the hub genes in the most aggressive tumors. And I wanted to emphasize one of the genes in particular, PTPRZ1, which is the most highly expressed gene in the ORG signature in these tumors, and which we had previously identified, as shown here in these violin plots, to be highly enriched in these outer radioglial cells that we've been talking about. So based on that, we wanted to look more carefully at what the role of PTPRZ might be in the normal biology of the outer radioglial cells. And we knocked down PTPRZ1 in the primary fetal cells. These are not the tumor cells. And as shown here on the right, it reduced the mitotic somal translocation significantly. In fact, the cells jumped half or even less than half the distance, and some of them didn't jump at all. So PTPRZ1 is part of the pathway that involves the motors, which turn out to be actin-myosin motors that drive the jump in these cells. And by knocking it down, it inhibits their jump. And the reason that was interesting to us is because, as I mentioned, one of the key features of the tumor is how highly invasive it was. And that made us wonder if this mitotic somal translocation and the ability of these cells to grow their fibers could contribute to the invasion of this tumor type. So we used an assay we developed where we took some of the tumor samples, grew them in a tissue culture dish, made a scratch, shown here under the microscope, creating a clear space, and then waited to see if the tumor cells would close that space, migrate essentially. And we did this both in controls and in the presence of a knockdown for PTRPZ1 and a ROCK inhibitor, because I failed to mention a number of years ago, we looked at the mechanism of the MST, and it was Rho activation of ROCK that actually phosphorylates ATP and actually causes the actomyosin contraction, and ROCK inhibitor prevents the MST in outer radioglial. And as I hope you can see in the lower panels that while the control tumor cells invaded that clear area, knocking down PTPRZ1 or adding the ROCK inhibitor to selectively inhibit the mitotic jump prevented the cells from actually crossing that gap and filling it in. So it seemed to us that this could be a mechanism that could explain the invasiveness of the tumor, the spread of the tumor. And we're very gratified to see this paper from Shi's lab, where they used a xenograft model where they took GBM tumors and put them into mice, and then knocked down PTPRZ1, which is shown in the middle and the right-hand panels, and either completely blocked or significantly inhibited the spread of the tumor. So we think that the jumping MST-type cells might help explain why some of the glioblastomas are as aggressive and difficult to treat as they are. And it might give us some new targets to treat. To prove this a little bit further, we took our tumor samples. We're able to actually selectively enrich the ORG compartment, the PTPRZ1 cells, which are the ORGs shown here on the right. And we took the enriched and depleted samples and injected them into organoids. We found that doing xenografts in mice took many, many months, but these tumor cells, in a matter of a week or two, grow very nicely in organoids. And I hope you can appreciate that the PTPRZ1 enriched fraction of tumor cells spread and reproduced much more widely than in the PTRPZ1 depleted cells. And if we look at the, I'm not showing you that here, but we look at the diversity of cell types, the PTPRZ1 enriched cells actually recreated the same diversity of cell types in the tumor. So this supports our hypothesis that these may actually be cells of origin for at least some of these very aggressive glioblastomas. A very unexpected direction of work in my lab because we don't usually work on tumors. But getting back to what we do work on, that is normal cortical development, having just those few tools to identify ORG cells has given us, I think, an insight into a feature of cortical development that I think is very important and helps tie in with what you just heard from Colette DeHay about the supragranular layers in primate cortex. And that's shown here, where we use two of the new marker genes, HOPX, shown in red on the left, which labels the outer radioglial cells entirely. So you can see their cytoplasm and the fibers that they produce. And then CRYOB, which is shown here in green, which labels the ventricular radioglia. And at ages like gestational week 18 or further on, which is when the upper cortical layers are produced, we notice that when we use both markers together, we'd expect to see green fibers all the way up to the cortical plate. That is, the ventricular fibers should be continuous. Instead, we only saw red fibers in the cortical plate and, and the PL region, suggesting the green fibers weren't going all the way up to the cortex. And in fact, when we just look at the individual channels, the green channel for CRYOB shows the ventricular fibers ending in the outer subventricular zone region. And so we looked at this in a different sort of orthogonal way by using dye crystals. And if we put the dye crystals on either the cortical surface or the ventricular surface, up until about halfway through neurogenesis, we saw continuous radioglial fibers, which are shown here in black. So they originate the ventricular zone and they go all the way up to the pia, which is the classical glial scaffold you'd expect. But after a week or two later, for example, gestation week 18, it's a very different pattern. If you put the dye crystals on the pial surface, they end in these fibers in the cell bodies of the outer radioglia in the outer subventricular zone. And if you put the dye crystals of the ventricle, shown on the right, it labels the ventricular fibers that also end in the outer subventricular zone, suggesting a discontinuous glial scaffold, sort of confirming what the marker expression suggested. And that's leading us to this another work in progress, a model of cortical development that we think now may be not just human, but maybe primate specific, where on the left you can see at the earlier stages, when the deeper cortical layers are produced, it's a continuous glial scaffold, and cells are produced both from the ventricular progenitor zone and also from the outer subventricular zone, which appears at those early ages. But then about halfway through corticogenesis, when layer 4 starts, there's a wave of production of outer radioglia that then move away from the ventricular zone. The cells they leave behind, because the outer radioglia have inherited their basal fibers, regrow fibers. But when they regrow the fibers, they don't go all the way. And they often end in the outer subventricular zone on blood vessels. So we're now calling those cells a different type of radioglia. We're calling them truncated radioglia because they're literally truncated. And the outer subventricular radioglia, the ORG cells, now are, we think, producing and guiding the migration of the upper cortical layer cells, the supragranular neurons. So a different lineage for the upper layer neurons compared to the deeper layer neurons, as shown here, ties in, I think, with this feature that you heard about with the supragranular layers of primates being highly cellular, containing maybe a larger diversity of cell types and a larger number of cell types. And I think what this shows is they also have a different lineage. And we think those things are related. And at this point, as I say, it's a working model where we're trying to fill in more details. But this came about largely because of those new markers that we had available. And that was just with a set of around 350 cells. So since then, we've looked at larger numbers. We've now been using the drop-seq method to look at hundreds of thousands of cells, but I won't show you that data yet. Instead, I thought I'd start by showing you the next step up, which was our study of about 4,200 cells, which we collected from different brain regions and at different ages. And as expected, that gave us a much larger diversity of clustering or cell types. In addition to just looking at gene expression, we also did what you heard about from Dan Geschwin. We did a weighted gene co-expression network analysis to look at modules of gene expression that were, in a coordinated way, giving us some information about biological processes. And I just want to talk about a few of these findings. So first of all, as I mentioned, we had a larger diversity of cell types, including endothelial cells, microglia, different types of excitatory neurons, different types of inhibitory neurons, different types of progenitors. I just wanted to highlight some of those features. First, the aerial map, and how is that actually, how does that emerge? And we had some insight into that because we sampled from the different brain regions shown in colors. And I want to highlight the two maybe most divergent regions, the blue, which is the prefrontal cortex, and the visual cortex, which is in pink. And what was interesting is that the progenitors, the radioglial cells, the intermediate progenitors, which are shown here in the dots, those each dot representing a single cell from either the frontal cortex or the occipital cortex. In this case, we've just looked at the progenitors in those clusters. And they're salt and pepper. Essentially, we don't see gene differences that label radioglia from the front of the brain any differently than the ones in the back of the brain. But if we look at young neurons as they start migrating, and those are the clusters shown here now that are circled, we clearly do have segregation of gene expression based on arealization, so that the same pyramidal cell type in the occipital region versus the frontal region expresses different genes. And these are kind of arealization gene expression differences. We look more carefully at all the different cell types that we were primarily interested in. These are shown on the left: radioglia, intermediate progenitors, deep layer neurons, upper layer neurons, and interneurons. And what these violin plots are showing are genes on the left-hand columns, individual genes, enriched in the frontal region, but not the occipital region, for each of those different cell types. And in the right column, I'm showing enrichment of genes in the occipital, these cells that are located in the occipital regions versus the frontal regions. And what this confirms is that there are genes that you can find that are regionally different for all the cell types that we've been able to look at. But what's interesting, if we look at the number of gene differences, there's a very clear pattern that the progenitor zones, for example, the radioglia, have the least arealized different gene expression. Intermediate progenitors have a little bit more, but the migrating deeper and upper cortical layer neurons have much more gene expression differences that are regional than the progenitors do. And then, interestingly, the interneurons have the least number of differences, and that may have to do with what you all probably know about now, the tangential migration of interneurons. They don't really arise in different progenitor regions of the cortex, and they distribute more or less across areas, and they don't have as many gene expression differences that are arealized either. So this leads us to this notion that there's a cascade of increasing number of gene expression differences as the neurons progress first from the progenitors into their migrating neurons and then into the cortical plate. And also, although I don't show it here, there's a decrease in gene expression areal differences with later maturation. So there's increasing complexity of gene expression, and then in some cases that decreases probably because those genes are no longer needed, connections and so forth have already formed. And then, of course, I'm really interested in the outer radioglial cells. That's my favorite cell type at this point. And there are a number of genes that our data set suggested were uniquely expressed by outer radioglia. And I just want to talk about some of those. Among them were the cells that are the genes that are involved in self-renewal, and in particular the LIF-R STAT3 pathway, which many of you may know is found in many other stem cell systems. It's an activated gene network that's used usually to self-renew or to expand cell populations. And we found it, interestingly, uniquely expressed by outer radioglia in the developing human brain. And we took advantage of that in an organoid preparation because there isn't a lot of intrinsic LIF, that is leukemia inhibiting factor, intrinsic in the organoids themselves. So we added LIF to these organoids, and after about a week, noticed, as shown here in HOPX expression labeling the outer radioglia, this huge increase in the number of outer radioglial cells, confirming the self-renewal that I mentioned that LIF-R STAT3 pathway is involved in, also the fact that it's expressed uniquely by the outer radioglial cells, and suggesting how you can, with this kind of information, optimize the use of, say, an organoid to reproduce a little bit better normal human cortical development. Not only does this exposure to LIF give you more outer radioglia at these earlier stages, but as you wait longer on, or later on rather, the organoids have larger numbers of upper cortical layer neurons, as you'd expect, by the expansion of this outer radioglial population. So that's one of the differences we noticed. Another thing we noticed was shown here. It's a little bit complicated, but let me explain it. We're looking at gene expression differences mapped, in this case, across pseudo-age, that's the x-axis, and then laminar position, which is the y-axis. So we could identify where the cells came from, if they were from the ventricular region or the outer subventricular zone, and the color map indicates where those gene expression, where the gene expression was found. And just to help you interpret that, we looked for the DAB1 gene, as shown here. It's a renin signal mediator gene, and it's expressed in outer radioglia at early stages of development along the ventricular zone, which is what the lower left patch indicates. And it's also expressed in outer radioglia, which arise a little later in development and are out in this outer subventricular zone. So those two cell types are enriched with this DAB1 gene expression. What's missing is down here where the truncated radioglia would be, because the ventricular radioglia transform into truncated radioglia. This is where you'd expect them to see it. And just to show you, here's a gene that's expressed. In fact, it's a marker gene we now use for truncated radioglia, shown here. And they're missing because they don't express DAB1. So the truncated radioglia are not expressing that renin mediator. And why I like this, the reason I'm going through this talk, mentioning it so much, is because the source of reelin is the Caharetia cell, shown here in blue, that's in the marginal zone at the top of the cortex. And the ventricular radioglial fibers reach to that area and could receive reelin signaling. So do the outer radioglial fibers. They reach to those Caharetia cells. They could also receive reelin signaling. But the truncated radioglial cells don't. And so we think that may explain why the truncated radioglia don't show expression of this reelin signaling pathway. And it helps confirm the discontinuous glial hypothesis I showed you earlier on. I hope you all follow that, but that was the reason I thought I'd show you that. The other feature that I wanted to emphasize is mTOR signaling genes, almost all of which are enriched in the outer radioglial cells selectively. And this was a surprise. mTOR signaling is a very ubiquitous pathway. It's found in many different types of progenitors. But in our samples of the human brain, it turned out to be uniquely expressed by outer radioglia. And this was confirmed in tissue sections where we looked at expression of a protein PS6, which is activated when mTOR signaling is active. And as shown here, it highlights the outer radioglial cells. It even highlights their basal fibers. And so mTOR signaling in human outer radioglial is especially enriched. And we think that's interesting because, as many of you probably know, mTOR signaling has been implicated in neuropsychiatric diseases, in particular in autism and tuberous sclerosis and in macrocephaly. So we think that the fact that it's cell type specific is very interesting. And I'll come back to that at the very end of my talk. And the other thing that I've been doing that I don't talk about much yet, but I thought I'd mention in this group, is we've been looking physiologically at how these cells signal to each other and how they could respond to signals that are in the environment prior to synapse formation. And we're doing this in a somewhat high throughput way by dissociating cells, loading them up with calcium indicators, capturing them in a microfluidic plate, which is a fluidine machine called the Polaris, which allows us to image them even up to a few days prior to doing single-cell RNA sequencing on the same cells. So when these cells are imaged, we treat them with a pattern, a barrage of different transmitters shown here. These were selected because either they've been shown in the mouse or rat to be active in progenitors or early migrating neurons, or because our data set showed that the receptors for these different agonists were present. So we used serotonin, ATP, glutamate, ACH, GABA. And as you can see in the calcium responses shown below, different cell types seem to have very characteristic or stereotype patterns of response to different agonists. And just not to show you the real raw data, but just to give you the bottom line, what we found is that the progenitors are uniquely responsive to purinergic and serotonergic activation. The young neurons, as they're starting to migrate, turn off the serotonergic and purinergic activation. They no longer respond to those, but they respond to ACH and AMPA, glutamate activation through AMPA receptors. And by the time they reach the cortical plate, you find GABA responses and NMDA responses very abundantly. So there's a transition, a sort of very clear, almost lamination to the receptivity of these different cell types. And then we're starting to look at what that might mean in terms of circuits, who's signaling to who, and what the consequences, functional consequences might be. And then the final thing I wanted to talk about was our study of evolution. And for that, we're very interested in comparing human outer radioglia and other progenitors to those in chimpanzee, our nearest living relatives. The problem with that, well, first I want to mention some of the differences that intrigue us. One of them is, of course, the human brain is much larger than the chimpanzee. It's 3 times bigger. There's been selective expansion of certain association brain regions. And as I mentioned, the upper cortical layers are different. So those are all features we'd love to be able to explore in more detail. The biggest problem, though, is that we don't have access to fetal chimpanzee tissue. And so Alex Pollan, who's a evolutionary biologist who joined my lab 4.5 years ago now, came with that particular goal in mind. And the strategy that we decided to use was to grow organoids from these different individuals, and we can do that from chimpanzees' fibroblasts, and then try to get single-cell sequencing and look at the differences that are species-specific. And it took a number of years, actually, to develop a protocol that worked equally well on all these different species, because we had to, of course, keep it very much the same. But that we were able to do. And then, as shown here, just in a few snapshots, we got pluripotent iPSL colonies from 10 humans and 8 chimpanzee individuals, made multiple lines, and then using a standard protocol, converted them into organoids as shown here, which in cross-section had progenitor regions and areas of both radioglia, intermediate progenitors, young and upper cortical layer neurons. That allowed us to start looking at similarities and differences across these different species. But before we did that, well, yeah, let me just mention that. So as shown here in the first column, from primary human and primary macaque fetal tissue, we were able to do single-cell RNA sequencing and have the same cell types expressed simultaneously in the same t-SNE plot. So here we have mixtures of both monkey and human cells from the fetal brain, and they're all mixed together. That is, you can identify the same cell types across these two species, and that allows us to look at the similarities and differences. When we look at organoids from human and chimpanzee, as shown here, we dissociated them and captured them, did single-cell RNA sequencing, and as shown here again, in these t-SNE plots that combine both human and chimpanzee cells, we can see them overlie each other. So we can see the same clusters enriched in progenitor cells or in excitatory neurons, inhibitory neurons, and this gave us the confidence that we could start looking at the same cell type in all the different species that we wanted to study. And that's what leads me to the next little bit of a departure, which is how well do these organoids actually mimic primary tissues? And the bottom line is that there are similarities and differences. And the differences I want to emphasize first is that the organoids are enriched in glycolysis networks, as shown here. The genes in the violin plots on the right are expressed in glycolysis. The first two violin plots show human and chimpanzee primary and macaque primary tissue that don't have those genes, but all the organoids, and not just our organoids, but those that have been published in other protocols, they all have very enriched stress signatures, namely this very enriched glycolysis pathways shown by the GO terms listed here. In addition, they also had ER stress networks that were very much more activated than normal, shown by some of the sample genes here. So these are organoids that are under a great deal of metabolic stress. So I think you have to be very cautious when you interpret disease phenotypes in these models, and especially when you're modeling metabolic disorders. But the increased stress might actually explain why some of the neurodegenerative disorders are seen in these fetal stage cells because of this enormous amount of stress that they're under. In addition, we also find that transcription factor expression is greatly increased if you compare the same cells in primary tissue to the organoid tissue. Not only is there an abundance of transcripts, but they're actually miscoded, as it were. We have mixed identity. This is shown schematically here, where the different cell types on the top each have their own signatures of gene expression. In the organoids, we find the same cell types have mixed gene expression. So the majority of the genes seem to fit, but there are exceptions that we don't find in primary tissue. And that could also be important when people are using organoids to model disease. So that's sort of the bad news. But the good news is that we do find all the principal cell types, and we can pull out these gene networks or modules of genes that are characteristic for the cell types that we're familiar with. And I've shown some of them here: radioglial cells, intermediate progenitors, excitatory neurons, inhibitory neurons, and the major hub genes and the networks of genes that characterize these different cell types we find in organoids as well as in primary tissue. So that allowed us to start looking at cell type specific gene differences. Before I do that, I just want to emphasize the good news, which is cell composition is largely overlapping with organoids and primary tissue. Gene co-expression is high. Cell type correlations are good. Patterns of neural differentiation over time are also correlated. So those are all things that we find are reasonably good, and we can rely on that. So now, just focusing on my favorite cell type, which are the radioglial cells, we looked at genes that are found in human but not chimpanzee radioglia, and vice versa. Genes that are found in the chimpanzee but not in human radioglia. And I'm showing just here two examples of genes that are uniquely expressed in the human, not in the chimpanzee or in the macaque. And this is an example of a gene that's found in chimpanzee and macaque, but not in human. So we find examples of this in both directions. And these are cell type specific genes. So in this case, just radioglial genes. And the final slide, or next to final slide, is this one. I mentioned that outer radioglia are enriched in mTOR signaling genes. So if we look at the mTOR signaling genes shown here, as I mentioned, in the primary human tissue, we find enrichment of that pathway. In the human organoids, we find exactly the same thing, which is very satisfying. But as you may notice, in the macaque and the chimpanzee genes, there's no enrichment for mTOR signaling pathway genes. So what that tells us that we didn't know before is that the mTOR enhancement enrichment in outer radial glia is human-specific. It's not found in other primate species. And just to confirm that, we have some fetal tissue from macaque. We went back and stained it for PS6, which is the sign of activated mTOR signaling, and is shown here in the quantified plots. Although the human outer radial glia are highly enriched in mTOR signaling pathways, the macaque tissue was not. So we now are very intrigued at this human-specific feature that might actually have some disease relevance. And that's where I was going to leave this story and just highlight the conclusions. There's a greater diversity of radioglial neural stem cell subtypes in human than we find in mouse, that combine comparative analysis in primary cells and the use of these organoid models can help reveal human-specific changes during neurogenesis and also, of course, later in other stages of development. And then I also want to leave you with the message that has really, turned over my lab, which is that single-cell RNA sequencing has become a hugely powerful tool, not only to give us new tools, but to actually give us new insights into what's going on during brain development. And the people who did the work in my lab are all shown here on the left, but I wanted to highlight Aparna Budari, who's a current postdoc, who's a terrific quantitative biologist who helped us with all the bioinformatics, and then Alex Pollan and Tom Nowakowski, who were postdocs in my lab and helped introduce single-cell sequencing. They both now have been recruited to UCSF and have their own labs. And I want to thank the funding sources, in particular the BRAIN Initiative, which really was key for most of the studies I showed you. Thanks for your attention.
I'm afraid we have to move on. Please remember your questions, and we can talk about it in the discussion later.
This one?
I was going to say pull it up first.
There we go.
So the next speaker is Barb Finley from Cornell University.
So first, I'd like to thank you for allowing me to participate in this really excellent meeting. But there's also one additional thing, which is I've been spending a lot of time in data gathering to assemble things like my translating time work. And a large number of you are the people who have generated the data for that enterprise. So I'd like to thank you collectively for this project that we've all engaged in together, whether voluntary on your part or not. So I will be talking about a lot of work about trying to integrate changes in brains in adults across species with changes and conservation and developmental models. And I want to talk about 3 different things, in each case looking at these 2 issues in combination. So I want to just review a lot of known cortical developmental mechanisms that have been discussed recently, just in the last several hours. And then I've been also going to combine that with scaling of volume and neuron number across species, looking for the whole brain by layer and by area. And I'm not going to be talking about any genomic analysis that I've done myself, but I'm going to point at places where some kinds of genomic analysis would be really useful, in my opinion, and places where they echo the kinds of findings that you see in these other 2 subjects. So first is the generation of overall cortical number and volume. Oh, and before that, I like to do my credits first, so I don't forget. So the set of people that generated most of the primary data, Alan Workman, Dermot Callan, and Christine Charvet, a lot of the counting and translating time work, and Nabil Imam, who's here, who worked on some of the modeling of cortical area generation, and my two long-term colleagues, Richard Darlington and Luis Carlos Silvera, who died a couple years ago, unfortunately. So, so then let's go straight to the first topic, generation of overall cortical neuron numbers and volume. So here's this old slide of, use the pointer over here, which is to, what we're looking at here is a number of classification of primates and insectivores and simians in an old phylogeny, and we've looked at the change in the volume of this from the Stefan data set, and doing the simplest possible plot, which is brain size on the bottom and the size of each structure on this side. And what I want to point out here is just the one thing. This is the human column, and we're looking at these brain divisions from neocortex, striatum, and so forth, all stepped apart so you can see them. And the point is, in this graph and virtually any other that you do of this type, that the human points are perfectly predictable, that they neocortex and all the others, and that there are taxonomic differences that we'll be interested in discussing. But let me just give a different version of this same kind of thing, which is now represented a little bit better. And so now we've added a whole lot more and more properly classified mammals to this collection. We're looking at, down here we have brain core, which is brain stem, versus the particular structure that's named here, again stepped apart, so there's no contamination of the two. And we're getting the same kind of thing. In this case, the human point here remains the largest, but remains predictable compared to all the others here. And this is, so who cares? I mean, what does that mean? So I just want to clear up this plot here so it doesn't go under with so much detail about what you are going to ask about the different species. So here we have just the human, the African camel, cat, marmoset, and a mouse plotted, and we've computed the regression lines here, but I've just sort of drawn them in for illustrative purposes here. So we have the cortex and cerebellum and hippocampus for these species only now plotted out, and we have the camel because it's pretty close in volume to the human cortex size. And there's been a lot of interest in why the unusually large human cortex gets that way. Well, the point is, it isn't unusually large. It's on the line. It's on the line with other primates. So if you're going to give an argument about why this structure is large, you are kind of obligated to also describe why this one in the camel is also large. And what about the primates that are using the same mechanism on this trajectory? Why the cerebellum, or why, in fact, any structure is the size it is going to have to be decomposed into these sort of species general and species specific ones. No, I want to make absolutely clear that it's not making a case for absolute uniformity, but what I am making a case for is an overwhelming variable that has been missing investigation, which is duration. So if you look across the different mammals, particularly looking across primates, what's the thing that really is the thing that's carrying a lot of the variance in cell number? It's duration of development. And there are very, very few numbers about how duration is selected on or differs from one species to the next. So you don't have to take my word for it. There are a whole number of demonstrations of this. So Passingham is one of the first ones, and I think it's a very cute graph in a way because and inventive when you can't get to the right kind of material. So he simply plotted the weight of the brain at birth in a whole bunch of mammals. So this is, 62, and those are common lab animals and a bunch of farm animals and a few cetaceans, so just a mixed set. So what you've got is essentially 1 point per animal, from small to large ones, born at different states, and those brains are still all in the process of growing at birth. And so when you do this 1 point grandma, you get a straight line up. So neural tissue per day in these mammals is being produced at a pretty constant rate, a very constant rate. Down here, this is a different way of doing this. This is Drew Halley, who's now in Leah Krubitzer's group. So what we have here is plotted against changes in body size, changes in brain volume, looking in this case at a number of different neuronal primates. And what you can see is the difference between the different animals from small to large bodies is sort of when each monkey in turn gets off the high brain growth trajectory curve. So in this case, duration as terms of exit from rapid growth is what correlates directly with absolute brain size. So from Homo sapiens down through some of the smaller New World monkeys here. And here's our version of the same thing, which is now comes to the translating time site. I'll describe it a little bit, or project, I'll describe a little bit more later, but I just want to point out the main result with regard to this. So this is different from all the other graphs, is that time is on this axis, and maturational state is on this axis. And we have 271 different indices of maturational state that we've ranked over now some 20-odd animals. So this is going from generating your first neuron to being 80% of adult brain size on this axis. And this one is how the model looks as animals are going through that. So this is the mouse line, the black one, and the animal starts off and races from that first neuron up to the 80% brain size in some 30-odd days. The human takes a leisurely 300 days to complete those exact same steps. So what's going on in the big brains is you're doing the same things. You're generating cortex, you're generating a retina, you're generating more volume, but they're bigger, and it's taking you longer to do them. And so if you look, in this case now, the slope of these lines, the relative slope, will tell you the rate of progression, how fast you mature. So, and the steeper the slope in absolute days, the more quickly you're racing through these developmental steps, and that will give you adult brain size that really R equals 0.95 to 98 or another sort of embarrassingly high correlation. So now my argument here is that this is not the only thing that's going on, but there should be maybe a little more attention paid to the control of duration from people who are looking at the how you get adaptations in animals, because this is a massive factor and I think counts for a great deal of variance. So number two, again, known cortical developmental mechanisms, and we've just described a whole lot of generation of the cortical column, and particularly the stuff that Colette has described has just cleared up a number of questions about the detail of this that I will hope to just sort of point at here a little. So I'm going to look at how scaling of neuron volume and neuron number, but in this case mostly neuron number, what goes on. So this is a figure that was the sort of aha here that was Christine Collins, when associated with the Cost Laboratory, did this version of a picture like this, which is the number of neurons or the density of neurons per column, which is in this case kind of interchangeable for analysis purposes, where if you look across the cortical sheet, there is a gigantic difference from the caudal to the rostral part of the cortex. Many, many more neurons in caudal cortex. It's from visual cortex up to the prefrontal areas. I don't think I need to, this is just the cartoon for other kinds of audiences. And so I want to contribute another thing about how you alter developmental parameters when you go from this size of cortex to human size of cortex. Now the one thing that's very interesting is that it turns out that prior to a lot of just assumptions early on, or assumptions that came from selectively sampling the cortex here and here and here, and then saying, well, that shows that the cortex is a uniform sheet. When you actually looked at the entire cortex, you found that it had a really pronounced and interesting gradient in neuron number. And it's long been known for virtually every species that's been studied that there is a gradient across the cortical plate as well, which is that neurogenesis and the first organizational effects in the cortical plate occur roughly at the same time, although the caudal part lags a bit. But the thing that really differs is the end of the period, where neurogenesis in rhesus will be going on for a full 3 weeks more at the caudal pole than it does in the rostral pole. And just a note here, when I'm talking about this, I'm talking about the cortex lateral convexity and no limbic cortex, no olfactory cortex, simply the lateral convexity. So we wanted to do this by layer, and so we, help of our neural monkeys from Luis Carlos Silveira, we were set about to do some massive counting and analysis of the number of neurons by location and by layer. So this is, I'm not going to take you through all of this, but what I'd like to point out is we've got everything here. So this little thermometer graph is the total number of neurons in the cortex, a whole lot in Cebus, and there really is some in the hamster. There are a few. Okay, and this, the circle is the absolute cortical areas, so you can see the difference that we have going here. Then we're doing these, in this case, rostral to caudal number of neurons for the whole cortical sheet. And so we're looking at these small and some fairly hefty rodents here, the hamster through the agoutis and pacas. And this just represents our total graph here now, not normalized and put on the same graph so that you can see how the change in number is looking. And our point here is we've been trying to use both, the way we've been trying to get a hold of neurogenesis from this time-related view is to treat intracortex variation and between species variation as if they're generated by the same mechanism, which has worked out fairly well. So looking a little closer at just our primates here, we've got, I'm going to just echo with a different set of primates the point that it's the upper layers that that is the source of the difference between these animals. So looking at our capuchin, the Cebus, we look at this as all over the number of neurons per column from the front to the back, and then this is layers 5 and 6. This is layers 4, 2, 3, and I guess 1. And you can see that the thing that is producing the difference in the number of neurons per column is pretty much exclusively the upper cortical layers. The lower cortical layers stay the same, and it's been interesting to see just how strongly that has been now reinforced by a number of other species. Okay, so here the upper cortical layers are the source of the increased neurons. So we've gone to model that by looking at both in this case, the evo between the species and the devo, that's the time for how each cortex gets generated, and going from a mouse with a short period of time, 21 days or so, to generate all the neurons, and then further days to elaborate them. You'll see that there is a well-known Cavaness and Nowakowski of a gradient across even the smallest rodent cortices, but it doesn't appear in a great difference in numbers, so that the numbers of 5 and 6, 2 and 3, are pretty similar. But then as you go to longer developmental times, this has quite nonlinear effects, so that we're getting a bigger cortex overall, but particularly a bigger cortex in a number of neurons in later layers 4 through 2. And then bigger still, if you go to rhesus, this same kind of thing where the lower layer is increased by a smaller exponent, and the upper ones are increasing precipitously. Now this is what a cortical column difference would look like if you just translate this into dots instead of volumes. And so I've been really quite impressed in the last several talks about what I trying to integrate this developmental timing perspective with the cell biology perspective. So what I think has happened here, or you can demonstrate, in fact, that the events in neurogenesis in primates compared to rodents have shifted, so that the primate neurodevelopmental effects are happening later, and by that I mean not later in absolute time, but later with respect to our cross-species developmental scale. So they're shifted with respect to the baseline. So these primate events are happening later in its own brain with respect to, like, generation of the midbrain. It's a relative movement of this developmental epoch. So if you look at the rodent and you say what's happening during this envelope of cell generation, so this is, you might think of this as exit from the stem cell pool, and this is the statistical specification of each neuron type. And then if you move this and multiply by this times this, gives you the primate distribution of cell types. So that this is now moved into a maturational time period that carries different specification outcomes for the cells that are generated from the stem cell pool. And I think that is a lot of stuff going on there. And I think what Colette and Kriegstein and a number of the other people have been looking at in this has been exactly what this is going to unpack into in terms of other kinds of cellular mechanisms that could be addressed, like changes in rate of the cell cycle that are specific to the late times, changes in migration, different migratory patterns, and so forth. But you can capture it in terms of just numbers and volume relatively cleanly, which is an interesting feature by itself. Now, I just want to get away from the notion that all I'm going to show you is things that are the same. So here's a couple things that aren't accounted for by neurogenesis and smooth changes. So here's, first off, older data, which I'd like to point out. This is gathered by Below and Clonier a long time ago, and it's using EM to get numbers per column. And so what we're looking here is rostral to caudal. So in the cortex, and what we're seeing is exactly the same kind of thing in these cats, where motor cortex has fewer neurons for the lowest grade and very high in posterior cortex. But notice this thing, which is the high dosage, the high number in primary visual cortex and also in somatosensory cortex. So what's causing that? Well, I think one thing that's definitely causing it in visual cortex is the fact that it's sitting at the end of the maturational gradient. It has the most time to generate cells. But look here, it also seems to be a bit kind of step function, so there's at least one other factor, and this is something I was involved in describing in a mechanistic way years ago. So if you look at all our primate sample, look at primary sensory areas, you see that they are above the line. They have more neurons than you would expect per column if you look at simply neurogenesis. And we do know that there are close to 80% more neurons that are generated for layers 2 and 3 than will survive to maturity, and that the presence or absence of thalamic fibrils will save some of those doomed cells. So I think cell death is part of the thing that gives this bump in neuron numbers in primary sensory areas, and the mechanism that Colette described of having some thalamic instruction or change in rate of neurogenesis is also another source. So we have, I think, several factors combining to put a step function on a curve, basically. So we're taking a general predilection for putting high neuron numbers in the caudal cortex, and then we're amplifying that as well with a few other features. Now, here's another thing. This is just to talk a little bit more about the translating time model. So, and this is my usual ad, so everyone who needs to be able to say what exactly is the equivalent day in the African camel or whatever versus your laboratory mouse can go to this site and either directly look up or get how to compute, how to equate days of maturation across species to go from particular states to actual days. So here's another numerical difference that you see in the cortex that our method picked up before it was fully understood. So what we do is iteratively fit the best regression that regression line for each species independently that will fit those 271 events, and then put it into an overall model. But what we like to, and the model fit is very good because a lot of developmental timing matters don't change very much. But then we look at fit of data to model, and there was one set of neurons, see these little guys floating above the line here, that didn't fit. And that turned out because we had equated appearance of GABAergic neurons in rodent cortex to emergence of those same neurons in primate cortex. So it turns out that the number of that those aren't completely the same sources nor the same timing, and the primate cortex is dosed with extra GABAergic neurons from a different source. And you can see that in the model's failure to, say, recognize the primate and the rodent ones as the same neurons. Now, there's numerous amplification or versions of this. So roster caudal gradient and several things. So this is the several massive studies by Lean et al. And some of the people here of differential expression of genes rostral to caudal. It's the major kind of gradient effect they show. If you look at the cellular level, if you go in the frontal cortex, cells have lots of, many more spines and processes. The cell bodies are bigger. And this is from the massive Markov et al. If you look at each cortical area, for each area, how many other areas project to it. So if you go from frontal to visual, we've got 60 plus areas projecting to the frontal cortex areas and very much fewer projecting to primary visual cortex, with primary motor cortex a extreme outlier, which somebody should look into. Okay, so this makes a cortex, I'd argue, that changes progressively in an interesting way as you go from a small to a large brain. In the posterior cortex, the neurons are small. If you think of the pixels grabbing a visual image, they're grabbing a lot of them and doing a lot of detail and being able to recombine them. But then progressively, as you go in the direction of the feed-forward connections, the cortex, they're gathered and gathered into fewer and fewer, basically relatively fewer cortical areas onto larger cells. And this is something that changes progressively and systematically with brain size. So the bigger the brain, the more the compression of dimensionality. So we have to talk about always two directions in the brain. One is the integration, the other is the prediction direction. So in bigger brains, more abstract things like going from time of day to time of year, or minute to minute, or local space versus geographic space might be changing for the different species as they integrate things on that large cortical axis. And the last point, which goes rapidly, I think. So how about cortical areas? Something we don't know a whole lot about. We don't know a whole lot about S1 and V1, but talking with Leah, and we think there's about one study where people actually physiologically identified the development of any secondary area, which is just astounding. There must be thousands of studies on the development of V1, and there's nothing similar on V2. So where are they coming from? Does the cortex, have the same divisions or not? So I just want to explain how I'm representing things here. So this is the Markov anatomical model. Here we have their blowing up of that and labeling the thing. Then I am taking it and devoing it. So what we're looking at here is highlighting the primary sensory areas, so visual, somatosensory, auditory, and motor. And then I'm just going to square it up because I want to emphasize certain interesting features to it. So one of these is something that's been described a lot in visual physiology. If you look at how big V1 is compared to V2, you find that V1 is bigger, V2 is smaller, V3 is smaller still. The receptive fields do the opposite progression, where the cells are integrating over larger areas. Then the map breaks up into smaller topographic regions, and this is a progressive change as you get larger cortical areas. So we have this change going on. I want to point out that topology is how we're going to be talking about these things, so how the visual field is represented. But this whole lateral convexity really embeds all the maps into the egocentric map. So so this is the bottom of the visual field, and if you recall your homunculus, this is the representation of the feet. And here we have, moving the feet. So up here is down. Here is up. So upper visual field, head, and so forth. And then we have a nice little island in the temporal pole where we have still topological organization of the cortex. So we have representation of the cochlea, the viscera, faces, all things that are topological but not egocentric, which the developing brain for apparently a long time has decided to sequester. So we've got two kinds of space embedded in this lateral convexity map. Now, where do areas come from? This is an old graph looking at what people reported the number of cortical areas in the entire brain for a bunch of different species. And this always kind of perplexed me because you get this. So here we have the area of the cortex and the sort of size over these different things represented graphically as a square, so you get an idea how things are changing, that you could fit the entire rat cortex into one of the squares of the monkey cortex. But anyway, the number of cortical areas shoots up, and that doesn't change a whole lot. And it suggested, though it certainly doesn't prove, that there might be some biological process that was generating it. And we were going for a Hebbian one. The other thing that we need to know is this topographic seed region, and this I'm taking straight from Leah's work, the fact that we have a certain number of areas that are stable across species, and what you do is add areas in the middle. And and these are special in a variety of ways. There are many more different genes are expressed related to recognition, thalamocortical recognition particularly, and axon wayfinding. Then they're called seed regions. What are they seeding? There's one piece of organization people haven't talked about too much. For each one of these regions, old study, that if you look from the thalamocortical connections, ignoring nuclei, you'll find a continuous representation of space from LGN to pulvinar. In the visual complex, a similar sort of thing in the somatosensory ones, but there's a break in smooth topography between them. So these sort of compartments at the cortical level can be defined by this thalamocortical mechanism. So what we're trying to do is understand, can we generate these cortical areas not by the usual notion that there are specified areas that have some particular computational role that to do some exact thing that's been selected on per se, but could a generic mechanism produce them? So in combination with Nabil, who's in the back, we've made a model in which this is to represent, V1, and this is periphery, center, up and down, say, or in this case representing the way I did before, lower visual field and upper. And what we want to do is see if we can generate these features of cortical areas going from the large V2 to the smaller V3 with then the increasing topography by using the minimal Hebbian. So the Hebbian idea here is that these neurons here are going to be trying to colonize this available cortex expanse, and they're going to do it progressively. The ones that represent the periphery here have some advantage. They're right there, and they can get there first. They can establish peripheral connections. Then when these guys get here, those things are already taken, and they have to go, and their best bet to be in topographic order is to skip that and go to the next one. So what we're trying to do is use Hebbian mechanisms, a anchor region, and we're using basically a graph model to establish a connection line if it meets a essentially Hebbian criterion. So this is just a picture of how this, whoops, that's what I'm going to do, how this looks. You can get to it. Let me just... So, here's to watch it grow. So we see that we're establishing periphery, then doing the mirror reversal. And we can get, using just these minimal constraints, generate 2 to 3 mirror flips now, and also reproduce the feature of increasing topographic disorder the farther you get, or precision the farther you get from the original seed region, and the decreasing size or representation of a visual field. So this will generate this feature of a cortical area automatically from well-known cellular processes. And that's it. So that's it. So this is, what is the significance of each one of these things? So we're generating overall neuron numbers by things that seem advantageous along the way. We put volume preferentially cross species in areas of integration. As you get larger, you progressively abstract through the rostrocaudal gradient. We generate areas in relationship to the size of the cortex and the integration of neurons that suggests a great deal more plasticity in how they're organized than people may have originally thought. So that's it. Okay.
Hi, Barbara. If I got it right, you're saying that the difference where you have the gradient of the densities of neurons in the cortex of primates, the difference across sites is really due to expansion of the number of neurons in the supragranular layers, and that happens in the occipital cortex compared to posterior cortex.
Across the entire cortex.
Across the entire cortex, but the part where you have the most
yeah.
expansion is in posterior cortex. Right. So that's what I don't get, because my understanding is that everybody else, when they talk about expansion of supragranular layers in evolution, especially in the context of human cortical expansion, that's presumably something that happens mostly in frontal areas where you have associative processing. That's because we tend to associate supragranular layers with associative processing. Could you comment on how those two things work together?
That's the point. So what's different is that there are many fewer neurons in frontal cortex, which is forcing the abstraction and dimensionality reduction in the frontal cortex, whereas the posterior cortex is generating a large number of neurons for, I would guess, perhaps, image analysis. So it's the thing that produces the volume in frontal cortex is neuropil. It's the larger cells, the larger neurons, and all the processes, the 100 areas that have to converge.
I completely get that part. That's not what I'm asking. What I'm asking has to do with what Arnold just spoke about, which is how having more of an outer radioglial zone and more development in that part would give you expansion of the cortex, and mostly through the addition of a supragranular cell, and that is layer, and that is always placed in the context of how the frontal areas, associative areas in primates and humans in particular, are preferentially expanded.
I don't think any argument was made being particular to, I don't think anyone claimed that there were more neurons in any one of those areas as a hallmark of human organization. In fact, humans have the highest numbers of neurons in
that's
striatal
that's
cortex.
What cortical expansion means. I mean, we should ask Arnold.
This sounds like a great
yeah,
topic
I mean,
to
I
take
think
up in the
I think the
discussion.
change applies to all the cortical areas, but that's not the question.
Great. Let's talk more about this later. Sounds very interesting. Yeah. So it's time to enter the data blitz phase. The first data blitz speaker from UCLA is Aaron Gordon. Thank you, Barbara.
Hi, everyone. My name is Aaron Gordon, and I'm a postdoc in Dan Geschwin's lab. I'd like to thank the organizers for inviting me to talk here today. And before I start, I just want to say that all the work I'm presenting was done in close collaboration with the Pascal lab at Stanford. So what we were trying to do is see, we have the 3D spheroid module, and we want to see how far into maturation we can push these spheroids. So what we did is we cultured, this was all done in the Pascal lab, they cultured the 3D spheroids, and basically they kept growing them for very long periods of time. And our hypothesis was that the longer we grow them, the more mature they will become. And it turns out that we can grow them for very, very long periods of time. We can grow them up to 2 years, and currently they're growing them for even 3 years. And so we have all these data points across time, and we want to see, well, how mature are these spheroids? So the first thing we did, we wanted to test was the kind of the age of these spheroids. And to do that, we used the epigenetic clock developed by Steve Horvath, which is a combination of 353 epigenetic markers, and combining them creates a measurement that correlates very, very strongly with tissue age. And as you can see here, we also get a very strong correlation between differentiation day on the x-axis and the predicted DNA methylation age on the y-axis. And 0 being the day of conception, birth is about 266 days, so that's 9 months. And so that gives us the predicted age of birth, and this is birth in quotation marks, is about 280 days, which is very, very close to the 266 days which it takes in vivo. So next what we wanted to do is look at the gene expression patterns and compare what we get in vivo to what we have in vitro to what we get in vivo. And to do that, we use the BrainSpan data set by Nanad's lab, which has data all the way from 4 weeks post-conception all the way into adulthood. And what we do is we look for changes, we look for overlap in transitions. So in the BrainSpan data, we take every stage and compare it to the initial stage of stage 2. And in our data, we take every day and compare it to day 25, our initial day. And the stronger the red diagonal, so the stronger the diagonal is in red, the more overlap there is. And as you can see, up to about day 200, the overlap is almost exclusively with the prenatal stages. At about day 250 to day 300, we see this transition from the prenatal to postnatal, where after about day 350, the overlap is almost entirely with the postnatal stages. To get a kind of more detailed map of what's going on in these spheroids, what we did is we ran WGCNA to get modules of genes that get co-regulated or change expression in a coordinated manner. In the BrainSpan data, we then made sure that these modules were preserved and overlapped with our data in the cortical spheroids. And what we saw is that the trajectories of these modules follow the in vivo trajectories. So for example, in the top middle panel, the neuronal modules the increase in expression to about day 200, somewhere between day 150 and 200, which is the peak of neurogenesis, and then they go down, whereas the glial modules on the bottom left-hand corner initially decrease in expression as radial glia go down, and then as astrogenesis occurs, the trajectories go up again. And we wanted to generate some kind of predictions of some of the canonical changes that occur at birth. One of these canonical changes is the changes in the NMDA receptor subunit switchover. So in blue, it's the NR2B, and in red, it's NR2A. And in vivo, what happens is that NR2B initially is expressed at high levels and goes down, and NR2A is initially expressed at low levels and goes up, and the switchover occurs around birth. And as you can see, this switchover occurs right around our predicted age of birth in vitro. And with that, I just wanted to thank both the Gershwin and Pascal labs for this, for all the help and assistance, as well as Luis de la Torre, Jason Stein, and Steve Horvath, who developed some of the methods that are used here today, as well as our funding agencies.
One or two questions?
Yeah. Any data on the neuron activity in these organoids? We're working on that. Yeah.
Anyone else?
So you are using the same media and same conditions before and after birth.
Yes.
Do you think that you should move to solid feeding? so I'm just wondering whether the metabolic needs of these organoids, they should be changed with age. That's my question.
Possibly. Yeah, possibly. I have a son at around that age, so I can ask him. But no. The media we give these spheroids at these stages is actually very poor media. We just give it some basic media without any growth factors. The astrocytes are the ones that kind of support the neurons. So if the changes need, the astrocytes also mature. And there's a paper by the Pascal lab showing that these astrocytes really mature over time. So they would change what they secrete and what they support, how they support the neurons.
Thanks again, Aaron. Okay. So the next Data Blitz speaker will be Ana Rita Ribeiro Gomez. Oh, that's as close as I get, sorry. Thank you.
Okay, thank you. So my name is Anna, and I'm going to present you some results we have on an ongoing project in the Kennedy and DeHay labs that is evaluating the development of the cortical spinal pathway in the macaque. And so the cortical spinal pathway in primates is a highly developed pathway, and is extremely important to the execution and control of fine voluntary movement. And paradoxically, much of our understanding of the development of this important pathway comes from the studies in the rodent, because in the rodent, the cortical spinal pathway develops mostly postnatally, which makes it a very nice system to study the development of the cortical spinal pathway. And so what we know is that if you do a retrograde tracer injection in the newborn rats, you have a vast territory, the cortical spinal, the cortical neurons projecting to the spinal cord are broadly distributed across the cortex, and so if you, and they project from regions that do not have cortical spinal neurons in the adult. So basically, if you do a retrograde tracer injection in the immature rats, you see that the neurons are distributed across the cortex and they project to your injection site. But if you do the same injection in the mature rats, only the rostral part of the cortex project to the spinal cord, and all these neurons, layer 5 neurons that have this transient projection, they lose this projection. And so what we did is that there is a restricted pattern of corticospinal projections in the macaque, and so we went and we evaluated if the restricted pattern of corticospinal projections in the adult macaque was developing from a exuberant projection. And so what we did is that we did in utero injections in the corticospinal pathway at 2 embryonic ages, 95 and 105, and the injections were made with retrograde tracers that are very good to trace pathways in big brains, and they are very sensitive, and these injections were unilateral. And so what we decided to do was to evaluate the distribution of retrograde labeled neurons in the cortex projecting to our injection site, and we started by evaluating the contralateral projection, because it is from the contralateral hemisphere that most of the projections are present in the adult across different species. So here you have a parasagittal section of the brain. O refers to occipital, F to frontal, and every blue dot represents a labeled neuron in the cortex projecting to our spinal cord, to our injection in the spinal cord. And so basically what you can see is that whereas in the adult brain you have a restricted territory projecting to our injection site, if you look early in development, you have a broad distribution of neurons across the cortex, from more posterior to more anterior parts of the cortex, that then get removed and give rise to this adult restricted pattern. And so, as you can see here, it's very difficult to make comparisons across ages, because we don't actually have all the Suki and all the landmarks, Suki and gyro patterns that we have in the adult. So what we did is that we used acetylcholinesterase, cytochrome oxidase, and Suki and gyro landmarks to try to parcellate the brain in different regions, and then assign the corticospinal neurons projecting to our injection sites to these different regions. And in some cases, we were able also to put them in specific areas. For example, you can see that there are no corticospinal neurons at these stages in the area V1, primary visual area, projecting to our injection site, but you do have it, for example, in the primary auditory area. And so by doing that, what we saw is that there are regions that have transient projections to the spinal cord in the macaque during development, namely occipital, temporal, prefrontal, and there's also limbic, and that in the regions that do have neurons projecting to the spinal cord in the adults, these projections are much more broader, namely the parietal, frontal, insula, and also cingulate cortex. And so, because the injections were mostly were unilateral, what we could do was then ask what is happening in the ipsilateral hemisphere. And basically, the conclusion is that the same exuberance that we find in the contralateral hemisphere, we also find it in the ipsilateral hemisphere. And the fact that we use these tracers and we count every single cell that we see in these brains allows us to say, to highlight that during development, at least at these ages that we looked at, there's very little lateralization of the corticospinal tract. And so where the monkey brain and where the non-primate and the primate brain diverges is that we do have this exuberant projection. The divergence between primates and non-primates is that this is actually bilateral. And in addition, we also have some bilateral subcortical spinal projections. And that raises many important issues, and particularly, I think I would learn a lot from a later discussion in what might be the function of all these transient projections, which are many, many projections, and why do they exist in the first place. And that's it.
Question? Eric?
Yeah, I can speak up. So where are you injecting in terms of the spinal cord, and not only in the column itself, but in the motor neurons or the reticular formation? Because that might make a difference as well.
So we are injecting beneath the pyramidal acquisition at the level of C1, and so we are really in the spinal cord.
Now, are you trying to hit the reticular formation or the motor neurons, or both?
That's the thing. When you do the injections, and you can see that, for example, in the studies in the adult macaques, is that when you inject the spinal cord, in our case, for example, we were trying to hit the dorsolateral funiculus, where you have the fibers in the primate descending in the spinal cord. But the thing is that this is done in utero. It's very tiny. And so basically what happened is that, for example, in the youngest fetus, the injection does, it's involving the dorsolateral funiculus, but actually the injection in the eldest fetus does not actually encroach the dorsolateral funiculus, so it's mostly constrained to the gray matter and the dorsal funiculus. And so, besides that, we do have a lot of exuberant projections, so it might indicate that these projections are not in the white matter, but are actually already in the gray matter during development, prenatally.
Okay, we'll have to leave it there. Thank you very much.
Thank you.
And the final talk of the morning will be Stefano Berto from Southwestern.
Thank you. It's a pleasure to be here among these amazing scientists. And I'm Stefano. I'm a postdoc in Konopka lab. And I'm going to share with you my project that is ongoing in her lab. So I'm interested in trying to understand the molecular mechanisms that underlie human cognition, and also how these molecular mechanisms are disrupted in neuropsychiatric disorder. And I have a simple hypothesis that neuropsychiatric disorder are a consequence of evolution itself. And we know that in the past decades, a lot of effort has been done to decipher how human evolved such a big brain from different point of view, from developmental, structural, and also system biology point of view. However, so far we don't know exactly how different cell type contributes to such human evolutionary achievement. To answer these questions, we basically use a BA46, a Brodmann area 46, that is a region in the prefrontal cortex strongly associated with the human cognition and neuropsychiatric disorder, and it's a orthologous region in chimpanzee and macaque. Using a fluorescence-activated nuclear sorting approach, we selected neuron oligodendrocyte labeling with new and holig 2 positive nuclei. We applied next-generation sequencing, and using a parsimony approach, we detect the species-specific differentially expressed genes. And as you can see here, the ratio between up and down regulated in human is higher compared with the other species, but probably you can point out that there is not much difference between neurons and oligodendrocytes. However, when taking into account divergence time between the three species, what we can see is that there is an acceleration of genes that are gaining human lineage compared with the other two species, more or less twofold. And more interesting is that in oligodendrocyte is even higher, suggesting that oligodendrocytes are subject to a more rapid acceleration of gene expression compared with neurons. And we then ask a simple question: what kind of function those human-specific genes have in this cell type? So what we found is that neurons, kind of boring, are implicated in synaptic etiology and presynapse, excitatory synapse, and so on. So very common synaptic etiology. And we know from independent study that this is something that is particularly under pressure in human evolution. However, what we found in oligodendrocyte is that, surprisingly and something unexpected, that human-specific genes are enriched for RNA alternative splicing. And these functions are specific for oligodendrocyte, are not found in neurons, are not found in other non-human primates, suggesting that alternative splicing is a function that is specifically evolved in oligodendrocyte. And we then ask basically a simple question: how can I relate a neuropsychiatric disorder with this kind of human specialization, in this case, gene expression? And basically, we take advantage of all the GWAS studies that are basically implicated in a neuropsychiatric disorder. And what we found is that genes that are associated with the human-specific genes in neurons and oligodendrocytes are enriched for genetic variants associated with neuropsychiatric disorder, which like ASD, autism, bipolar disorder, major depression disorder, and schizophrenia. And interestingly, none of the non-brain-related GWAS are enriched. It's kind of interesting. And when taking into account all the non-human primates differential expressed genes, we don't find any enrichment, suggesting that there is a link between the human-specific genes and genetic variants. And I would like basically to summarize what I found, again, is that oligodendrocytes are kind of cool from evolutionary point of view. And they're like, it seems to be undergone a gene expression acceleration in the human lineage compared with neurons. And we have like this kind of unexpected function in oligodendrocytes, since RNA metabolism and splicing are strongly associated with the oligodendrocyte human-specific genes. We found a lot of RNA binding proteins, and this is something completely unexpected and kind of interesting for us. And also we found that brain-related genetic variants are linked with human-specific genes, again, basically linking a neuropsychiatric disorder with some human specialization, in this case, gene expression differences. And I would like to acknowledge my boss, Genevieve Konopka. She's a great mentor and helped me to brainstorm this project, and also other postdocs from the lab that have like independent career in Japan, and also collaborators from Emory University and Georgia Tech. And thank you very much.
Thank you.
Hi.
Do you have any insights into the spatial or developmental patterning of these human-specific genes that you found, albeit in a single-cell setting?
It's a great question and something that I'm interested in because I would love to understand. I mean, we know that humans have a prolonged myelination, and we know that something happens also during development, as you heard from different speakers. And we analyze adults, and yeah, this is like an interesting question, and we're looking forward to answer this. Right now, we don't have like enough data to support and to. Yeah.
Could you step back, I think 2 slides? There.
Yeah?
Here. So it seems to me that the oligodendrocyte signal is coming from specific psychiatric disorders. It's not present in schizophrenia, for example. And it's present very strongly in major depression, bipolar disorder somewhat, and autism. So I'm kind of perplexed of why you presented this as referring to any psychiatric disorder, basically.
Basically here is like a sort of enrichment for genetic variants. What we have also, it's kind of hard to explain, but basically we have data from schizophrenia, from neuron and oligodendrocyte, and what we found is that the genes that are differentially expressed in oligodendrocytes, human-specific differentially expressed, are also enriched for basically downregulating schizophrenia, suggesting that there are like some different layer of regulation of those genes affecting. In this case here is genetic variants, and it's basically I use everything I could from genetic point of view, what we have from GWAS. But there is something else that's going on with the oligodendrocyte. Here you can see that schizophrenia is basically specific for neuron, but on the gene expression level, there is something changing schizophrenia in the oligodendrocyte. So,
Longer history of studies that have shown, mostly candidate gene studies that have shown downregulation of oligodendrocyte specific genes, but I mean that could also be completely consequent to treatment effects or other issues. So yeah.
Okay, let's thank Stefano once again. And please join me in thanking all of the speakers from this very stimulating session. Thank you so much.
Good afternoon, everyone. Thank you all for coming. Lovely to see you all here on the last session of the two-day event. I'm really excited to introduce the speakers that we have within this session. And the strand, I think, that kind of connects the talks that will be presented from the 4 speakers and the 3 trainees in this session is that they consider how these macroanatomical, cellular and molecular patterning of the brain during development constrains the unfolding of genetic risk for neuropsychiatric disorders. And as you'll see, there's a sort of diversity of talks that address this question in different phenotypic levels and across different model systems, including us. We have Ed Bullmore from University of Cambridge first, speaking about adolescent development of brain connectivity and networks.
Okay, so thank you, Armin, and thank you, David, for inviting me. It's been a very interesting meeting. And I'm going to talk about a range of stuff that we're doing in Cambridge to look at adolescent development of brain connectivity and networks. I should say at the outset, just to make sure you're aware of it, I work half-time for GlaxoSmithKline, which is a pharmaceutical company, as well as working half-time for the University of Cambridge. But nothing I'm going to say today has any commercial sensitivity. There's no GSK angle. I just wanted you to be aware of that. Now, we've seen this slide once already over the course of the meeting, and I just put it up to remind myself and others why adolescence is important for psychiatry. And it's because it's a particularly critical period of risk for the emergence of many psychiatric disorders. And therefore, it's a very important pathogenic question for us to understand what's going on in the brain, the human brain, during the course of adolescence. How might that process, developmental process of adolescence, be perturbed in ways which lead to various disorders? And we want to understand that not just at the level of behavior or imaging. We really want to understand that at the level of cellular and molecular processes which are likely to provide us with targets for intervention. So if we're going to do anything therapeutically, we need to have an integrated understanding of what's going on over the course of adolescent development. Now, I'm not going to present a completely satisfactory integrated account of human brain development over the course of adolescence, of course. What I'm going to do is just talk through some of the work that we've been doing, and it'll, I think, give you some hints of where we're hoping to go in the future. I'm going to start by talking about adolescent brain structural changes in connectomics, say a bit about this phenomenon of cortical shrinkage and how that might be related to intracortical neurogenesis, say something about that, how that relates to the connectome, and speak a little bit about new techniques that we've been developing to look at structural networks in individual subjects, which allow us to relate the connectome to individual differences in cognition and IQ. Then I'm going to put that human adolescent work into context, and then finish by saying something about linkage to genetics. So this is the main study that we've been running for a couple of years now, and let me just introduce the design to you briefly. It's an accelerated longitudinal design. That means that we have, over the age range 14 to 24, we've got these 5 age strata. We've got a balance of males and females in each stratum, 300 subjects in total. Everybody has a baseline scan and at least one follow-up scan 6 to 18 months later. This is a cohort subsampled from a larger age-stratified cohort of about 2,500 young people where we have genetics and cognition. And it's roughly representative of the healthy population of Cambridgeshire and London. I put healthy in inverted commas there because actually health, pure health, complete absence of any subclinical mental health symptoms is actually rather unusual in adolescents, we discovered. Only about 20% of that 2,500 have no subclinical symptoms, although they were all free of any history of treated disorder at the time of recruitment into the study. The imaging data, which I'll talk about, we collected multi-parameter structural MRI, multi-echo resting state fMRI, and some diffusion-weighted imaging. I'm going to focus mainly on the multi-parameter MRI technique. And for those of you not already familiar with this, I'll just introduce it. It's a sort of, I think, a very ingenious approach, which allows us in the same scanning session of about 15 minutes in duration to collect a number of different magnetic resonance parameters, R1, R2, proton density, and this MT stands for magnetization transfer. It's an imaging sequence that is particularly sensitive to the contrast between grey and white matter. I think you can see that here, and that is because it is particularly sensitive to the proportion of fatty versus watery molecules in each voxel. And the first thing we did, or one of the crucial things we did, was to look at MT and look at it not as a way of disambiguating central white matter from the cortex, but looking at it as a way, as a marker of myelination within cortex, intracortical myelination. So this is at 3T. You can see here's the surface of a single gyrus. Here's the grey-white matter boundary. You can see the individual voxels with variable levels of MT. By interpolating between voxels in a single region of interest, we can estimate the MT signal with subvoxel, sub-millimeter resolution, and we can see how that signal increases in intensity as you go deeper in the cortex from the peel surface down to the grey-white matter boundary. That is one piece of evidence to suggest that this is indeed a marker of myelination, because we know from histological studies dating back to the turn of the century that there is a much deeper, denser quantity of myelinated fibers in the lower level, in the lower layers of the cortex. So this MT signal obeys the prediction you'd expect from the histology. What do we see when we look at this developmentally? There's a very busy slide, but let's just start at the top. This is a map of cortical thickness, which, as you can see, is around about 2.6, 2.8 millimeters. It's thinner in the visual cortex and the primary somatosensory motor cortices, and that's a well-known observation. It also shrinks somewhat over the course of adolescence, and again, that is a anticipated and therefore here replicated observation, this phenomenon of cortical shrinkage. There's been some debate in the literature about what that represents, and it may be relevant to that debate just to take a look at what we see in terms of the MT signal. Now, I'm calling this myelination, but bear in mind it is strictly magnetization. But you can see there is increase of this MT signal again in primary visual and somatosensory areas, which is compatible with what, again, we've known for a very long time, dating back to Flexig, that those primary cortical areas are particularly densely myelinated early in development. Over the course of the 14 to 24-year period that we're studying, we can see that that myelination signal becomes stronger, particularly in association cortical areas. And indeed, the increase in myelination is negatively correlated with the change in cortical thickness. That is to say that areas that shrink fastest over the course of adolescence are also the areas which show the greatest increase in this MT signal. And that suggests that one possible explanation or one contributory factor to the observation of shrinkage may be a kind of partial volume effect that, seen through the prism of MRI, the cortex appears to shrink, at least partly perhaps because the lower layers of cortex are becoming more myelinated and therefore appear to a segmentation algorithm as if they're white matter rather than grey matter. So these are some sort of basic local observations, but of course we were interested in how they related to the connectome, and you've had a little introduction to the connectome already. This is a topological representation of a human brain network from an fMRI data set, which I just put up to remind ourselves that there are typically these modules or sub-networks in the system. There will be some hubs, shown here as the larger nodes, which are highly connected. Some of them may be highly connected only to other nodes within the same module. Larry yesterday made the analogy between a human brain connectome and the airline network, and these hubs shown here as squares are the kind of long-haul hubs, the equivalent of London Heathrow or LAX, if you will. They're the nodes in the network that do a lot of flights, but they also mediate most of the intermodular traffic, the intercontinental, if you will, traffic in the brain. So that's the kind of organization that we were interested in understanding, but we remember we're looking at myelination. So we need to start with structure. How can you get a connectome from a structural MRI dataset? Well, as some of you may know, there is a technique that's been around for a while. It's called structural covariance network analysis. And basically it works by parcellating the brain, measuring a single parameter, typically cortical thickness, in each region, and then correlating between regions. So here you have regions X and Y. You can see there's a correlation between those regions in terms of the cortical thickness, estimated over multiple subjects. Okay, so you need a lot of data for this, at least 100 scans to get a reliable estimate of correlation. And you repeat that estimation for every pair, you end up with this association matrix, you threshold it, and you've got a graph which you can topologically analyze and look at properties like the hub. Now, when we do that in these data, what we find, interestingly, is that here, first of all, is a map of the nodal degree, the centrality or the hubness of each of our cortical regions, and you can see, I hope, the darker colors, the bigger nodes, representing the hubs, particularly in the frontal cortical areas. Interestingly, those connectome hubs are the nodes that show the sharpest decrease in cortical thickness and the greatest increases in myelination. The same is true if you use an alternative measure of centrality or hubness. Instead of degree, this is closeness. It's very similar, and it shows the same relationship. So these brain regions, which locally are shrinking, perhaps because they're myelinating, but they certainly are showing an increase in magnetization transfer over the course of adolescence, are typically the hubs of the connectome. But we also want to understand not just how does local change in myelination or thickness relate to a sort of static representation of the connectome, but what can we learn about change in the connectome itself over the course of developmental time? So in another study, we took this sort of sliding window approach. So we constructed a structural covariance network using the methodology I've just described, not just once for each, for the entire sample, but repeatedly for subsamples that we estimated by essentially sliding a window from 14 to 24 years of age. So we ended up with 5 subsamples of overlapping age, and for each of those subsamples we could estimate the covariance network, and we could estimate some topological properties on it. And what we found was that over the course of time, the degree centrality in these frontal and other association cortical areas in particular decreased. So the hubs became less hubbier over the course of developmental time, and that was not a linear process. It was quite sharply nonlinear in many brain areas. And we could estimate both the drop and the maximum decrease in degree centrality, and also the age at which the degree, the hubness, reached a minimum. And you can see here in these two maps, this is where this is a map of the difference in degree over the course of adolescence, showing the brain areas that had the sharpest decrease in hubness, and this is a map showing the age at which the minimum degree was reached. And those were correlated with each other, so the greater the decrease in hubness, the longer it took for that particular brain region to reach its kind of plateau level of degree centrality. And this nonlinear process of developmental reduction in degree was greatest in the areas that showed the most rapid changes in magnetization. So those rapidly myelinating areas of the brain seem to be associated with this decrease in connectivity of connectome hubs. Now, that is quite interesting, but it has one obvious problem, which is that we've degraded the temporal resolution of the process that we're interested in understanding. We've had to aggregate data over multiple time points even to get this level of resolution. And that prompts a desire to have a technique for estimating structural connectivity networks that does not require tens or hundreds of subjects, as structural covariance network analysis does, but can be done on a single subject. And this is the work of, very largely, of Jacob Seidlitz. Professor Molnar yesterday said he thought the NIH Oxford Graduate Partnership Program was a great thing, and I agree with him almost entirely. It's not just Oxford that benefits from this program, it's also Cambridge, and it's really a terrific program. We have had many outstanding students come to Cambridge, and it's a great gift, actually, of NIH to the UK. And Jacob is one of those students, and his idea was that instead of trying to build a network, a covariance matrix, from a single observation, a single parameter in multiple subjects, why not use multiple parameters in a single subject? And of course we can do that because we've got these multi-parameter MRI data that I mentioned, plus we've got the diffusion-weighted imaging data that we also collected. So we've got multiple parameters on each subject. We can parcellate those images, and then from the parameters we've estimated, we can derive a number of features. We end up here with a 10-feature vector for each region. So you can see there's some markers of myelination, MT, and some of the diffusion-weighted imaging metrics. We've got some macrostructural parameters like grey matter volume and cortical thickness, and we've got some measures of curvature. So for each region in each subject, we've got a 10-length feature vector. And we can construct an association matrix simply by correlating between regions across that feature vector, which gives us an association matrix that we can render in this kind of block diagonalized way. We can threshold it to produce a graph which we can show topologically or in anatomical space. So this looks like an interesting technique. We've got a single subject way of getting a complex network out of human MRI data. But is it real? Is it valid? And that, I think, is a very important and recurrent question for all of us working with human MRI, where we are way above the ground truth of cells and synapses. I think it's important for us to continue to ask, particularly when we do these sort of rather sort of more adventurous modeling exercises and try and make maps of human brain network architecture, how can we be sure that what we're deriving from the data is really indicative of the underlying biology? And these are some examples of the way in which we basically tried to break morphometric similarity, to try and refute it, or more affirmatively, to try and convince ourselves that what we were seeing was plausible. And we took a couple of different approaches. One was to leverage the prior histological observation that brain areas that are cytoarchitectonically similar to each other are more likely to be axonally connected to each other. Therefore, we reasoned that if our method of morphometric similarity network analysis was biologically accurate, we should expect many of the connections to be between cytoarchitectonically similar areas of cortex. And that, in fact, is what we found. This is the morphometric similarity network for a single subject. The colors here represent cytoarchitectonic classes, and you can see, I think, that there's quite a lot of connectivity between nodes that belong to the same cytoarchitectonic class, rather more than there is in a comparative structural covariance network, and quite a lot more intra-class connectivity than there is in this diffusion-weighted network. You can see those data shown here as well. The other point of comparison, which alludes to something I'm going to get back to a little bit later, is that Jacob and colleagues were able to leverage macaque MRI data collected here. And of course, the great advantage of looking at the macaque through the prism of MRI is that we know the ground truth in terms of track tracing data, or at least we know part of the truth. So we could construct the morphometric similarity network from the MRI data on the macaque. We could compare it to prior track tracing data on the macaque, and it turned out, encouragingly, that those two measures were quite strongly correlated. So the strength of morphometric similarity as predicted, was positively correlated with the density of connectivity estimated by track tracing. And this is an approach that allows us to begin to look at individual differences and how, for example, individual differences in IQ might be related to the topology of brain networks. There's a prior theory out there, it's been out there for a long time, that particularly when it comes to more fluid or domain general aspects of cognition, it may be the integrative components of topology, the connector hubs that link the modules, that they may be particularly important in mediating or sustaining these kind of fluid intelligence functions. And that is roughly what we found here. If we looked at the relationship between IQ in these 300 young people, it was very strongly correlated with the degree, the hubness of nodes in the morphometric similarity networks in brain areas specialized for language and logical functions. There was also an association between performance IQ and degree, but it was in a anatomically distinct set of regions with distinct psychological functions. So we're beginning to think that this is a technique that could be useful in terms of understanding how networks relate to cognitive functions. It's not the only approach we have available to link these data to cognitive or indeed mental health related differences. And I just put this slide up to remind myself and us that here we have repeated measures data. Everything I've shown you so far is based only on the single baseline scans that we have for the subjects, but we have at least one other scan. So we can, in fact, estimate individual rates of change in myelination for each subject. And you can see it's quite noisy. Each of those connected points represents the intra-subject, within-subject difference in magnetization transfer over the course of time. But if you look at the relationship between these trajectories and the individual differences in terms of compulsivity, it turns out that there is an association, and the individuals with higher scores in terms of compulsivity show slower rates of change in magnetization, putatively intracortical myelination, particularly in these frontostriatal circuits that we know a priori are important for compulsivity and for obsessive-compulsive disorder. So this is pointing in a direction that we will continue to go over the next few years, I'm sure. Now, I just want to put that in context a little bit, because I've spoken quite a bit about 14 to 24. But, of course, life goes on before and after adolescence, and it's just worth remembering that we have the opportunity to look at life cycle changes in some of the same properties that I've already described, in particular, for example, myelination. And this is work in collaboration with a Norwegian group, several hundred individuals aged 8 to 80, using a different MRI marker of myelination, which is the ratio between the T1 and T2 times. And you can see that there's a curve, it looks a bit like life. It goes up to begin with, it reaches a plateau, and then it dips towards the end. That is the inverted U of intracortical myelination. It's obviously a non-linear process. So we can model it, and we can take the derivatives of a fitted function to that curve, and we can estimate milestones. We can find, for example, the time at which there is peak growth in myelination. We can estimate the onset and offset of that middle-aged plateau in myelination. And we can look at how those milestones vary over the course, over the surface of the cortex. And what we find is that for each of these milestones that I've mentioned, peak growth, onset of maturity, onset into the decline of old age, it seems like there are kind of waves. There's certainly a bimodal distribution for each of those milestones. So the the primary somatosensory and visual cortices particularly belong, have peak growth before puberty and an early wave. Most of the cortex comes later. And we can see those bimodal waves of change across all of those milestones. We can also see, going back to the connectome, and this is a map of the degree estimated from these data, the degree centrality in each of the nodes in the network. We can see that the higher degree is associated with the second wave of change in each of those milestones. So it looks like the connectome, which I suggested might mature later in terms of adolescent myelination, might also, those connectome hubs might also show later, more delayed rates of change in terms of reaching middle-aged maturity, and then be more resistant to the sort of attritional effects of later life. And this, I think, also is important for the future, and certainly is an important part of how we're thinking about adolescence in Cambridge, is we have an opportunity to work across species, in particular with the marmoset. And the marmoset has a lot of attraction for somebody that's interested in human adolescence, because it has quite a protracted period of adolescence itself. It has quite a large frontal cortex, and we can scan it. And we have here used a rather primitive scanner, but just to give you a sense of what we've done. These are, again, individuals scanned in a kind of an accelerated longitudinal design, so each animal has provided a couple of scans, and the age of the animals allows us to look at a period of time that collectively subtends sort of 3 to 27 months. We can take those data and use very similar modelling techniques to the ones I've already described to estimate the nonlinear trajectory of change. And here we're just looking at cortical volume, not myelination, but we can fit that nonlinear function using a technique called splines and estimate the derivative of that curve, which again gives us the opportunity to look at milestones of change in cortical development. So, for example, here you can see in an area of visual cortex, I think it is, or possibly auditory cortex, there's quite an early peak in cortical volume, which then declines, whereas in this area 17, the peak is flatter and more prolonged, and the decline is later. So we can estimate milestones, and we can see, for example, that there's considerable variability within the cortex. The frontal cortex shows later maturational change, as do the subcortical nuclei. And we can take a bottom-up approach and ask the question, are there clusters of cortical areas and subcortical nuclei that show similar developmental trajectories over time? And in this analysis, it turned out that there were convincingly 6 different classes or clusters of cortical and subcortical nuclei, which showed similar trajectories of developmental change to each other, ranging from this rather sort of peaky early change in here in the occipital cortex particularly, right down to this sort of slower, flatter, longer term change in the temporal pole. That's just I think a beginning of an approach that we hope to extend quite considerably in future. We have got a more powerful machine installed. We will be able to reproduce all the imaging sequences that we are doing in the human, in the marmoset, and as I think this example shows, many of the modelling techniques are applicable across species as well. Now finally, I just want to get to genetics, and there are many different ways you could try and link what we now know about gene expression, particularly in the human brain, to these kind of human imaging data. This is an example, this is inspired actually by Fenner Kreenan's work on this particular gene set. There is a set of 19 genes that we know are expressed particularly in supragranular layers of human cortex. If you've got a candidate gene or a candidate gene set like that, you can simply take from the Allen Institute or some other reference data set on brain expression the expression levels of the candidates you're interested in, map them, and correlate them very directly with whatever imaging phenotype you're interested in. Here we're just looking at the relationship between human supragranular gene set expression and the nodal degree in the structural covariance network. And you can see that the higher degree nodes have higher levels of this gene set. That's encouraging as a sort of proof of concept. Since we know that those genes are important in long-range cortical connectivity, it's consistent with the idea that the higher degree hubs, as I mentioned earlier, by analogy to the airline network, may be important, particularly in the long-distance intermodular connections in the connectome. But what if you don't know what gene or gene set you're interested in a priori? What if you're curious to understand the transcriptional correlates of an arbitrary imaging phenotype, and you don't want to be constrained to any particular prior hypothesis? You want to look over the whole genome, 20,000, 24,000 genes. This is obviously a very high-dimensional problem to solve, and a technique that Petra Vertes in particular has pioneered in Cambridge is using partial least squares to solve this problem. So partial least squares will essentially combine these two data sets and find a ranked list of genes that have an expression profile optimally similar to the phenotype of interest. So you get a ranked list of genes which you can then analyze in terms of their biological function. I particularly like this picture. This was taken, I think, backstage before Petra and Kirsty went on stage to pick up a Global Thinkers Award. I think it was Petra, Kirsty and Hillary Clinton who won it on that occasion. Anyway, this is what PLS does. More graphically, here's the phenotype we're interested in, which is the adolescent neurogenesis and association cortex. And the PLS result finds the combination of genes with an expression pattern as similar as possible to the phenotype of interest. And in this case, we can see that that gene expression profile shown here as partially square scores is highly correlated with adolescent myelination and negatively correlated with cortical thickness and enriched for risk genes for schizophrenia. I'm going to take two more examples, if I may, just to talk about the relationship between imaging and gene transcription, because I think it's likely to be of interest to this meeting. So this is a different starting point. So here we have correlated magnetization transfer intracortical myelination putatively with individual differences on schizotypy. Schizotypy, of course, is a personality trait that many people think of as a form fruste of schizophrenia and perhaps a marker of genetic risk for schizophrenia. And we found that there were a number of areas in the brain where myelination was positively correlated with schizotypy. Those brain areas had functions that we know are impaired in schizophrenia. And we tried to understand, well, what gene expression profile is most consistent with this cortical pattern of schizotypy-related myelination. We used the partial least squares technique that I've described. We ended up with a ranked list of genes weighted in terms of their importance in explaining the pattern that most closely corresponded to the phenotype we were interested in. And it was very interesting. You know, the positively weighted genes in this list, the ones that were most themselves positively correlated with schizotypy-related myelination, like this example, ANK1. You can see ANK1 expression positively correlated with schizotypy-related myelination. These positively weighted genes were very strongly enriched for the gene set that Dan Geschwind and colleagues published earlier in the year. We call it here the downregulated Gandal gene set was highly enriched in these positively weighted genes, whereas in the negatively weighted genes, it was the Gandal upregulated gene set that was positively correlated. Now, there are a lot of interesting details and implications of that result, but I again find it quite encouraging that if you rank genes in terms of their differential expression based on post-mortem data in schizophrenia and look at that in relationship to the weights on this PLS component, which we've derived from an in vivo study of a schizotypy-related imaging marker, they're quite highly correlated. I think that is suggestive that this approach may be picking out genes that are of real biological relevance to the imaging phenotypes we're interested in. Here's another example. This is looking at schizophrenia and using the technique that Jacob introduced, this morphometric similarity network analysis. So here's a map similar to the one I've already shown you, indicating where in the normal brain you have particularly high morphometric similarity, that is equivalent to high degree in a morphometric similarity network. So these are the hubs here in red. And these are the results from 3 different case control comparisons. Patients with schizophrenia compared to healthy controls. You can see there's a fairly consistent pattern of reduced hubness in the frontal cortex particularly. And if we apply the PLS technique to uncover what gene set might be most related to this difference in degree, we get a network with many protein-protein interactions enriched particularly for genes that are important in nervous system development and GPCR signaling, instantly also enriched for schizophrenia risk genes. So, a bit of a whirlwind tour, just to give you a sense of some of the things that we're interested in doing to try, again, to get at an integrated understanding of adolescent brain development that can help us understand from behavior through imaging right down to cells and molecules and genes what is going on during adolescence, what is going wrong, perhaps, in adolescent brain development in people with schizophrenia and the many other mental health disorders that have their peak incidence in that age range, and ultimately what can we do to intervene on that basis. I won't repeat all of that, but I do think we have an outstanding opportunity, and I'm not talking about Cambridge, I'm talking about all of us just at the moment, to understand how the brain is sculpted during adolescence and how that might relate to risk genes for mental health disorders. So finally, let me just thank the people in the lab who contributed to the work, the funders, and if you're interested in more detail about the methodology of brain network analysis, I can unbiasedly recommend this book as a possible source of further information. So thanks very much for your time, and I look forward to questions.
If you use the marmoset to define the changes in myelin or any other variable that you were detecting with the MRI and find important hubs of change and so on, have you gone back into the animals and done proper neuroanatomy to then understand what it is actually you are detecting with the machine?
No, we haven't done that yet, but that's exactly what we hope to do and plan to do. The study I showed you is really our first outing. So that was just to establish that we could see relevant developmental changes, but we've got a more powerful machine, and we'll do exactly as you suggest.
Yes, thank you for a great talk. My question is, there are reportedly sex differences in brain maturation. I was wondering if you disaggregated your data by sex or gender.
We've looked quite hard for sex differences in brain network changes, not least because there are some pretty obvious sex differences in terms of mental health over the course of adolescence. In particular, there's a sort of spike of depressive symptoms, at least in our data, epidemiologically, in young women from about 14 to 17 compared to men, have a very much higher incidence of depressive symptoms. So we have been looking quite hard for some kind of gender dimorphic change in brain network development that might help us to understand that. So far, we have not found it. In these data to date, gender doesn't seem to be a very important factor.
So that was a very excellent talk. Thank you so much, Dr. Bullmore. I was curious about if we could take a step back, since I know you were focusing on schizophrenia, but at adolescence in general, in terms of the later development of association cortex. What are your thoughts on since sensory development it would be obvious that sensory development precedes it, but why do you think there's such a delay between primary sensory area development and myelination and the association cortex?
It's always difficult to know the answer to the question why. And I can only hazard a guess. I mean, I would have thought for association cortex to have anything to kind of integrate, for there to be an associative function of those areas, there has to be prior maturation of more specialized cortical areas, primary sensory or primary motor areas. I mean, that seems intuitive. One could argue that there's a sort of finite metabolic budget available for development, and it has to be rationed, and the program has to be phased. It could be that the development of those areas is contingent on environmental exposures or other sort of age-related changes. I honestly don't know the answer to the question why, but what is clear is that the brain, particularly in those association cortical areas, is developmentally active well into late teens, in fact early 20s.
Here is Fenner from Harvard University, who will be presenting on genetic contributions to neocortical specialization in primate brains, some recent insights from single-cell transcriptomics.
Now everyone can read my email. Let's just get rid of that. Okay, great. Hi, everyone. It's great to be here. I'm a postdoc in the Mitochondrial lab, and today I want to tell you mostly about work that I've been doing in that lab for the last couple of years. But I want to start by just acknowledging the really thought-provoking discussions we've had so far on these questions about how far you can take an experimental system or a model in another species or even an organoid to explain features that we see in the human brain. And I think we all know by now that the human brain is unexceptionally large, and there are many other aspects of human brain organization that can be well predicted by what we already know from other species or can divulge from other species. And so in the spirit of that, my newer work is really focusing on this question of comparative aspects of organization and genetic contributions to that in multiple species. But I just wanted to start by motivating this talk with some older work, and really my interest in this effort started with some work that I did actually in functional neuroimaging in humans. So we know that we can't access the ground truth of connectivity in the human brain in the adult in vast majority of cases directly, but through interesting quirks of the neuroimaging signal, we can sort of start to infer connectivity patterns or coupling patterns across different brain structures. And one of the interesting aspects of neocortical organization as it pertains to coupling patterns or connectivity is that the association cortex, of course, has expanded relative to primary sensory areas, and it has this odd tendency that we've heard about a little bit already to couple to itself, that is to couple to other association cortex in regions that actually span large swaths of the neocortex. So you can sort of animate this property. So this is using resting state functional connectivity to sort of start to infer these properties. And I'm moving that little seed region, that ROI, around the lateral parietal cortex. And one of the things that you can appreciate is that the heat map, which is a measure of regions that are functionally coupled with the spontaneous fluctuations in the signal that we're measuring from that seed region, are distributed widely across the neocortex. And it seems to be the case that association regions tend to be preferentially coupled to one another. So prefrontal cortex preferentially coupled to the temporal lobe, and the prefrontal cortex to the parietal lobe, and so forth. So we can start to make maps of the type that we have been so far lacking in the human brain, but can really help us corroborate what insights that we already knew a long time ago from track tracing in other species such as macaques. So for instance, Pat Goldman-Rakesh in the 70s and 80s knew already that the association cortex in a macaque will connect across long distances, the parietal cortex to prefrontal and the temporal lobe and so forth. And we can start to color in this map in the human brain to gain similar insights. And one of the things that is striking about these maps is that regions or networks that are centered around primary sensory areas, so visual cortex, somatomotor cortex, and auditory cortex, have the tendency to be more locally coupled, whereas this association network, again, has this odd property of connecting across long range. And one of the questions that we became interested in is just this question of whether we can anchor some of these insights of topography or topology of coupling patterns with patterns of molecular variation or gene expression across the neocortex. So here's a general rule, though, and we've heard about this a little bit in previous talks, which is that there are really strong gradients. There's a fundamental sort of reflection of constraints of gene expression in the neocortex in multiple species, which is that nearby regions tend to have more similar gene expression patterns, and this sort of falls off with distance. And there's been a lot of interest in this rostral-caudal gradient of gene expression that stretches across the neocortex. And the question then is, are there exceptions to this rule, and are those exceptions somehow explained or predicted by the coupling patterns that we see with these indirect methods of estimating connectivity patterns in the human brain? And this is work that I did with Chet Sherwood, who's here at GWU. And so I'll just show you the punchline, which is that remarkably there are some, and it's a very small number of genes, but there are some genes whose expression is not easily explained by simple gradients or spatial proximity. So now I've taken, and these are taken from microarray data from bulk sequence tissue from the Allen Institute. But if you just order the regions by their rostral-caudal location, or if you just order them by their spatial proximity to one another, for this particular set of genes, and I'll show you who they are in a moment, you don't see the correlation structure following the spatial distance metric. But if you just rearrange them in a different way, and now I've taken all the cortical regions and I've rearranged them by their network order, and I've grouped all the sensory guys together in the top left, and then paralimbic, so these are cingulate and orbitofrontal regions, and then the higher order association regions, prefrontal, temporal, and parietal together, you can immediately appreciate then the correlation structure becomes more of a block design. And just to visualize what that looks like back on the surface, it turns out that regions can sit quite far apart on the neocortical surface, but by virtue of something that is similar within those cortices and potentially reflecting some broad tendencies for coupling patterns, long-range coupling patterns, you can see that the gene expression for these genes is quite similar, whereas regions that are much closer together on the cortical surface can have very distinct very distinct gene expression patterns for this particular set of genes. So who are these guys? Well, it turns out that in an extensive profiling in situ profiling of many different, 1,000 different genes from the Allen Institute, only 19 of these had a very peculiar property, which is to be expressed in the lower layers, and particularly layer 5 in mice, and they've shifted to the upper layers in humans. And that could mean that they're no longer expressed in layer 5, or it could mean that they have gained expression in the upper layers. But it's an interesting feature in a very small number of genes that do this. And again, since we've had this extensive discussion on what it is about the supragranular layers that seems to increase in the primate cortex or the human cortex, and what kind of molecular remodeling might be going on up there, that really gauged our interest and sort of brought us to this gene set in particular. And of course, we did some controls, so all layer 3 selective genes that are conserved across species don't show this property. Genes that have annotated function related to connectivity don't show this property in humans. It was really this selective and quite small set that have shown this shift in and diverged across these two species spatially that seem to have this relationship to broad topography of connectivity. So, and again, just that was interesting to us because, of course, we think of the upper layers as the main player, the main role, and Arnold Krickstein has mentioned this earlier today in being the sort of the basis for cortical connectivity. So then we can think of this as sort of an exception, an interesting exception to a general rule, which I think still, of course, applies, which is that some genes don't have more similar expression patterns by a simple distance rule. So that was pretty cool. But of course, there were many things that were sort of missing from that study in terms of our understanding of what these genes, why these genes shift, and what their actual relationship to connectivity is. And we didn't know in which cell types these were expressed, and importantly, we didn't know the extent to which this is a conserved feature of primates overall. So I wanted to get a better insight into the ways that neocortex differs across regions and across species, but now looking at gene expression at the single cell level. So you've heard in several talks now these powerful new techniques. So when I moved to the Mitochondrial lab, they had just published this technique for measuring gene expression in single cells in really high throughput. So I came to the lab with the idea of starting to build comparative data sets to really answer this question at the single cell level. And to date, we have now probably too many cells. We have millions of cells isolated from human, macaque, marmoset, and mouse brain tissue. So yeah, I'm using this technology to build out a comparative program, and this is only possible with a large number of collaborations. And the most critical one for me has been working closely with the Guoping Feng's lab at MIT to build a single-cell atlas of the marmoset brain. So we're taking many different cortical and subcortical regions from the adult marmoset, and we're trying to sort of get a sense of this question of cellular diversity and molecular characterization of that species. And of course, as compared to the other primate species that we have available. So this is all enabled by our participation in the NIH Brain Initiative Cellular Census Network. So our part of this is to, we're sort of the first non-human primate grant of this type, but we're trying to add to this understanding that has already been built out in, or is being built out in the adult and developing mouse brain and the developing human brain. So it's not without some challenges. I mean, generating high-quality data sets that involve isolating acute and healthy single cells is a challenge from adult primate tissue. In particular, it's heavily myelinated. The cells have been surviving for years before we try to take them out of the brain. And we're trying to adopt an atlas-guided or atlas-based approach to standardize our dissections and recovery of each cell type, so that we both have good biological reproducibility and get a handle on the technical artifacts that arise from these methods. So we can do this and dissociate fresh tissue into single cell suspensions. We have now mostly moved to single nuclei because of the scalability and ease of translating our findings to human brain. But we knew we were doing an okay job, at least with this in the primate, when we could see at least some of our cells seem to be pretty active. And these, I think, are probably ependymal cells that still have their cilia attached and sort of running wild. And these are dissections that I did right next to the, taking out the striatum, so you probably have a bit of the ventricle lining that was captured along with the suspension. But as I said, most of the data I'll be describing today come from single nuclei RNA-seq. So the first question that we can sort of motivate with these 4 data sets is taking a conserved cell type. So let's take upper layer projection neurons, or putative upper layer projection neurons, ones for which we know that from in situ work and from work in non-human primates and mice, that the broad classes of markers seem to be conserved across these within this particular cell type. And we can take it across multiple regions of each brain, and we can simply ask recalling some earlier talks that have really addressed this question of regional specialization. We can ask the question, well, to what extent do regional specializations in one species translate to the same specializations in other species? And one way to visualize this is, again, let's just take one cell type, upper layer projection neurons, in the marmoset, and color all the genes that seem to be most differentially expressed in that species in the prefrontal cortex relative to primary visual cortex. And then we can look at our other species, and in this graph, just to orient you, the dots that are above the 0 and that are in the upper direction are enriched in the same way as in marmoset in each species, and if you're going down below 0, then you're in the opposite direction. And what is sort of interesting to see is how conserved many of these differentially expressed patterns are within a given cell type across the primates. So 89%, 87%, so a vast majority of the specializations that exist in one primate species seem to be conserved in the other primates as well. and the picture changes sort of dramatically when you look at the mouse. And that's not to say that there aren't gene expression differences within a given cell type, particularly within excitatory neurons in a mouse brain, and that's work that is recently published by the Allen Institute, looking at ALM and primary visual cortex in that species. And they find many gene expression differences within conserved cell types across those regions of the neocortex. But the point is different here. The point is saying that for a given gene, the odds that you're going to be expressed in the same and specialized in the same cell type across the 4 species is basically at chance for the mouse relative to the primates. But within the primates themselves, you have a high degree of conservation. So we can start to sort of build out a list of the ways, and this is a simplified sort of cartoon and can maybe get you thinking about other ways in which the neocortex might specialize, either across species or across regions. And one simple way of specialization or changing aspects of the neocortical circuit is to just change the abundance of existing cell types. Another way you could do this is change genetic programs in existing cell types. The example I just showed you is an example in which you can change genetic programs within conserved cell types across different regions of a species, and then see the extent to which that is conserved across multiple species. Or you could invent, and this is maybe the most dramatic case, you could invent new cell types altogether, of course. So I'll just give you some examples of how we're thinking about doing these comparisons and how they might touch on some of those possibilities. So we've already been introduced to cortical excitatory neurons. And then, of course, there's a whole other main class of neurons, the GABAergic inhibitory interneurons, which are highly diverse in the neocortex. And I'm going to show you some examples now just from interneurons. So if we take all of our cortical interneurons isolated from the mouse brain and cluster them together by transcriptional similarity, what consistently happens is that they generally fall into 2 lobes by their transcriptional similarity, which, based on the gene expression patterns, you can divide into interneurons that likely arose from MGE origin. So interneurons are born subcortically and then migrate to the cortex. Interneurons that arose from MGE origin, and then those that came from other origins, which most likely are from the CGE, if you're talking about the neocortex. And if we're interested in changes in proportions, we can simply compute the relative fraction of interneurons that come from each of these different sources. And in the mouse brain, and this I think is corroborated by stereology, it's remarkably consistent across the cortical regions that we have measured. So the fraction that come from the MGE, and to just give you an intuition of who they are, they're the somatostatin positive and parvalbumin types, are about 62% in across cortical regions that we've dissected. And so you can simply reflect that ratio as the ratio of MGE derived in both regions. And then what is interesting with the primates is that, and this is sort of known for expected from the literature, is that the ratios don't stay balanced in the same way across multiple cortical regions. So the proportion of MGE-derived interneurons decreases in primate association cortex. And I say association cortex because it doesn't seem to be just selective to prefrontal cortex, but rather is true of temporal and parietal association cortex as well. So that's one simple way. It's not simple developmentally, but it's one simple way that you can specialize cortical regions, is just change the abundance of existing cell types. Now, what about changing genetic programs in existing cell types? So we had one example from upper layer projection neurons already. But here, staying with cortical interneurons, one of the nice features of this study is that it made us realize that the main cortical markers of cortical interneurons that we know from mouse are basically still conserved in the other species as well, in the primates. And they basically form 4 non-overlapping, mutually exclusive categories of interneurons. So that turns out to be very useful. You have your parvalbumin and somatostatin MGE-derived types, and then your other types, neurogliaform, AID2 positive, and VIP positive types. And those 4 markers pretty much exhaust the possibilities for your cortical interneuron flavors in each of these species. So once we have that sort of partition, we can start to ask, well, how flexible is our gene expression programs across these 4 canonical types then? And one interesting feature about being able to look at this across the whole genome is that you can start to appreciate how rapidly and how dramatically some of these changes or switches can occur even within markers that we have good intuition about in the mouse. So for instance, the neuropeptide Y gene, NPY, is expressed mostly in somatostatin-positive cells, but actually also a little bit in neurogliaform and a bit in VIP-positive cells in the mouse. But in primates, it seems to be quite selective to somatostatin-positive subtypes. Another example is netrin G1, which is preferentially expressed in parvalbumin-positive interneurons in mouse, and in sort of a dramatic flip, is pretty selectively expressed in neurogliaform subtypes within the primates. And I just pulled this example because many of us know this gene from the neuropsychiatric genetics literature. Of course, CACNAs are voltage-gated calcium channel subunits, and some of them have known associations with schizophrenia and other major psychiatric disorders. And I give this example not because it's a dramatic example of a total flip in specificity, but rather that if you were building your intuition for CACNA1E based on the mouse alone, you would be tempted to say it's very specific to somatostatin populations, but actually in the other primates, you can see what is probably more striking, at least from the human, is that it's selectively depleted from parvalbumin-positive subtypes. And then there are very vanishingly small examples, or few examples, of classes of genes that are completely depleted from the mouse or from one species and arise anew in other species. And one example was published in the literature a few years ago of osteochrin, which is a known gene in osteoblasts in the bone and is expressed in the mouse in bone, but is apparently absent from the brain in this species, and yet seems to be a specific marker for parvalbumin-positive interneurons in primates. Okay, so those were a couple of examples of how you can change genetic programs and the sort of kind of dramatic examples of complete switches among subtypes of even known or disease-related genes. And how about this last possibility? So how about the possibility of inventing new cell types altogether? So do we ever find any evidence of this? This is, again, focusing just on cortical interneurons. So the previous examples take as an assumption that we can divide up the cellular space or the subtype space equally and apportion similar types across species with known markers. But of course, what you'd really like to do is have an unbiased way of sorting cell types or cells together and determining to what extent the same types are conserved across species. And so we sought a different approach, an unbiased approach of aligning transcriptomic signals across different species and different data sets to allow the possibility of cell types that are either not conserved across species or that are greatly expanded in one species relative to another. And so the tool that we used was canonical correlation analysis. And so that is the plot on the left, and the plots on the right are actually independent component analysis, so ways of partitioning single cell, dimensional reduction of single cell data sets within each species. And I've simply colored the cells together that the CCA analysis would have called or would have put within the same cell type or cell class. And when you do this, you can find correspondence between many of the cell types that we already have intuitions for and that we know and we can even give names to. But when we did this, actually one cell type in particular stood out. And what was interesting about this type is that it seemed transcriptionally very similar to existing flavors of neurogliaform cells. So there are two main classes within both primates and mice. But this third category expressed additional markers that we've never seen before in cortical neurogliaforms in the mouse. And this is work done with Gord Fischel, who's recently moved to the Broad Institute in HMS. And so we're really interested in this feature because when we looked, this was discovered first in our analysis of marmoset to mouse, but when we looked in the other primates as well, we found that they represent a relatively constant proportion across all of the cortical areas that we had measured in all of the primate species, and were virtually absent from the mouse. I won't say totally absent because we do see a very small proportion of them sort of hugging the white matter in layer 6B. But they're a vanishingly small fraction in that species. And we did some in situs, and we're characterizing their density and distribution over the lamina, but I'll just show you one zoom-in shot of the upper layer 2, where you can see examples from the marmoset of this type. And so we wanted to know, well, simply where do they come from? How do you get a new type or a vastly expanded type in other species? And one of the insights came from a point that has been raised several times today, which is well known, which is, of course, that interneurons are not born within the cortex itself, but rather migrate from proliferative zones subcortically within the ganglionic eminences. We talked already about MGE and CGE as different sources of the different flavors of interneurons that we know of. And this is work done by in Arnold Kriegstein's lab as well as in Gord Fischel's lab, making the point that when these cells are born within these subcortical structures within the ganglionic eminences, they don't just migrate, of course, to cortex, but they can migrate widely to other structures as well, including the striatum and the hippocampus. And so we wondered, if we don't see them in great numbers in the mouse cortex, might there be a possibility that we see cells of this flavor, of this type, in other structures within the mouse brain? And so we'd recently published an atlas of the adult mouse brain at single-cell resolution across 8 major structures of that species. So that includes the cortex, of course, as well as the cerebellum, the thalamus, the striatum, the hippocampus, substantia nigra, and entopeduncular nucleus. And so we could use this large data set, curate out all of the interneurons from each of these structures, and simply ask if we use a canonical correlation analysis to pit the cells that we're interested in the primates against all of these possible structures and sources of interneurons in the mouse, where might they come from? And the answer was unambiguous, which is that these cells uniformly and preferentially map to a single cluster of mouse interneurons. And I'll even tell you who they are. It turns out that the vast majority of them come from the mouse hippocampus, with a very small number, again, as we see from our in situs, there are a small number of cells that share these common set of markers in the mouse neocortex, but they make up such a small fraction as to be negligible. But we actually see a large number of them in the mouse hippocampus. And so what this suggested to us is actually that there's an opportunity to look, that being able to collect regions across multiple, data across multiple structures is actually a powerful way of starting to understand what may be shared origin relationships, at least for the interneurons that we're interested in. And it turns out that in primate brains, there are really closely matched types across the hippocampus and the neocortex. And the newer expanded population that we had found within the primate brain falls in quite nicely in this concept of having matched populations in each of these two structures, which of course is not the case that you see in the mouse. And in general, we think of the id2 population, the neurogliaform population in the neocortex, as arising from the caudal ganglionic eminence. But in this case, and we've known this for a while in the mouse hippocampus, that a large proportion of neurogliaforms comes from the MGE. It seems to be the case that in the primates, the neocortex may have co-opted or repurposed a population of MGE-derived neurogliaforms, and they now happily settle in multiple layers, both in the upper layers and in the infragranular layers of the primate neocortex. So we can maybe update our list and say maybe it was a bit of a tall order to ask for a complete invention of a new cell type, but rather an easier way of making changes to the sort of canonical microcircuit within the neocortex is to simply repurpose types from existing structures. So I'll just conclude by giving a sort of summary of what I've shown, the work I've shown today, which is there are many ways that the neocortex can specialize within a species or across regions. I showed you some work that linked gene expression patterns to something about network topography, or perhaps, as Ed Bullmore said, something about the cytoarchitecture of regions that may lie very far apart from one another but are somehow linked in terms of their molecular composition or potentially their developmental progression. That genetic programs in specific cell types can vary across cortical regions and, of course, between species, but knowing something about the relatedness of species can give you some more insight into in which direction these expression patterns changes might go and how they might be conserved across more closely related species. And finally, that a very flexible way of introducing changes to the cortical microcircuit might be repurposing or recruiting existing cell types that were not in this structure before within certain species. And with that, I will thank you guys so much for listening and take any questions.
Very nice talk. I was wondering if that last group of cells that you said were repurposed still exist in the human hippocampus?
They do. They exist in the human hippocampus. They exist in the marmoset hippocampus. And so once we found them in the mouse hippocampus, of course, the obvious thing was to look within the same species directly. And we find that they have very similar gene expression profiles within the two structures within the same species.
So about the CG part of the talk, the comparison between mouse and human brain, or mouse versus primate brain. The other feature of the CG neurons is they're really highly biased toward the superficial layer. I wonder that the proportion of the increase of the CG neurons in more associated layer areas, whether there is also even more exaggeration of the CG neurons being more concentrated in the superficial layer versus a deeper layer. I wonder whether you checked that.
Yeah, that's exactly the prediction that we had, and we're doing the in situs now to do the quantification. But that's exactly the prediction. So the MGE-derived interneurons tend to populate the lower layer. I didn't say this, but the lower layers of the neocortex, and are also born earlier and migrate earlier. We saw some of this work already. And it's the later wave of CGE-derived interneurons that tends to populate either all cortical layers or preferentially the upper layers. And so one prediction might be is that if you're already messing with the upper layer compartment, particularly within association areas, you might need to do some population matching with interneuron subtypes as well. And then the clear prediction would be that they'd be derived from CGE.
In the genes that within that group of interneurons that is from the mouse hippocampus that is equivalent to the primate superficial interneurons, is there any clue from the genes that are different in terms of possible clues for the difference in migration, or they have to connect to different cell types, so there must have been also other genes that are different, right?
Yeah, so I sort of emphasized that the gene expression differences were quite minor between the hippocampus and the cortex of these matched neurogliaform subtypes, and even true for this novel or expanded population in primate neocortex. But there are some gene expression differences. And I think one of the frustrations, of course, is that we're looking in adult tissue, so we don't really get to know or maybe retain the migration signal that we would hope to see, because the obvious question is where they come from and how they choose to migrate to the cortex rather than the hippocampus, if indeed they come from the common lineages, which is a common lineage, which is the prediction. But one of the most interesting things about them is that they express the transcription factor NKX 2.1, which in the mouse should be completely absent from the neocortex, and in fact is known to play a role in biasing migration of interneurons to the striatum rather than to the neocortex. And so for some reason, these guys have managed to retain this transcription factor, which leads to a cascade that that biases them, that should bias them away from the striatum once NKX 2.1 is downregulated, which is involved in growth cone collapse as you're migrating across this around the striatum from the MGE. But for some reason in the primates is retained on and even into adulthood. So we're wondering whether that pathway can give us some insight into why they both retain NKX, manage not to fall into the striatum, and then are integrated into both lower and upper layers in the neocortex.
This is related, I think, and I'm not sure I have this right, but in a coronal section, to get to the hippocampus, the medial pallium, you sort of have to go through the dorsal pallium or go right by it at least. And then in the mouse, it ends up that this subclass of neurons is only in the hippocampus, so it never makes that radial reach into the dorsal hippocampus. So is it possible that the mouse is the weird species here?
Yeah, so now we're actually doing in situs in the ferret right now, just to make sure. I don't think I ever said that this is a primate-specific phenomenon. We're hoping that it's not a question of just losing it in rodents or in mice in particular, and it's present everywhere else. But we're using an outgroup to confirm that. We've tested this now in multiple strains of mice, and we don't see them in any of the strains that we've tested. But of course, it remains a possibility that that mice are the weird ones, which is probably not the first time that has happened.
Are there any cardinal markers such as the HT3A receptor, 3C receptor, that are present in mouse and are absent in human?
5-HT3AR is a great example. It's completely absent in CG-derived primate interneurons in the neocortex.
And how do you explain that? Because up until now, we've been hearing in this seminar more and more that the cardinal stuff, the stuff that is VIP, parvalbumin, and so on and so forth, is really maintained across species. But this particular one that seems to be even higher in the hierarchy than VIP, for example, is actually lost.
No, I can't explain it. I really can't explain it, and I don't know from a developmental perspective whether it's lost over time, but it's a feature of all three primates that we've tested that we don't see that as a marker. It's expressed in maybe one cell in 100,000. So it's not that it's not well annotated in the genomes, especially in the human genome. So we're not really sure why we have sort of an absence at the upper layer of such at the upper level of such a cascade that should determine fate. We also, I think, lack really good markers for CGE-derived fate and transcription factors that are really specific to CGE fate. So it's been, while it's gratifying that those 4 canonical markers are conserved in the types of neurons that we expect them to be conserved, really being able to say for sure that they all originate from the CGE in the primate case is something that we're still working on.
Yeah, now thanks for a really interesting framing of this problem. I'm curious whether it seems like, at least from the initial work you're doing, is looking for things that are really quite qualitatively distinct. And I'm wondering if you find differences, regional differences in the proportions of very specific cell types to each other, because that would have a kind of profound impact on local circuits.
No, I agree with you. That's a great question. And the example that I showed you is a pretty dramatic change and a pretty blunt change of just proportion of MGE to non-MGE derived interneurons. But of course, then you can make that a more granular comparison. And we do find fluctuations that seem to be stable within a species, but differ across species, again, with the primates tending to be more similar to one another than to the mouse. We know that, of course, and there's something that Barb Finlay said in her talk, there's an overabundance of GABAergic interneurons in the primate neocortex, and we also recover that in our data. But moreover, the fraction of CG-derived and subtypes within CGE seem to be quite flexible within, although stable within a species, quite flexible across species. So that is a dramatic, fairly simple, but a very dramatic way to change the microcircuit.
Yeah, back here as well. Just a comment and question. The comment is, I think it'd be good to be cautious about immediate early genes, or in which there are many. And depending on what one animal is doing beforehand, some genes might be activated in some brain area, not the other. I doubt that's what's happening here in this mouse, but I think it's worth considering.
Yeah, well, are you thinking about OSTN? Because osteochrin was actually in the study that was discovered in the macaque, was shown to be sort of an immediate early like gene in that it was very responsive to a visual stimulus or the previous presentation of visual stimulus in the macaque right before sacrifice. Here we can show that if that is true, if it is really activity dependent, it's activity dependent within a very specific subtype of interneurons. But I absolutely take your point, and we can't really control as well as we might like to for prior experience immediately before sacrifice for all the
so for all of our experiments except for humans, we keep them in the dark in a quiet state beforehand, so we have baseline across species. But I'm wondering, in the initial part of your talk, you showed two different cortical areas that have similar gene expression profiles that are far apart from each other, and wondering, have you looked into whether or not, I know it's human, but thought about whether that could be due to different, the same developmental origins, or they converge on a similar expression profile in different parts of the brain?
I know. I think that's really one of the, that's the million-dollar question for me. And I know that people like Arnold Kriegstein and others who are actually actively working in neurodevelopment are trying to ask this question of regional specialization. And one of the things that he was able to show is that you have sort of lack of diversity of progenitors that arise from different regions of the neocortex. But as the excitatory neurons become post-mitotic, then you start to see these differences emerge. And we see that also in our developmental series of human prenatal data. But yeah, that is the question: how you get regional diversity, a regional specification of at least of post-mitotic neurons, and then at the same time, at the beginning of the talk, showing you that there are some cases where, at least as far as we can measure in bulk, that the global gene expression profile of cortical regions that seem to be somehow related, either in terms of cytoarchitecture or maybe in terms of connectivity, maybe ontogeny, have similar profiles. So we're talking, of course, about different sets of genes and different methods of specification, but I think that's really the open question that we all need to work on.
If there are no more questions, I hope you will join me in thanking
Hello everyone, thanks for coming back for the final session. Excited to have Genevieve Konopka here, who will be presenting cell-type-specific transcriptional networks regulating striatal development and behavior. Thank you very much.
Thank you, and like all of the speakers have said, I just want to reiterate to the organizers, thank you for an awesome two-day symposium. I'm really honored to be among the speakers. And my talk today is going to be a little bit different than within the session and even across the 2 days, because I'm going to talk about the striatum. We haven't really heard a lot about the striatum in this symposium. And although my lab works, more than 50% of our projects are human or non-human primate related. This particular project is focused on work in the mouse. But hopefully I can convince you that it's quite important and potentially relevant to understanding human neuropsychiatric diseases, in particular autism. So the large, big question that my lab is interested in addressing is trying to identify the molecular mechanisms underlying cognition. We do take an evolutionary approach to a lot of our studies, asking what features of cognition might be unique to human, and are these features perhaps related to the emergence of human-specific disorders, in particular autism. And if you heard my postdoc's 5-minute talk, he kind of mentioned this hypothesis that's been around in the field for some time that we're following up on with molecular studies. Okay, so there have been many features that have contributed to human evolution, but I would like to argue that language is perhaps the most compelling and maybe unique human feature. And Eric yesterday talked a lot about language and how there are definitely conserved, in the vocal motor learning aspects across species. But I think if you took that whole pie that he presented, one could argue that there's something kind of unique about human language. And in addition, language is frequently disrupted in many cognitive disorders, including autism and schizophrenia. So a number of it's been some time now, almost 20 years ago, we made some headway into trying to understand molecular mechanisms or brain developmental patterns that might underlie language by the uncovering of the gene FOXP2 being related to speech and language disorders in humans. And again, yesterday you heard from Eric some information about FOXP2 and its relationship to vocal motor learning. And so we feel that this is an entry point into really understanding brain developmental patterns that underlie this, what we think of as this human unique feature, and may also give us insight into these neuropsychiatric diseases. And so a lot of the work in my lab has been focused on trying to understand the role of FOXP2 in the brain, particularly in the human brain, and it's a transcription factor. We're a genomics lab. We've been trying to understand transcriptional targets, human-specific targets of FOXP2. However, what I'm going to speak to you today has some link to FOXP2, but is more about the related transcription factor, FOXP1. And I got interested in FOXP1 a number of years ago because it had been shown that it's highly homologous. Again, it's another transcription factor, highly homologous to FOXP2, and that these two transcription factors could heterodimerize to regulate gene expression. And so I thought it would be really cool to try and understand what are the shared versus distinct transcriptional targets of these two transcription factors in vivo in the developing brain. Well, that particular study has been quite challenging to flesh out, although we are continuing to work on that and have a couple of projects related to their interaction. But what I'm going to focus on today is just our work on FOXP1. So when we look at where and when FOXP1 is expressed in the human brain, we get a pretty interesting picture. And these data are derived from NANOD's human brain transcriptome database. And the solid line here indicates birth. So hopefully what you can appreciate, there's a peak of expression of FOXP1 in the developing human brain. And as we have heard throughout the day, this is a very important time point in human brain development. And as Dan mentioned in the morning, there seems to be this convergence of genes related to neuropsychiatric disorders, in particular autism, that are important during brain development at this period. Each of these colored lines indicates a different brain region, and the black line here indicates expression in the striatum. So FOXP1 has its highest expression in the developing human brain during mid-fetal gestation in the striatum. So to me, this was very interesting because, as you've already heard today, there's been a lot of focus on the neocortex as it pertains to various disorders and related traits. So while I was setting up my lab almost 10 years ago, a number of studies started to emerge. While I was initially only interested in FOXP1 because of its interaction with FOXP2, it turned out that if you have mutations in FOXP1, you have increased, or you almost certainly will have a diagnosis of autism and or intellectual disability. And using more recent data, we saw this graph yesterday from John where he talked about TBR1 of these high confidence de novo genes. FOXP1 is among these high confidence genes. So that list he mentioned of 85 genes, FOXP1 is among it. And I've seen recent presentations this year of unpublished larger data sets where FOXP1 really rises kind of to the top of these candidate risk genes. So we think it's really important, a really important single gene involved in autism. And if you look at where the known mutations occur, they don't cluster specifically, for example, in the DNA binding domain, but they really are found throughout the gene. And they're all primarily loss of function, though there's at least one study showing a duplication of FOXP1 in a patient with autism. And so in terms of autism spectrum disorder, what you'll probably hear from clinicians, or if you're a clinician yourself, you know for the most part, with idiopathic autism, every case of autism looks different from the next case. However, what the clinicians who have been studying these patients with FOXP1 mutations have noted as they've uncovered more and more individuals with mutations in FOXP1 is that they all tend to look very similar in a number of features, so much so that a recent review was going as far as calling this FOXP1 syndrome. So what are the shared features? Well, hopefully, as you can appreciate from these photographs, they have quite distinct craniofacial phenotypes, but that are similar among the patients. What has been noted that they all have language delay, and in fact, I was talking to Joe Boxbaum's group lately, most of them or all of them don't have any spoken language, absence of language. Of course, they're almost all diagnosed with autism, and some are also comorbid or co-diagnosed with intellectual disability. But one of the other statements from these recent reviews that struck me, thinking about the expression pattern of FOXP1 in the human fetal brain, was the statement that all individuals with these FOXP1 mutations are delayed in achieving motor milestones. And I thought this was really important, thinking about FOXP1's potential role in striatal development and function. In addition, there had been a paper published a few years ago from Gudrun Rappold's group in Germany, where they had made a Nestin-Cree conditional knockout mouse for FOXP1, and the major neuroanatomical difference they reported was this significant reduction in striatal size. And so what we had decided to do was to take, a number of years ago, a different approach and make what we called a patient-relevant FOXP1 mouse model. And so we basically characterized the full-body heterozygous mouse, which we thought would mimic the haploinsufficiency found in the patients. And this was published, so I'm just going to go quickly through this because it sets the stage for the unpublished data that I'm going to show to you. So one of the first things we wanted to do was show that there was a reduction of FOXP1 expression in brain regions with known FOXP1 expression. What was really, and so this is quantification of some immunoblotting. What was really striking was that in the cortex, we didn't really see a significant reduction in the FOXP1 heterozygous, even though it's highly expressed in layers 2 through 5. And so last year, my lab published a couple of papers on the role of FOXP1 in the neocortex. This, to me, suggested that it's so important in the cortex, there was upregulation on the other allele, and in fact, we've shown it's important for cortical migration and learning, spatial learning, et cetera. The hippocampus had roughly 50% reduction, as you would expect for a heterozygous mouse. The striatum had at least 50% or more reduction in the striatum. Since we went to the trouble to make a mouse model, we can study behavior. I know yesterday we talked a lot about pros and cons of trying to investigate behavioral phenotypes in model systems. These mice, for the most part, are quite normal when we put them through a battery of behavioral tasks. However, there was one phenotype, if you will, in these heterozygous mice. We measured pup ultrasonic vocalization. So this is the task where you take the young pup away from its mom, it gets cold, and so it cries. It has nothing to do with language. It's not learned, it's innate, but it's a nice way for us to measure motor function in a young mouse pup. There are only a few tests that we can really do at this age point if we think this gene might be important for early development. And what we found was that the heterozygous mice had a significant reduction in the number of USVs emitted by the pups, as well as a number of other features. Why it's also interesting is because model systems of FOXP2 in mice have also shown similar phenotypes, and this gets back to this idea of how these two transcription factors might be working together to regulate brain developmental programs that underlie perhaps behavioral traits. Now, of course, we want to understand, it's a transcription factor, FOXP1. We want to understand what are the gene expression programs downstream of FOXP1. And so we carried out RNA sequencing in these 3 brain regions. We didn't see anything changing in the cortex, not surprising because there's no reduction in FOXP1. But we were able to identify genes changing in the hippocampus and striatum. We carried out WGCNA. You've heard about WGCNA in several talks today. And because this is published, I'll just kind of summarize the main finding. We were able to identify a number of modules that were, the eigen gene was related to a striatal gene expression. And these modules were enriched for genes involved in ion channels and dopamine receptors. And so we thought it would be really important to see if there was a functional consequence to reduction of FOXP1 in the striatum at the level of neuronal function. And so to do this, we teamed up with Jay Gibson at UT Southwestern, who does electrophysiology, and we crossed the heterozygous mouse models to reporter mice that would mark the two major cell types in the striatum, the D1, DRD1 or DRD2, expressing spiny projection neurons. And what we found was quite striking. When you reduce FOXP1 in the DRD2 SPNs, not MSN anymore, according to the folks who study striatum, there was a significant increase in intrinsic excitability. In contrast, when he carried out the same experiment in the D1 SPNs, what we found was, in fact, it's not significant, but it was tending to hypoexcitability. So this suggested to us that FOXP1 may be having different roles in these two major cell types within the striatum. In addition, thinking about this idea of how could these two transcription factors perhaps be regulating these two cell types, maybe we took our differential gene expression in the FOXP1 heterozygous mouse. We intersected it with publicly available data from Wolfgang Enard's lab, where he had made a FOXP2 heterozygous mouse and did RNA-seq in the striatum. We found a significant overlap of genes, suggesting that there indeed may be shared targets. We further intersected that with publicly available data that had indicated genes that are more enriched in the D1 SPNs versus the D2 SPNs. And what we found was that the FOXP2 targets tended to be more enriched in the D1 SPNs, whereas the FOXP1 targets were somewhat equally distributed. And this fits with the known expression patterns of these two transcription factors. FOXP2 has more enriched expression in the D1 SPNs, whereas FOXP1 has equal expression in these two major populations. It also is not, I should say, is not expressed in any other of the cell types in the striatum. It's not expressed in interneurons or in non-neuronal cell types. Okay, so this is my only slide actually using human materials. In the lab, we also grow human neural fetal progenitors. So these are cells derived from discarded fetal tissue. I think, Dan, some of the work presented today this morning uses the same model system. We can manipulate gene expression in these cells. We can differentiate them into post-mitotic neurons. So we did that. We manipulated FOXP1. We carried out RNA-seq. We did the WGCNA. And then we carried out what is known as, many of you in the room know, module preservation. And we basically intersected our human gene expression data with the gene expression from the mouse in the hippocampus and striatum. And what we found was that the genes that we had identified in the mouse that are strongly regulated by FOXP1 tend to be more preserved with the human data. Again, this together really focused on FOXP1 regulated gene co-expression being important in the striatum, and that we could make this correlation to human data set, I think, was very important for us. In addition, if I need to further convince you that the striatum might be important for autism, there have been a number of studies that have integrated genetic variants in autism with gene expression in various cell types. You already heard this morning about enrichment in cortical neurons, but this recent paper from Evan Eichler showed that de novo mutation signals are enriched in genes with striatal neuron expression. In addition, a paper a few years ago from Dennis Vitkup, not only did he note the enrichment in the cortical lower layer cortical neurons, but also in the D1 and D2 spiny projection neurons. So this was important for us that we could make the case that the striatum is indeed perhaps important for autism pathophysiology. But from the clinical side of things, there's also supporting evidence. So there's definitely this prevalence of motor abnormalities in children with autism. MRI studies have shown changes in striatal volume. And fMRI studies have found reduced striatal activity and connectivity in social reward processing tasks in patients with autism. And there are some nice recent reviews that talk about the role of striatal circuits in autism pathophysiology. So just to remind everyone in the room a little bit about striatal circuitry, there are two kind of main distinctions in the striatum. So as I've already mentioned, there are two major cell types. the direct pathway that contains the dopamine receptor 1 expressing spiny projection neurons, now being called DSPNs, and then the indirect pathway containing the DRD2 expressing neurons that are now being called the ISPNs. And these cell types are both expressed within two neurochemical zones, the matrix and the striatum. A lot of this work has been pioneered by Anne Grable and a few others in the field. And I would like to argue that while there's been a lot of work on circuits and identifying, work by John and others in the field, I think we still don't yet know all of the molecular mechanisms underlying the specification of these organizational features in the striatum. And so based on our finding of this differential activity of spiny projection neurons with loss of FOXP1 in one cell type versus the other, we hypothesized that FOXP1 may regulate striatal signaling pathways in a cell type specific manner. In particular, we thought that it might be very important for the ISPNs, or the D2 expressing SPNs, and that these cell types might be particularly at risk in autism. And so to address this hypothesis, a very talented graduate student in the lab, Ashley Anderson, decided to make a number of conditional knockout mice. And so she removed FOXP1 from the D1 SPNs, from the D2 SPNs, and then made a double conditional knockout mouse. And so this image here is just showing a reduction of FOXP1 by RT-PCR and also a reduction by immunoblotting. This is from the double conditional knockout mouse, and you can see basically a complete reduction of expression of FOXP1 because, as I already mentioned, it's only expressed in these spiny projection neurons, and these Cres are quite efficient. And I'll show you a slide to that effect. Okay, so it's a transcription factor, so we want to know what are the cell type specific transcriptional targets downstream of FOXP1. And so in order to do this, we carried out single-cell RNA sequencing. We used the 10x genomics platform. We had 4 animals per genotype, and we chose for this study postnatal day 9 for this experiment. This was for a number of reasons. One is because initially when we started doing single cell a year and a half or 2 years ago, we were using whole cells. And it's really hard to do that from adult animals, as we just heard. And now we're only doing nuclei, but these data are from intact cells. In addition, all of the electrophysiology that we had done was around P18. So we wanted to capture the gene expression patterns that were before the changes we were measuring by electrophysiology. Okay, so in total from these 16 animals, we were able to obtain over 60,000 cells that passed all of our QC. And we were able to find all of the major cell types that are known to be found in the striatum. And this is on par with, for example, roughly the proportions that have been found in the adult striatum by Steve McCarroll's group and Stan Linnerson. If you look at the distribution among the gene expression from the 4 genotypes, hopefully what you can appreciate here, they're color-coded by control, D1 conditional knockout, D2 double conditional knockout mice. They're equally dispersed among all these major cell classes. And this was reassuring to us because we weren't hoping to find, a major kind of restructuring within the striatum. Other QC is just showing that the median number of UMIs, number of genes per cell, these numbers are on par with what others in the literature have reported using the 10x platform. And again, this graph is just showing the major cell types across the 4 genotypes, and the pie charts indicating, again, roughly equivalent distribution of these major cell types among the 4 genotypes. What you might note is that we, for probably technical reasons, had many fewer cells from the D2 conditional knockout mice, but we've now done downsampling among the other 3 genotypes to this number, redone the proportional contribution to these major cell classes, and we find the same thing. So we don't believe that there are any sort of artifacts due to sampling in this data set. Okay, what we're really interested in, though, although we can do a lot on the non-cell autonomous effects of FOXP1, are the spiny, how is FOXP1 affecting expression in the spiny projection neurons? So what we did is we pulled these cells and reclustered them, and what we could find, again, was really nice, that most cells are either DRD1 expressing or DRD2. It's pretty rare especially at this time point, to find SPNs that would express both the D1 and D2. this is a violin plot, basically showing the same thing, adding in additional markers, so TAC1 for DRD1 and PANC for DRD2 expressing clusters. So this is really nice that we were getting kind of the two major cell types you would expect that we had defined from the spiny projection neurons. Now, how does this distribution look when we start looking at the different genotypes? Well, what we found in the control animals was there were about 2/3 of the D1 expressing SPNs and 1/3 of the D2 SPNs. This was the same in the D1 conditional knockout. However, when you reduce or you lose FOXP1 from the D2 SPNs, you see a significant reduction of the cell type, and it's somewhat mitigated in the double conditional knockout mice. Now, again, getting back to that idea of sampling and perhaps Cree efficiency, Ashley wanted to show that she could get the same result in an independent manner. So what she did is she crossed the mice to a DRD2 GFP reporter, and so this is the expression of D2R just in the the control mouse, and this is what it looks like in the D2 conditional knockout mouse. And hopefully just by eye, you can appreciate there's a reduction in the GFP signal, and when we quantitate it, we indeed see a significant reduction in the number of D2 SPNs, but there's still quite a number of remaining D2 SPNs, and so one could think, well, perhaps there was a Cree inefficiency. So to look at that, she then co-stained with FOXP1, indicated here in red, and there was no overlapping signal of GFP in red, suggesting that indeed the Cree was quite efficient. The remaining D2 SPNs do not express FOXP1, and this raised the question, what is this remaining ISPN population? And so she went back to thinking about this, the neurochemical distinctions in the striatum, and she used a marker of striasomes, the mu opioid receptor, in purple, and this is just showing these zones in a control mouse where we're indicating the D2R, the D2 SPNs. And this is what it looks like in a D2 conditional knockout mouse. And if you zoom in, hopefully you can appreciate that these cells really are found kind of in a peristromosomal pattern. So they're found around the striasomes. In addition, whereas DARP32 or PPP1R1B is traditionally used as a marker of SPNs, these remaining D2 SPNs that don't express FOXP1 also don't express DARP32. So they're a bit mysterious to us, but we thought, well, we can use the single cell sequencing we've done to try and tease apart what these cells might be. So here we've just color-coded all of the clusters that indicate the D2 SPNs, and we focused on this cluster 7 that contained primarily cells from the D2 or the double conditional knockout mice, but were marked as being D2 SPNs. And we asked, the genes that are expressed in this cluster versus the other D2 SPNs, what are they? And so this is just a plot indicating the genes that are upregulated and downregulated in cluster 7 versus the other. And the genes that are upregulated were surprisingly genes that are enriched for cell adhesion, cell migration, neuron projection, so that we have one hypothesis that perhaps these remaining cells actually have migrated in an abnormal fashion from somewhere else. And so that's one hypothesis that we would like to test. In addition, we found that the downregulated genes tended to be genes that are involved in ion channel activity. And this was really interesting based on our previous electrophysiological findings. And so we went back and collaborated with Jay Gibson again and asked him to basically do the same experiment, but this time in the conditional knockout mice that have been crossed to the reporter animals. And what he found was, again, in these remaining D2 or ISPNs, that we had increased intrinsic excitability in these cells. In addition, he found that there was an increase in resting membrane potential and input resistance. So together, this suggested to him and to us that perhaps there's alteration in a potassium leak channel in these ISPNs with loss of FOXP1. And we have at least a number of candidates, including this potassium leak channel, KCNK2, which we had found was also altered in the FOXP1 heterozygous patient-relevant mouse model. In addition, folks who study the basal ganglia circuitry are always very interested in how, if you're altering one aspect of it, how the connectivity might change. And since we're seeing a reduction in the D2 SPNs, how might that affect the kind of circuit formation? And so to do this, again, we're crossing our animals to various reporters. This is a control mouse crossed to a D2R GFP reporter, and you can see nice projections onto the GPE. However, in the FOXP1 D2 conditional knockout mice, not surprisingly, since we see a reduction in the number of D2 SPNs, we see a significant reduction in the projections onto the GPE. And I can say significant because we've now taken all of these brains and run them through a whole brain imaging facility at UT Southwestern, where they get registered to the Allen Brain Atlas, and we can do quantitative analysis of these tracks. I just don't have the graphs with me. In contrast, we can also see what happens in the other cell types. So this is, again, a control animal with a D1R TD tomato marker. You can see that there are not projections onto the GPE. However, now in the D2 conditional knockout mice, we see abnormal collaterals of D1R onto the GPE. And if anything, this is enhanced in the double conditional knockout mice. So we're seeing non-cell autonomous effects, loss of FOXP1 in the D2 on the D1 projections, which is pretty cool. And we can now use the single cell sequencing and look at how changes in the other cell population might be affecting this projection pattern. So those are kind of our main cellular findings from the D2 conditional knockout mice, but what about the D1 conditional knockout mice? And this is pretty new data, but I thought I would share it with you because I think it's pretty striking. So Ashley quantified and looked at, again, this mu opioid receptor, so marking the striosome. So what I had shown you before was just in the control animal or in the D2 conditional knockout mice where we had that perichromosomal expression. But when we look in the D1 conditional knockout mouse, we see almost a complete absence of striosomes. And indeed, we can also use the single-cell sequencing to identify known markers of striosome and matrix genes, find clusters, and now what we're doing is trying to integrate this information and hopefully explain this loss of the striosome in the D1 conditional knockout mice. But I would like to end with some behavioral results. I know we talked a bit about the caveats of this yesterday, but I think it was important for us to do, given the fact that we really were hoping to dissect out, go all the way from genes to behavior in a cell type specific manner. So these are kind of all the known clinical related phenotypes of these FOXP1 patients, and just focusing on what we think are the D2 regulated behaviors. What we find in the accelerating rotorod, which is kind of a test of motor learning in these animals, as well as in the open field, which is a measurement of activity, that these are significantly impaired in the D2 as well as in the double conditional knockout mice. And this is on par with other, many papers in the literature that if you kind of impair D2 signaling, that you'll see these effects on motor function. In contrast, we see changes in quite different behaviors in the D1 conditional knockout mice. So we again went back to the ultrasonic vocalizations, and we see a change in the number of calls in the D1 conditional knockout mice, and a change in the kilohertz frequency of the calls in the double conditional knockout mice. And I think this fits really nicely with some of our work that we've published on the EMX1 Cree conditional knockout mice, suggesting that cortical striatal circuitry is really important for this early motor behavior in mouse. Also, we found a significant reduction in nest building in these animals, which might be a measurement of another socially related behavior in mouse. So this would suggest that FOXP1 regulates distinct distinct pathways in terms of relating it to behavior D1 versus D1. And I'm going to, D2 versus D1, excuse me, summarize all of these data here for you. So what I've shown you is that single-cell RNA sequencing of the mouse brain can uncover cell type-specific mechanisms regulated by FOXP1, which is this high-confidence autism candidate gene. That loss of FOXP1 reduces the number and alters functional properties, so reduces the number and alters functional properties specifically within the ISPNs, or these D2R expressing SPNs. In contrast, loss of FOXP1 in the other cells, in the D1R expressing SPNs or DSPNs, this affects striosome matrix development. And when we look at behavior, loss of FOXP1 in the D2 SPNs disrupts motor relevant behaviors, and in contrast, in the D1 expressing SPNs disrupts social behaviors. Okay, so I would like to give a big thanks to Ashley Anderson, the graduate student in the lab who's carried out this work, and she's had a lot of help from a postdoc in the lab, Ashwina Kumar, who helped with the bioinformatics aspect of the single cell. All of my lab, another shout out to Stefano for giving a great 5-minute talk earlier today. my funding, and as well as Jay again for the electrophysiology, and Haley Tucker, who provided the Flox allele for all of these studies. Thank you for staying.
Back here, I'm wondering, I've asked this to a lot of people lately, but I don't get a clear answer, and that is, with these D1 and D2 neurons, there are several D1 genes, and there are several D2 genes as well. And I'm wondering if, because, there's...
You mean isoforms?
Yeah, different genes, D1A, D1B, or I forget the...
DRD1A, DRD1.
Right. And so... I'm wondering if, in the case of where you find no difference in the FOXP2, or you find the D2 gene, cells, in the FOXP2 heterozygous, if this could possibly be another D1 paralog, as opposed D2, that is, as opposed to the one you're looking for. Should I ask that again?
In the FOXP1 mice or in the FOXP2 mice?
Sorry, the FOXP1 mice.
I know, it's a lot of ones
just,
and twos.
Just like FOXP1 and FOXP2, they're two
Right.
different, and there's actually FOXP3
right.
and 4.
Yeah, the Crees that we're using, are they really separate genes? I have to go back.
No,
and
because
look.
you did RNA-seq in this case.
Right. We could look at the expression, certainly, of all, the different genes with loss of FOXP1 in each population. And I think we've done a pretty good job showing that when we use the DRD2 Cree, for example, I'm not aware of a highly similar gene for DRD2, that we have complete loss of FOXP1 in all those cells, but yet there are some cells that remain that are not typical SPNs.
Right,
but I'm not
it's the
sure that's
same
answering
Cree.
your question.
Right.
Exactly.
That is the same Cree. I didn't think about that.
Yeah. John.
Striasomes are born before matrosomes, and since you have evidence for a loss of striasomes in the D1 Cree, have you looked to see whether or not the early born medium spiny neurons are missing or died in the case of that particular conditional mutant?
That's a really good question. What we're essentially doing right now is going back through development and trying to answer all of those key questions. This difference in timing, the difference in the timing of the Crees turning on. They were available. They were thought to turn on in ET18, but they turn on way earlier. We don't know if that's a timing question, but I would say that it's maintained at least into adulthood, so that's good. It's not like something's going to happen later on. But yeah, and then we have these weird things with interneurons too that I've told you about.
Beautiful talk. Just a question, follow-up on Eric's. Do you know, does FOXP1 or FOXP2 regulate the D1, D2 family gene expression?
That's an excellent question. We have unpublished evidence that FOXP2 in the cortex regulates DRD1, and there's evidence from the songbird from Rich Mooney's lab that FOXP2 regulates DRD1 as well.
Right. So I'm just wondering, maybe you can look at your single-cell sequencing data, whether the Cree, because these are transgene drivers, so it's very hard. They may not follow the endogenous regulation pattern. But basically, when you see changes, some of them could be due to the driver was.
I believe the Crees are knock-ins into the endogenous locus in this case.
I guess either way, it's maybe worth.
It is a caveat, and I think we do have to look at that more carefully now that we know, or we had known previously of this, regulation. Yeah, but those were the best. The other only thing we could do would be RGS9, but that's going to knock out both populations. If you know of other, I guess there's a Dora 2A Cree as well. Yeah.
Yes, so what's the relationship between the D1 type with the vocalization behavior?
In our model system.
Yeah, just more in general, how does it work? Because they said the pub vocalization, as you said, is relatively innate. There's a basic circuit is relatively simple in the brainstem or PAG. So how does the striate?
Well, I think it's part of the pathway that's important for this output in these cries, and I think even though it's been shown that you could take out the whole cortex and not affect ultrasonic vocalizations in an adult animal, I really think cortical striatal circuitry is important. And we also have published evidence for FOXP2 in the cerebellum being important for these pup USVs. So I think there's a lot of connectivity among these brain regions at different times, different brain regions can compensate depending on what you're, genetically altering. So.
Very interesting. Thank you.
Should I put in?
Yale, who will be presenting why to be human development and evolution of neurological circuits.
Okay, I'll start speaking. So, thank you and other organizers for inviting me to present today. Also, would like to thank all speakers for 2 days of very stimulating talks. I really enjoyed it. It's really one of my favorite symposia. It almost reminds me on Gordon conference. Like really, I felt every talk I was like tuned in. Okay, and thank you all for that. And I think we are near the end, and I think it's obvious by this time that the cortex is at the root of the cognitive and behavior repertoire that makes us uniquely a species, and that underlies at least some aspect of our susceptibility to neuropsychiatric disorders. And I will add to these 2 topics in my presentation, and it will be divided into 3 parts. First, I will talk about our ongoing comparative transcriptomics. I'll use mainly transcriptomics of primate brain development, human, chimpanzee, and macaque. It will be a mixture of published and unpublished, and then I'll tell you about the insights into evolutionary organization and development, human cortical circuits or cell types. I shouldn't say probably circuits because we are not at that level. And again, mixture of published and unpublished, and then again another mixture of published and unpublished. Insight from these studies into neuropsychiatric disorders. The work I will present is the work of 2 very talented postdocs, Andre Sosa and Ying Zhu. Andre is a wet lab scientist. She's a computational biologist, and Ying is just about to start her own lab at Fudan University. And most of the work will be their unpublished work. And so, as some of you have already mentioned, over the last, exactly will be now decade, our lab, in collaboration with many labs, have been really using genomics and primarily transcriptomics to really get an insight in human brain development. So we did this relatively ambitiously, from really starting from embryonic 5 weeks of post-conception all the way to adulthood. We have used multiple type of platforms, and some of these resources have already been mentioned, human brain transcriptome, BrainSpan, and there will be a lot of data hopefully coming out soon from the PsycEncode. And in addition, and so think about this as BrainSpan, and then also we're ambitious enough and fortunate enough to really do this in chimpanzee, our closest relative. At the moment, we only have adult samples, and also we finished doing this in macaque. And the way we did that is we focused on 16 brain regions. I am obviously a very corticocentric person, and the reason is so we have multiple areas of the prefrontal cortex, primary motor cortex, primary somatosensory, inferior parietal cortex, primary auditory, superior temporal cortex, inferior temporal cortex, and primary visual cortex. So we cover major lobes, hippocampus, amygdala, striatum, dorsal nucleus, and cerebellum. Most of this has been done at tissue level, but we have also been doing this at a single cell level. And the idea is to have a really comprehensive analysis as we could of this, and we really follow these 60 regions in the 3 species across the development. And so here is some of the recent data that basically Ying just plotted for you. And so the first problem is, how do you translate across time? I think Barbara has really eloquently tried to explain that. And so what Ying is doing here, so this is a RNA-seq data, so the red dots is a human brain. We are just using one region for the simplicity of the plot. No, she's combining all 11 neocortical areas. My apologies. This is a real time in post-conception days for macaque, human, and these are our chimpanzees. And so what I would like to point this, and this is basically prediction based on a algorithm that she developed called transcriptome age. And basically, as if you follow carefully, that there is some divergence in early fetal development, but early mid-fetal, late fetal, this corresponds to those periods that that couple of you have mentioned in brain span, and then infancy and early childhood. But what was really surprising to us, I mean, it's, you know, there is already theory about this, is that basically the samples that according to age, and this really fits very well with Barbara's prediction, pretty much your dates were all correct if we use our data, just to let you know, all 16 regions, which is really remarkable. But I know that you don't have data after that because it's hard to find the data. And it was really interesting that our multiple samples, which should be within the childhood, do not fall within the childhood. Pretty much macaque is lacking within all 16 regions signature of late human childhood. Okay, so we have to be careful about interpretation of that, just because why is that reason? Is that really species or our environment, food? We don't know that yet. But basically using this strategy, we cannot see that. And just to let you know, none of these samples have been fallen. And by the way, we have around 60-something human brain. We already have now over 300. And whatever you permutate, this is really something is odd about those macaque. They are all from our colony. They all have pretty much same mother, and not same mothers, but pretty much same fathers. So that the genetic variations and food should not be, but something about our late childhood does seem to be different than macaque. So that's the first point. The second thing is we also can now look at the divergence across the two species, across the entire basically development of the brain. Not to bother you with the data. This is so basically, I'll just show you the main finding is that the number of the genes that diverge between the two species, this is divergence between macaque and human across the entire development. We cannot do this for macaque, chimpanzee. I'll show you in the next slide adult data. And basically what Ying and Andre are doing here, they are plotting the level of divergence. Basically red means more genes are divergent. Each line is a one region. So these are 16 brain regions and including 11 neocortical areas. And you can see that there are some differences. So for example, we see early on that prefrontal cortex seems to have a little bit more divergence than the rest of the areas. But once you go, that divergence changes across the development, which it's not constant. We don't have a constantly same the number. Also, the genes that are divergent at this period are different largely than this. And they are largely associated with constructing the nervous system, building up the cell types, while here they are largely related to functional activity, neuronal activity, and many other things, and glial cells. And then the third point I would like to convey here, we see a dramatic downregulation of the genes that are differentially expressed before birth. This is not associated with birth. We have a lot of evidence, and basically starts around 20-something weeks in post-conception in humans. So basically, the number of differences, even within the human brain and the species collapses at that time point, stays low in infancy, and then goes up in late childhood and adolescence. And that is probably why monkeys might not have this signature of late childhood in humans. And so the other thing, at the moment, we can do this in adult. And so this is a 16 regions done with adult macaque human. And so I'm going to plot just human-specific upregulation. To do that, we are looking at only genes that are conserved in expression, both spatially and a little bit temporally, even though it's adult, between, this is pan, which means a chimpanzee and macaque. And you can see that the number one region actually that has the largest number is striatum, comes medial dorsal nucleus of the thalamus, primary visual cortex. And you can see that actually prefrontal cortex and some cortical regions that are really probably all our favorites are not exceptional. And there was a similar number of downregulated genes, slightly less. That's exactly what I think Dan has observed and Jenna many years ago with arrays. Basically, the slightly less genes are downregulated, even with the new technology and new annotations. But we see similar profile of again, striatum. And so there is something interesting about striatum. And it was actually perfect after Jenna's talk. So around 12% of genes using this data set, that's 16 regions. And of course, they are not randomly distributed, these genes. They do follow with specific functional categories that I'm not sure how relevant they are. And so as many others have done before us, we have also used a gene co-expression analysis developed by Steve Horvath and Dan Geschwin in I think it was 2005, and basically you can now cluster genes that have similar spatio-temporal distribution. In this case, it's mainly regional. So we are again comparing human, chimpanzee, and macaque. This is adult data set. This was published last year. And red is human, and green and blue are macaque and chimpanzee. So here is a set of genes that are highly regulated across the telencephalon in humans. That's the neocortex, hippocampus, amygdala, and striatum. And if you look at the network of these genes, many of them are uncoding, but two of them caught our attention, which was TH and DDC. These genes were really interesting because, as I'll show you later, they are really important enzymes to make a dopamine. And then we intersected this data with the single cell data from human, from multiple sources, including ours. And just to show you that, so basically, to make story short, this TH is expressed in DDC in somatostatin, interneurons, small subset of somatostatin, and basically what we are plotting at the direction of changes in the chimpanzee lineage. And so you can see that these genes are downregulated or not expressed in chimpanzees, and some of them are expressed selectively in chimpanzees. Also, we plotted, just to show you interesting, some neuropsychiatric related gene, neurotransmitter related gene, and ion channels, and you can see that multiple categories are changing at cell type specific level between human and chimpanzee. And then we were fortunate to set up a collaboration with Chad Sherwood and Marian Raganti, and we did not discover these cells. Many before us have characterized, especially Chad, carefully, but basically we wanted to validate that what we see at the gene expression fits what has been observed. And so pretty much these are cells. They are very rare. They are in deep layers, 5, 6, a little bit white matter. This is in chimpanzee and other non-human African apes. We have more data just for simplicity. You can see the axons positive probably from VTA. We can see some cells in macaque. They do look differently, but they are there, and so it really seems lost in non-human African great apes. What are the tyrosine hydroxylase? I mean, even Arturo mentioned this. So this and DDC are important enzymes to make a dopamine. And so we look whether they are colocalized. And so what we observed actually that around 60-something% of these cells, which are rare, I'll get to the numbers, co-express DDC. First, not all of them co-express DDC. So we already observed that there might be some heterogeneity. I'll get to that a little bit later on. So, and then we wanted to a little bit more characterize again, and to make story short, they do not express any of the proteins that John, Arturo, and others have analyzed to be classical or canonical markers of interneurons. We know that they are somatostatin mRNA, but they do not express, not a single one of them expressed somatostatin protein when you use immunohistochemistry. I'll get to that. And then we wanted to see their distribution across species. So again, with Chet and Marianne, we are able to really systematically analyze this in multiple regions of the human, chimpanzee, and macaque forebrain. And first, we look at olfactory bulb, whereas remember, Arturo, these cells are present in other mammals, and they are really well characterized in olfactory bulb. And we can see them in olfactory bulb in all species we have analyzed. Then we look at the striatum, both caudate and the putamen, and what we observed is actually there are 1.5% of neurons in the striatum that have characteristics of tyrosine hydroxylase. But what was really interesting is that's almost twofold increase comparing to chimpanzee. Also, we have data for bonobo, but just not enough to do the analysis, and gorilla and others. So what it is saying us that we humans have way more these cells in the striatum than other non-human primates analyzed. The same trend is in. And then when we look at the multiple areas of the neocortex, carefully, serologically, we couldn't find any of these cells, which fits beautifully the gene expression data and the analysis. And so where do these cells come from? That was also another interesting twist in the story. And so basically we look at the expression of TH, and then we observed, as you would expect, there is a lot of TH in the striatum. So this is basically brain span data, and so what Ying is plotting here where the TH is expressed, and you can see that it goes up, and then you can see it a little bit in the neocortex, but you see it a little bit more in the hippocampus and amygdala, and the expression and the levels go up as you age, which is a very interesting. And here is why it's interesting. When we look at the immunohistochemically, when do these cells appear in the human cortex, we see a labeling of axons. These are probably nigrostriatal axons express dopamine, but we don't see any cells in the human neocortex or even in the striatum at mid-fetal. That's here. The striatum has already a lot of mRNA, as well as other regions a little bit less, but we don't see any protein using a well-characterized antibodies. And then where we see the first neurons is actually in a newborn human, and basically we see a lot of labeling in the striatum. Don't get fooled, these are not neurons, these are actually axons from striatal axons. And then also on the side, we saw this stream of axons, which are probably VTA axons going to external capsule. And when we look carefully there, and slightly in the white matter, we saw this bipolar migrating like. We don't know whether they migrate because we didn't do any assays. But they look, and we can see clearly that at this point, they already have a lot of mRNA in the striatum and even in the cortex and amygdala and the hippocampus. But that's where we start to see first immunopositive cells, means that those that express protein. So we look at the chimpanzee newborn, and we see the same pattern when it comes to staining of the fibers. This is a pristine sample. Actually, it's like a PMI of less than 1 hour. And you can see that there is a lot of labeling in the striatum of axons as well as the external capsule. These are probably axons from VTA going to the neocortex, but we couldn't find a single immunopositive neurons, suggesting that these cells probably have never existed in chimpanzee cortex when it comes to developmental origin. And I don't know how this is related to what Arturo has shown about whether these cells are really migrating. I don't think so. I'll get to that point a little bit later, because first thing you have to keep in mind, there is already a lot of mRNA in these structures, but not protein. And so one other question we wanted to know, where do these cells come from? Work from John and Arturo and Arnold has shown that a little bit us basic that most of these interneurons in the human cortex come, but these are rare from the ventral part, and so we wanted to do it. So what we did is we culture a human MG, CG, and LG, which are the origins of interneurons, and also neocortical, because my advisor, Paško Rakić mentor, had also evidence that some interneurons come from the ventral, so we wanted to see whether these cells come from ventral or dorsal. To make story short, so when we make culture from LG and MG and CG, we see the TH cells there. And also we use the canonical markers that John has really linked to specific lineage, such SP8, NKX2.1, and NR2F2 for these 3 lineages. And you can see that they express appropriate marker depending on their type, place of origin. What was really important that these cells in culture mimic basically what we see in vivo. A large majority of them are GABAergic and express DDC. And then we added BRDU, asking, do these cells come from ventral or dorsal part? And so when we add BRDU, we can see, sorry, let me just show you a graph. We can see that they basically proliferate in MG, LG, CG, that they are derived from progenitors in MG, LG, and CG, but none of them from progenitors culture from the neocortex are positive, indicating that these cells that we can do come from ventral part. But here is an important aspect. These cultures are maintained for several weeks, and we can see a positive TH cells even in the neocortical SVZ, VZ cultures, but they are not generated there. Actually, they are early generated. But remember, we don't see them up to until newborn. This is a 20 weeks, so that's another 20 weeks of maturing. So what this suggests to us, and it's really that these cells come to neocortex early, express somatostatin and THRNA, but they do not express protein. So it is probably post-translational regulation of TH, and I'll tell you why this is important. Okay, so the next thing, we wanted to know whether we can get these cells in IPCs for 2 reasons. One, we wanted to test whether they can make a dopamine and release it. And also we wanted to a little bit more characterize them. And to make story short, this is nothing novel. Many of you have done this. And so if you culture these, if you can directly differentiate IPCs in cortical progenitors, and then you can create, you get excitatory, but after 80 days in vivo, you get also, in vitro, sorry, you get interneurons. We do get TH cells, and they have all of the characteristics we could find in vivo. It means that they express somatostatin, but they switch. Either it's mRNA, but not protein. So that's really, so we, okay. And so we wanted to see, also they express DDC, they are also GABA, and then we wanted to see whether they can transport dopamine, and to do that, basically what you do is you add a dopamine, a monoamine uptake assay, which are the They are not specific for dopamine transporter, but basically it's a fluorophore that is caged when it's outside, or when it can be transported, becomes fluorescent. So the cell that can transport it becomes the fluorescent. And here is a live imaging of these two cells, and you can see that only those two cells, but not axons that are not labeled with TH, actually the one that can transport the dopamine. And basically, this is the quantification, basically, and the cells that express both DDC and TH are the ones that are more positively labeled. And then we also tested using HPLC whether these cells can make a dopamine. We don't see any other TH positive or DDC positive cells in the culture. When we use control cultures or media, we don't see anything, and this is a primary neurons from a mid-fetal LGE or IPC. We can clearly get substantial levels of dopamine, considering how this is actually really rare, and indicating that these cells, at least in the context of in vitro, can make a dopamine. And so here is the model so basically a small proportion of 0.5% of somatostatin interneurons express TH mRNA, but what is happening is that these cells can either express somatostatin or TH protein. We really have never seen combination of this. And we have looked at other mammals. Actually, this is not the case. So this could be uniquely human. And so we don't know whether they make dopamine in vivo. That's really, and while we were writing the paper, we realized actually Nick Spitzer discovered similar phenomenon in a rat hippocampal, where they also switch between somatostatin and dopamine production. So we think it's something similar, but this is not the case in the mouse and other mammals we looked. Okay, so now I will just tell you some insights into neuropsychiatric disorders we get from this data set, and it will be one part is unpublished, and there will be an old published story that I really decided to present. Just I felt that it's really intriguing, and I hope you will appreciate it. Again, the work is, again, unpublished work from Andre and Ying, and there will be some old published work from Ken Kwan, who has his own lab at the University of Michigan. And so just we'll show you one slide. So now we have this macaque data, I'll call it macaque brain span and human brain span, and now we can really compare between the three species and ask simple questions. How much divergence do we see? But then we can also ask how much the genes that are linked to disorders that most of you have talked about it are divergent across the two species. This is just across the two species, so we don't know whether how many of these are because it's human specific. And so we look at high confident autism spectrum disorder, which are now around 75 of them. Then neurodevelopmental disorders that are excluded from high confidence autism genes, because as some of you have already pointed, there is an overlap. ADHD, schizophrenia, bipolar depression, Alzheimer, and Parkinson's disease. And now we are looking at two types of genes, those that diverge based what we call heterochronic, which is a timing, means that they are expressed in both species, but their developmental trajectory has a different shape, okay? And you can see that basically, like, for example, there are only like 70 of these genes, but already 10 of them are actually have a significant divergence in developmental trajectory. And we see a lot of schizophrenia, but the schizophrenia is our correlation with the GWAS study, so you have more genes. But then we also look at heterotopic, so these are genes that have their developmental trajectories are similar or not different, but they have a different expression, means they are expressed in one place in one species, but not in the other. So they didn't, in the first case, they have to have same spatial expression, but different temporal trajectory. In this case, the temporal trajectory has to be the same, but you are expressed in one species, in one area or brain region, but not in the other. And you can see again that autism and schizophrenia genes do have changes, not just in timing, but also in some cases, these are non-overlapping, just to let you, genes. And it was also interestingly, schizophrenia, we were able to find actually really 5 distinct clusters. So for example, we see cluster of genes that are really different, diverge between human and macaque in prenatal, prefrontal cortex. We also see that diverge in frontal cortex only during early postnatal period, and only those that are in adult. So most of these are also transiently expressed. And now I'll finish something that was really published, because I don't know if you know this story, but it really tells you how complex is what we are trying to solve and how little do we know about human brain. And so many years ago, Ken Kwan, who was a postdoc in the lab, really discovered something that I think it's really mind-boggling. And so what he discovered is a set of mini columns in a frontal operculum during mid-fetal development from 13 to 23 weeks of post-conception. what I'm going to describe you is a molecularly transient phenotype. And so what he was, when looking at nitric oxide synthase staining, and so this is the labeling for nitric oxide synthase. In the middle of frontal operculum is the future region, which consists of orofacial motor cortex and future Broca speech area. And what he observed is that nitric oxide synthase protein is expressed in this future layer 5 cells. He also see layer 3 cells expressed in anterior cingulate and a little bit dorsal frontal parietal cortex. But what he observed is this. If you look careful just to get to appreciate this pattern, he observed that cells that have BC11B, which are subcerebrally projecting corticospinal tract or corticobulbar, in this case, but not corticocortical, which express marker SATB2, have high levels of NOS1. So basically you have alternating mini columns. These mini columns are 15 to 20 neurons, and he found he actually completely reconstructed 2 brains. He found 45,000 of this. So it's not a huge number. It's actually so small, so whoever looked before us missed it because it's so small. It's actually basically 2 millimeters by 3 millimeters. So that tells you. But it's in the really region of the human brain that is really, anyway. And it has these beautiful alternating patterns, and I'll tell you a little bit more. And so if you now look at the nissel of this region, you see this what's called ontogenetic column that Pashko and Stefan Polyakov in Russia described. Basically, cortex is form of these radial columns, okay? But look at this. This is adjacent section. You can see that these columns are not just based on NOS1 protein, and this is enzymatic activity for NOS1 NADPH diaphorease. You can see it also with FESF2, which is one of our favorite genes, which also has BC11B, is important for corticospinal tract. And you can see alternating column. One expresses set of one genes and another. But this is really restricted to this region, which is really interesting. But here is what turned the project upside down when I said, we need to do in situ hybridization. It's a human brain, small region. You know, you want to be sure. He did in situ hybridization. He can see the columns, but he sees the expression of mRNA in other cells as well. Okay? So then we look at, okay, let's look at the macaque. And so when we look at the macaque, we do the time series in the macaque, and you can see something that is reminiscent of these columns. Okay, but they are not as beautiful as these and clearly delineate, but you do see them, okay? And this is an enzymatic activity. But when you look at the mouse, basically mouse NOS1 is a marker of interneurons. That's well known and established, nothing novel. And it's clearly, and you can see same interneurons in the human brain, but in addition to interneurons, at least in this region, they express these mini columns. But what was really striking when he did mouse in situ, he also observed that mRNA is transiently expressed as well during mouse perinatal development. And this we confirmed this by looking at Allen Brain Institute, their developmental mouse. So both in humans, macaque, and mouse, we see transient expression of mRNA for NOS1 around what would be equivalent of mid-fetal development in human brain. But for the reason that I'll get to that, only in human brain and slightly in macaque brain, we see these mini columns only in that region. So what Ken did, we suspected, or he suspected, that this is an mRNA binding stuff. So he did a pull down using a NOS1 and control mRNA. To make story short, he pulled down a complex of proteins, did mass spec Western blot, and ended up being a FMRP, which is a gene that is a first gene linked to intellectual disability some 25 years ago. It's a RNA binding protein that is deleted or altered expression in Fragile X syndrome, which is one of the most common forms of intellectual disability and often is associated with autism-like features. And so then can look at, okay, can I find something in a human? And so we know that we see these columns in human and macaque. Okay, and we don't know chimpanzee because we don't have tissue. But when we look at all the New World monkeys and prosimians and others, we don't see. And so he was able to find these sequences only in humans, apes, and all New World monkeys of these what's called G quartets. So G quartets are sequences that FMRP binds. Also there were, and so basically he predicted 3 of them, did the pull down and showed that actually 2 of them FMRP binds to it, and then he did the EMSA assay showing that this binding is specific because he can inhibit it with FMRP antibodies. Now is this all functional? And so what he did now, he went back to mouse and asked, okay, what if I forcefully expressed FMRP in a mouse brain? Remember, mouse does have transiently expression, so he electroporates control GFP when the layer 5 is born, so he can express GFP in layer 5, but of course there is no NOS1 in protein in these cells. the first neurons start to come exactly at the birth. This is birth, so you will not see any interneurons. And then when he forcefully expressed mouse NOS1 using a CAG promoter, which is one of the most powerful promoter, the mouse has mRNA but has no machinery to translate this into protein. But when he puts stoichiometrically, means same basic hemolality of the human NOS1, he gets the expression robust in pyramidal cells. Then he does experiment when he does, is this dependent on FMRP? Because mouse has FMRP, so if you do this in FMRP knockout, or which is FMR1, you lose this expression. And then he takes a mouse, he takes a human sequence and replaces those G quartet with the mouse, and he loses this ability to express. So what he was able to show that it's actually really human NOS1 has that sequence, and it does not depend on FMRP. Is this all relevant? We are also fortunate to be collecting brains, and you really don't know sometimes you will need something, so we collected FMRP prenatal and postnatal brain, and basically there is a loss of NOS1 expression in the mid-fetal frontal cortex. When we look at the mouse, there is no loss, really demonstrating that this is not just relevant biochemically, but it's relevant probably to a certain extent in Fragile X syndrome. So I will now summarize 4 points that are absolutely just repetition of what many speakers have said. So human brain transport is not continuously divergent, means that this divergence is temporarily regulated, and it forms this cup shape, phylogenetic shape. Macaque lack transcriptomic signature of human late adulthood. We don't know to what extent this is due to our lifestyle and food we eat. Our children are all exposed to activity, they are not. It could be that that is one problem. And as many others have said, human circuits have undergone both molecular and cellular reorganization. Even the same region, same cell type has undergone changes in human lineage, means last 6 million years. And we think that they could have a distinct forebrain dopaminergic interneurons, maybe even distinct mini-columnar syndrome, because we do see some quantitative differences between human and macaque when it comes to these mini-columns. I didn't have time to tell you. And also that some disease genes risk have primate or human-specific expression, and this is a repetition of what other speakers have said. And I already acknowledge my colleagues, also Brainspan and PsychEncode, NIMH, for really being generous, and my collaborators. And thank you for your attention.
Do you think the TH-positive neurons you're seeing could also be specialized in parts of the cortex, like some speech areas, or are you seeing it all over the cortex in humans?
We don't have that resolution at the moment. But
you can tell if it's ubiquitous or not in the sections that you've looked at.
Oh, we didn't look at that carefully, just to be honest. Yeah. But it is conceivable, yes. And one thing that I forget to tell you about, when we look at the mid-fetal cortical cultures, we see only all of them are LG, they express SP8. So just to let you know. So it is conceivable that they, but we see some diversity, so it is possible that later on they come from MG or somewhere else.
I'm just puzzled about the Broca's area of mini-column.
Yes.
They're only found at one particular stage of development?
Yes.
You don't see anything in adults?
No.
So what do you think is going on?
So we don't know, but we are continuing working on this, and so even though it's been already 6 years since, by the way. So the one problem is that we see mRNA everywhere, FMRP is everywhere, but only that region turns on the protein. So we think it's activity dependent, okay, just to let you know. And we have some evidence, because the same columns also, if you use CFOS, they also light up with CFOS. So one simple interpretation, and also in macaque, we do see the columns that are similar, but one thing is different. In macaque, they appear slightly later if we use a translation cross time from Barbara and our transcriptomic. And actually, I didn't tell you the whole story, actually. They disappear after 23 weeks, but they turn on in humans around the birth for a few weeks, when you get what's called the Grunt-type cortex of Brodmann, okay? So there are 2 phases in humans. The first one only future, let's not call it Broca, frontal operculum, okay? And then all over the cortex. In monkey rhesus, we only see all over the cortex. We don't see this early phase. And we have a lot of monkey tissue, just to let you know. So what it is possible that this is uniquely human, but I cannot say that because we haven't looked at any other non-human primates. And also it is possible that cortico-bulbar, that is cortico-bulbar pathway, is one of the first pathways descending to get to the brainstem, and it is possible that they form circuits earlier, and so that it's some retrograde, or because activity is important, because only those columns that have NOS1 also turn on the CFOS. But that's speculation.
Thanks for a fabulous talk, Leonard. I was really intrigued by that cup-shaped pattern
Yes.
of transcriptional divergence, and
Yes.
particularly the late fetal one.
Yes.
And I was trying my mind to make sense of how one might see a surprising loss of transcriptional divergence during a period of prenatal brain development that presumably is when many microscopically visible divergences are apparent. So that was one question I had, what's actually happening in terms of the developmental process of corticogenesis during that period. And then the other question I had is the heterochronic gene set.
Yes.
Do they have, what's their regional profile of expression? In which regions are they most highly expressed?
Okay, so first answer to first question, this is a speculation. So I've been really very interested in late fetal development because of this. So, we had this data. And so I think it's a unique part of the development that we know very little. But let me tell you what I think we can make. So first of all, that's when you have a huge explosion of astroglia, oligodendrocytes, and synaptogenesis. And secondly, Brodmann described a phenomenon called Grunt-type cortex. And this is why he called it isocortex, not neocortex, because he said everybody has this layer 4. And so what it is, it's probably thalamocortical fibers. I think what drives the expression difference is thalamocortical fibers. They know to go to prefrontal cortex using transcription factors that others have described. But once they come, we see they also peak at the same time neuronal activity, genes, synaptogenesis. So you have a humongous, now the whole system is being synchronized to all neurons pretty much are there. They send their long projections, and now you need to build your synapses, you need to get to the glia. Those programs seem to be less regionally diverse. That's just hypothesis or speculation.
Yeah, beautiful talk. Just one question. I remember there's some old literature about Th-positive cells in the mouse cortex, and they seem to be transient only in the neonatal, then disappear. Do you know?
Yes, that is true. We have, that is all true. So, and we have been analyzing, but they do not have these features. So I can tell you that, yeah. So it is a homologous cell type that has undergone tremendous divergence.
So that was such an interesting talk. One of the things I'm curious about, and this is again just to ask you to speculate, but with the SST expressing and the TH expressing neurons, you said that it ends up sort of being a choice almost, where either they express one or they express the other. What kinds of signals do you think, or have you looked at what kinds of signals might be stimulating them to one direction or the other?
I don't know. We are working on that, and we have absolutely nothing to tell you. So Nick Spitzer has linked it. He has found these cells similar, not these cells. They are different cell types. He's famous because he discovered neurotransmitter switching and I think Story-Landis. But what he has shown that it's regulated in case of Xenopus to light. We don't think it's true for us. Even though there are this, because we have cases that unfortunately die during night and morning, and we don't see that difference. And it's always all or none. There is no in between. There is no gradient. So, and it's just not that easy. So that's why actually we are now doing this and try to get this to the mechanism in IPCs. Because the cells in IPCs do the same thing, which is really good. So we have the functional mechanism or assay, but I have nothing to report to you. Everything we have chased did not work.
Do you know whether the TH cells, cortical neurons, die in humans with Parkinson's?
Yes, they do die. That's not our study. There was a study from Japan that they basically show that in late stages of Parkinson that do correlate with cognitive decline, these cells are gone. Whether that's a correlation or just we don't know that.
So are these cells interneurons or no?
They are interneurons.
They are
They make GABA.
They do make GABA.
Yes.
And but they can't make dopamine at the same time.
So they do make dopamine when they have TH, and some of them have TH and DDC protein. And at least in the culture, both primary human fetal cultures as well the human IPC derived, they do make dopamine.
So they can make dopamine and GABA at the same time?
That I cannot tell you for fact because I haven't tested that.
Okay.
That I cannot tell you.
Okay,
That's
so
beyond my expertise.
So basically, when we detect SST positive interneurons in single cell experiments, if they also express TH, and this occurs in humans, we are not making a huge mistake, basically. It could be there.
Yeah.
Yeah, okay. All right.
Okay, thank you.
Great, so we're going to finish off with the final set of Data Blitz talks. We'll begin with Saivash Darbandi, speaking about neonatal TBR1 dosage controls cortical layer 6 connectivity, and then Damon Pouliadakis, a single-cell transcriptomic analysis of human neocortical development, and finally Andre Teppenkamp, speaking about the 7q11.23 protein DNA JC30 interacts with ATPase synthase and links mitochondria to... I can't believe I managed to get that out.
Okay, so I'll just start. So I'm Siavash, a postdoc in Dr. State and Dr. Rubenstein labs, and we're going to talk a little bit more about TBR1 today. Driving down memory lane back in 1995, when I was a wee lad, Dr. Rubenstein's lab discovered TBR1, and over the next several years, what they showed is that TBR1 is a T-box transcription factor that's mostly expressed or exclusively expressed in postmitotic projection neurons of layer 6 and subplate, and then they went on to show that TBR1 is important for regional and laminar identification of cortex. And it wasn't until more recently, through the work of Dr. State and his collaborators, that they used de novo loss of function mutations to identify autism risk genes. And through the list of genes they identified, you can appreciate that TBR1 is one of the top 20 high-confidence autism genes. But the question becomes, what are the postnatal functions of TBR1 that, lack thereof, may contribute to ASD risk? To answer those questions, I took advantage of this TBR1 conditional mutant that was generated between Dr. State, Dr. Rubenstein, and Bin Chen from UC Santa Cruz, and we showed in the paper that the mutant allele does in fact act as a loss of function allele. And since these autism genes that have these loss of function mutations are mostly enriched in deep cortical layers, I used NTSR1-CRE to delete TBR1 in layer 6. And amongst many different things I addressed in the paper, one question was, does TBR1 contribute to synaptic development and function? So the first step for that was using immunohistochemistry and look at the changes in synaptic density in the same cortical lamina of layer 5 for mutant and control. And what I saw was that in case of the heterozygotes in green, homozygotes in blue, there's a decrease in synaptic density compared to the wild type. And this data corroborated with what we got from the recordings from these cells, where we saw a reduction in the intrinsic properties of spontaneous iPSCs and EPSCs. But the question becomes, how is TBR1 doing this? Is this a non-autonomous effect, or is TBR1 really playing a role in controlling these synapse numbers? So to answer that, so the conclusion was TBR1 is required to maintain these synapse numbers. So to answer what is the mechanism behind this, I went through my list of dysregulated genes that I generated by doing RNA-seq on these sorted layer 6 neurons of wild type and mutant, and then took advantage of literature to see if we have any dysregulated genes that are involved in synaptogenesis or synaptic plasticity development, etc., and used an in vitro assay to screen these genes to see if I can identify any candidates that can rescue the change in synapse numbers. So to do that, I harvested cells at P0, transfected them with my expression vector at P1, and then analyzed the cells 2 weeks later at P14. And what I saw was amongst the 4 different genes I assayed, Wnt7b was the only one that rescued the changes in synapse numbers, both excitatory and inhibitory. So the question was, if we can do this in vitro, can we reproduce that in vivo? So to do that, I generated a flex lentiviral construct that is, the cassette is flanked with 2 different variants of LOXP site, and in the presence of the CRE, it causes a gene inversion, or what's referred to as stochastic gene activation. And this was pertinent to make sure that my construct is only expressed in the layer 6 neurons that have the CRE, and it doesn't disturb any other neighboring cell types in that region. And then pups were born at P0 and genotyped. I injected the virus at P1, and 4 weeks later, looked and processed the brain to see if there was any changes in their synapse numbers. And this is just, I don't know how well you guys can see, but you can see the injection site, the GFT from the construct. I used that as a landmark for my imaging to which dendrites to analyze. And what I saw was in wild type, uninjected with my construct, there was no effect on their synaptic density, both excitatory and inhibitory. Here I am only showing the excitatory ones. But in case of the heterozygotes and homozygote mutants, I was able to rescue the synaptic deficit in vivo using this method. So the bottom line is that TBR1 promotes excitatory and inhibitory synapses in layer 6 neurons, and restoring Wnt7b expression, at least in this case, helped to rescue this synaptic deficit. And these are some other conclusions from the paper that Dr. Rubenstein presented yesterday, and it was recently published in Neuron. And with that, I'd like to thank both my supervisors, Dr. Rubenstein and Dr. Slade, for their unwavering support, lab mates, collaborators, and funding agencies. And I just want to leave you guys with one thought where this work is going right now is I'm trying to take advantage of the assay we have in Dr. Rubenstein lab using MGE progenitor transplantation to use these progenitors as vehicles to deliver the protein locally and see if we can get a rescue in vivo in a paracurrent fashion. And with that, I'd like to thank you all for your time and being invited to the meeting. They're usually at the, where are they? Well, probably at the spines, or no, hang on. So the question was, where are the receptors for Wnt7b? One speculation would be probably there might be the Frizzle receptors, part of the Wnt signaling, or the protein could be localized into the dendritic spines or being somehow involved in forming those. Yeah.
Next speaker is Damon Poliudakis.
Okay. And F5, yeah.
F5, they'll go to the beginning anyway.
All right, great. Good. All right. First of all, thank you to the organizers and the speakers for an absolutely fantastic conference. It's been both thought-provoking and inspiring. So I'm Damon Poliudakis. I'm a postdoc in the lab of Dan Geschwin. And for this project, I worked very closely with my co-author, Luis de la Torre Ubieta, who's now an assistant professor at UCLA. So as many of the exceptional speakers before me have already highlighted, many of the fundamental questions about the human brain remain relatively unanswered. Single-cell technologies are giving us a new way to address these questions. for my purposes today, we'll focus on using single-cell transcriptomics to catalog cell types in human fetal neocortex. To that end, we collected frontal cortex from 4 donors. It's a snapshot, so they're all gestation week 17 to 18. This is a good snapshot, though, because it should capture both the end of deep layer neurogenesis as well as upper layer neurogenesis. It is pre-gliogenesis. That would be the one caveat. And so from these samples, we ran DropSeq, processed the data and analysis to construct a catalog of 40,000 cells from human fetal cortex. From this catalog, we are able to identify all of the expected major cell types. So I'm highlighting the expression of groups of marker genes from some of the main cell classes here. And so the rest of the talk, I'll highlight a few of the insights we were able to gain from exploring this catalog. The first question we asked was, what transcription factors are enriched in these major cell types in human fetal cortex? And so I'm just highlighting a couple of the cell types up here. So all of the transcription factors that are in black from combing the literature, and some of them are very well known, were previously associated with these cell types, but the few I've highlighted in red were transcriptional regulators that were not previously associated with these cell types. I'm going to focus here on ST18, which is clearly enriched in a couple of glutamatergic clusters. And so we're interested in this because when we zoomed in on it further, we saw that it really split this cluster in half. It seemed to overlap pretty well with known subplate markers, and the other half of the cluster was deep layer. So to explore this further, we went in and subclustered this cluster of cells, and it clearly subdivided into cells that were expressing subplate markers, cells that were expressing deep layer markers, and this transcription factor, ST18, seemed to overlap very nicely with subplate cells. So this was the first single-cell transcriptomic isolation of subplate neurons, and from that we were able to go in and identify multiple novel markers of subplate neurons. So I'm plotting them here in a bulk tissue LCM lamina data set, and you can see that generally there's fairly nice enrichment in the subplate. The second thing we were very interested in doing with this catalog was beginning to map disease risk genes to different cell types in human fetal cortex. To that end, I'm highlighting just one of the diseases we looked at, autism. So we've got autism risk genes here, and you can see that sort of as expected in recapitulating the work of others in bulk tissue, there's a clear enrichment for many of these genes in glutamatergic neurons. There's also some pan-neuronal expression and some that are more enriched in interneurons. We are particularly surprised, though, to see clear enrichment in a couple of other cell types, namely oligodendrocyte precursors as well as parasites. So we think this is the first time to implicate either the maintenance of the perineural environment or the blood-brain barrier in autism risk. And it holds up when we look in adult human brain as well. This is a nice data set from Lake et al. So again, most of it's pan-neuronal, but we see some expression in other subtypes, cell types. So the last thing that I want to touch on is this is well and good looking at ASD risk genes, but as this conference has highlighted, a lot of disease risk localizes in non-coding regions. To that end, we used an open chromatin data set to link regulatory elements in human cortex to promoters, and then ask which of those genes that are linked to these regulatory elements are enriched for disease risk using partitioned heritability. And the one I'll highlight up here is schizophrenia, which had again previously been linked to, of course, to glutamatergic neurons as well as interneurons. But again, we now see some enrichment for various progenitor cell classes as well as oligodendrocyte precursors and fetal microglia. And so with that, again, I want to thank Dan and Luis, as well as our collaborators in the Plath Lab who were responsible for the DropSeq apparatus, and postdoc Justin, who helped us actually run the DropSeq. So thank you all for sticking to the end and your attention.
That last slide you showed, wouldn't that indicate that almost all the cell types you're looking at is enriched for something that's related to schizophrenia, that it's not so specific except that it's the brain?
Somewhat. So we didn't pick up parasites, but definitely everything in sort of the neuronal differentiation trajectory from progenitors on through to maturing neurons. Yeah.
What's the sex of the brains?
It was 3 female and 1 male.
Do you have any plans on sort of expanding to the sex differences in cell representations?
Yeah, I mean that would be very interesting to do. We tried to see if we could do it with what we had, and it's underpowered. But yeah, I would love to look at that. All right. Oh, yeah.
Did you have orexin receptor? You have orexin receptor in substrate. I didn't see that. It's called hippocretin receptor because the.
So I don't remember off the top of my head. I know we had a lot of the classical marker genes were pretty clearly expressed, but I don't remember specifically. We can definitely look afterwards, though.
Thank you.
All right. Thank you.
Final speaker, please introduce Andre Teppenkamp.
So, Andrew, formerly, actually. It's okay. With a name and title this long, I would also just start cutting letters. So I'll just echo everyone's gratitude for the fantastic seminar that we were treated to these last couple of days, not just for me personally to present my work, but just for the opportunity to stand on the shoulders of giants in the field and see where we need to go next. So I'm really interested in genetic brain disorders, and my focus is to try to understand the mechanisms that link the specific genetic defect to the neurobehavior that we see is observed in patients. And so one opportunity I took to do this was to look at the 7q11 locus. So this locus is susceptible to variations in copy number. Deletions in these 27 genes results in Williams syndrome and is marked by hypersociability, weak visual spatial skills, and reduced brain size. By contrast, duplications of these 27 genes is associated with schizophrenia or autism spectrum disorder, where ASD is associated with hyposociability, delayed language, and an increased brain size. So the question is, how does the dosage of one or more of these genes contribute to the spectrum of cognitive features that we see here? The strategy we chose to pursue this question was to look just within these 27 genes, what is their normal expression profile in the developing human brain? And when we looked at just the expression, actually it's easier for me to just talk from here. We look at the expression of these 27 genes to see whether there was any co-expression which might infer a conserved or coordinated molecular process. We found 3 clusters of co-expression. Focusing now on just clusters 1 and 2, we took the top 1,000 co-expressed genes within each cluster and did gene ontology, and we found for cluster 2 genes things related to chromosome organization and DNA metabolism. And in cluster 1 genes, we found things like synaptic transmission and ion transport. What was surprising was the GO terms related to respiratory electron transport chain and the redox process, things that are related to mitochondria. So surprising because we not come across any data that had suggested mitochondria might contribute to these disorders. Doing some more spatial temporal analysis to gain some additional analysis to gain some spatial temporal resolution of these gene ontology terms, I summarize this here in panel D, where basically cluster 2 genes and their ontologies are enriched prenatally throughout the brain, and cluster 1 genes are enriched postnatally and more enriched in the neocortex. And when we looked at the 7q11 genes that were driving these ontologies, DNA JC30 popped out at us basically because it has been functionally uncharacterized up until this point. So there's no data on its expression, its function, or anything. So this is really a wonderful opportunity for us to explore some fundamental biology as well as see how it relates to these disorders. So first we looked, DNA JC30 is expressed in neocortical neurons. It begins mid to late fetal and is expressed continuously throughout development. We wanted to know what are its protein interactions, protein interacting proteins, partners, and so we did a yeast 2 hybrid, and the strongest and most frequent interacting protein is ATP6. This protein is a component of ATP synthase. It is the part that loads hydrogen ions back through the intermembrane space into the matrix that coordinates ATP synthesis. We confirmed the yeast 2 hybrid results by isolating mitochondria from post-mortem human brain. You can see in the input lane that we have representative proteins from the entire oxidative phosphorylation pathway, but when we IP with complex 5, we get back specifically complex 5 and DNA JC30. We also confirmed this using immunogold EM in macaque prefrontal cortex, so each black puncta is a DNA JC30 molecule, clearly in pyramidal neurons. When you zoom in, the black puncta are associated with mitochondria. When you zoom in even further, they're associated with the intermitochondrial membrane. So then the question was, well, what happens if you remove DNA JC30? And so we acquired Williams syndrome cells and postmortem cortex. On the left is just to show you that DNA JC30 expression is in fact decreased in the fibroblasts as well as in the brain. We then use these cells to do seahorse mitochondrial stress assay. This does direct and live measurements of oxygen consumption. In the first part on the left, this is just resting conditions. You can see that Williams syndrome cells consume less oxygen. When you inhibit complex 5 with oligomycin, both oxygen consumption goes down in both control and Williams syndrome cells. But then when you uncouple oxidative phosphorylation from the active ATP synthesis itself, effectively maximizing cellular respiration, you can see a big difference between control of Williams syndrome cells. And then when you inhibit the entire oxidative phosphorylation pathway, then you both conditions go down to nothing. And then when you do an endpoint luciferase assay, just in resting conditions, so it mimics the conditions used here, you can see that actual ATP is decreased in Williams syndrome cells. So we wanted to ask again further, like how is it really, like if it associates with complex 5, how is it really, affecting ATP synthesis? And so J domain containing proteins are co-chaperones. It's a really small co-chaperone, so we thought that most likely it's just kind of stabilizing the interactions. And so what I did was to, again, isolate mitochondria from control postmortem cortex and Williams syndrome postmortem cortex and use a light and heavy detergent treatment to try to dissociate the complexes from the mitochondrial membrane. And what you can see is that in Williams syndrome, you can see a lot more of the complex 5. This is a Blue Native page Western blot for a protein of complex 5. You can see a lot more of complex 5 dissociate from the mitochondrial membrane. What's interesting is that the complex 5 actually exists as a dimer, and so you can see more dimer here. And then the monomer also comes out. But then when you switch it to high detergent treatment, you can actually release the entire complex from the controlled conditions as well. So this is kind of elucidated here in the model. We have some other evidence that DNAGC30 might also interact with some of the other proteins of the oxidative phosphorylation pathway that exist as supercomplexes. So we think that DNAGC30 is kind of like a glue that kind of helps some of these proteins stick together. Their concentrated supercomplex formation helps facilitate ATP synthesis to be more efficient. So to summarize here, I've gone over the molecular component, but some of the stuff I didn't get to talk about today was some of the cellular features that we saw in Williams syndrome cells and in DNAGC30 knockout mice, which is that fewer spines, less than dendritic branching, thinner axons, and altered electrophysiology and mitochondrial morphology that are consistent with less ATP synthesis. And then we also did behavior in these mice, and they have a hypersociability-like phenotype and a increased anxiety-like phenotype that is reminiscent of Williams syndrome. So with that, I'm happy to take any questions. I'd really like to thank the Neurobiobank, who we got the brain tissue from. Like without this, really wouldn't have been possible. So now, thank you, and questions.
Hello.
Yes.
Very nice talk. Can you say a little bit more about the postmortem Williams syndrome specimens, age, and et cetera?
I can say a little bit more. I don't have all of their metadata memorized, but they are, I think there's an 18-year-old and a maybe 38 and 40, that age. The two are 38 and 40, I think, something like that. I don't recall the sex off the top of my head. PMI is for sure less than 24 hours, which I appreciate is kind of long, but we matched them at least by age and by sex, and PMI as best we could with our in-house control samples. So we don't think that that is a covariant that might explain this phenotype. MFC, and OFC.
Thank you.
Yeah, you're welcome. Sorry, should I medial frontal and orbital frontal? I'm used to the chest and nomenclature, but just so it's...
You also mentioned that in duplication it can lead to schizophrenia. In autism, there's something that mitochondrial dysfunctions implicate in schizophrenia. Can you hypothesize that how duplication could...
Sure. So in this case, we didn't, we didn't study it. It's not that I didn't show it. We didn't study the duplication situation here. In part because we had a little bit of conversation at lunch that some of these CNVs are not fully penetrant. So we felt that the knockout was in the mouse and then a Williams syndrome would be a cleaner and more likely chance to see a result. With the duplications, it would be hard to know how much more for DNA JC30 you would need to see an effect if there is one. Regarding the link to schizophrenia, we also see the highest expression in layer 3C pyramidal neurons of the neocortex, and these are also really affected. David Lewis has shown a lot about the role of mitochondria and how they are affected in layer 3C neurons in schizophrenia. So this actually just kind of fits in. It's nothing new in terms of that aspect, but it kind of fits in with the work that he's been doing as well as others.
Could you show again the location of this gene in the Williams syndrome chromosome region, please?
Oh, in the chromosome region, sure.
I also didn't quite get how you arrived to this specific gene. So where is it exactly?
So it's right in the middle.
Oh, okay.
And so here it's just colored by anything, right? And then we colored it by cluster post-analysis just to the other concern would be that they're a cluster of genes, and so you could say like maybe they're all just expressed together in one group, but that's not the case. They're kind of staggered clusters 1, 2, and 3. And so we use that as partial evidence to say that if they're co-expressed, they would have the potential to work in a similar molecular process. The reason we picked it up was because when we did the gene ontology on cluster 1, synaptic transmission was not surprising, but the mitochondrial processes was surprising. And then DNA JC30 had one of the highest p-values for driving the mitochondrial gene ontology. And we saw gene ontology is top of the list, or DNA JC30 is top of the list. What does it do? There's no literature on it. So this was, as a cell biologist, this was beautiful. It's a chance to clear, blaze a trail for some new biology.
Uh-huh. So, but we were used to think more of LIMK1 as a critical gene in this region. So, but I see that they are in the same cluster.
Yeah.
Do you know, because the processes that LIMK1 regulates that have to do with actin, cytoskeleton, and so on and so forth are bound to require a lot of mitochondrial energy to be carried forward. So do you have any thoughts about this?
Absolutely, yeah. So this still is, so I don't think that DNA JC30 is the smoking gun. This is a mechanism, a part of many mechanisms of a very complex disorder. So you sound like you're very aware that GTF2I also has a very big role in Williams syndrome, as well as limb kinase. And so some of these features that we see in Williams syndrome might be cooperative in terms of which genes are controlling which molecular processes that are aberrated. We do have the single gene knockout mice that confirms a lot of this stuff. The killer experiment would have been a rescue in the cells to overexpress DNA JC30, but that wasn't, it's not a trivial experiment to do because of what I mentioned to Karthik in terms of how high do you rescue the gene expression level. So absolutely, I mean, limb kinase does play a role. This is a part of the game.
Thanks for a great presentation, Andrew. Just a question for me. I really love this approach of using co-expression to find candidate sort of coordinated subsets of genes. The CNV we look at is the whole X chromosome, so I'm very attracted by the idea of passing some of the large candidate space. Did you find that any one of these clusters, if you use them as seeds to catch other genes in co-expression, enriched for candidate genes associated with autism, for example?
We could have done that. We did not. Just as a first pass, I mean, these genes jumped out at us. But if we were to go down the list or intersect with some other gene expression atlases, so this is used with brain span and psych-encode data sets. But if we were to use some other data sets from postmortem brains, that we would certainly look for intersection of genes that might come up as being novel players in developmental disorders, absolutely.
Thanks, that's a lovely approach.
Would you happen to know, there's been several microdeletions that have been defined. Have you had or you or somebody else had an opportunity to see if DNA J30 is also affected in those microdeletions, and which ones correlate with them? Because they also have different effects that affect cognitive versus not cognitive.
The microdeletion patients are very interesting. I've only seen one that, it's not a clean microdeletion of DNA JC30. It's the right half of the locus from DNA JC30 on, so it wouldn't be very clean, but it would be something to do for the next step of experiment. So, you know, we're continuing with this project to do more transcriptomic analyses. It'd be great to have some of the microdeletion patients to tease out some of the smaller parts that you mentioned, but we don't have that yet.
Great, thank you, Andrew.
Thank you.
Thanks to all the other DataBits speakers and the other speakers today. I just wondered if the speakers would be okay to come up to the seats at the front for our final discussion session. That would be great.
Thank you very much.
If the will is there, I know it's been a long and fabulous couple of days.
You'll be on the bus by 6 o'clock, I promise.
Gin on tap.
We have one speaker. I just looked to see what time it is, and my Fitbit has died. I think that's sort of a metaphor for Friday evening. So anyone can start asking a question at any time. This is just free for all. We have a question. Go ahead.
So I just want to ask about transient projections, transient connections in the developing brain. What are your thoughts about why are they there, especially from layer 5, and how are they removed, and what might be their function in development, if you have any thoughts about it? In general, maybe for you, I don't know. Or somebody else in the room.
So I think it's just, I really, the reason, if I go back to what Barbara was talking about, you know, you have these rules that seems to be very conserve how you build a brain, you know? And one thing that brain likes is the redundancy, you know? Brain is incredibly plastic. We sometimes forget that, you know? As long as you are young. And so I think it's probably some way of mechanism to ensure that you survive as a species. It's also a lot of others. So that's what I would say. And then in monkeys, I mean, it's probably some of these features are a little more exaggerated just because of the long gestation, size and stuff like that. But I think it's a greater problem to plasticity and to be able to survive. That's pretty much the whole point of evolution.
I think Leah's work and a lot of the other, when you look at really, really simple organisms, it's much more deterministic pathfinding and cell-type specification. As you get a little more complex, there's more of a kind of role of experience and other stuff. So, I think that's part of it as well. It allows for more shaping of the brain, not by some genetic hardwiring, but the genetic hardwiring actually allows it to be more shapeable and plastic in a kind of ironic way, kind of reduces the physical constraints, opening up other opportunities for growth and plasticity. hand waving, of course.
I was wondering, there's been a lot of, in the past years, the importance of excitatory inhibitory balance and how they may contribute to these disorders. And today we've heard a lot, and yesterday also, about the differences primarily in glutamatergic cortical neurons. I was wondering if you could just mention into your studies, I mean, do you see variation in the GABAergic neurons? It seems to be less, and you have any comment about that in general?
Across a lot of disorders, I'll just say autism and schizophrenia, among the most downregulated genes is parvalbumin, somatostatin, a lot of interneuron markers. So it's clearly what you're seeing in the brain is circuit dysfunction. It's not a unitary cell. So the mutation might occur in a parasite or in an oligoprogenitor, and that affects how circuits are set up and then later. And so that's what we're seeing there.
Yeah, and I mean, that's a great question. I'll just say that I think you're right. I mean, from the Allen Institute data in mouse recently, one of the striking features that they show is that if you look at regional gene expression differences in excitatory projection neurons, you find an abundance, as we've all found in our different species. But there's very little difference when you go to the front or the back of the brain interneurons. And I think Fetal has thought a lot about this as well. So it sets up a puzzle because on the one hand, you have this specification that maybe regional and environmentally driven for one type of neurons, but there's a whole set of interneurons that have to migrate in different directions and at very far distances, and they end up being
check.
essentially the same thing.
Who's that? That's not coming up.
And so where is that missing variability, and at what point do they become so specified that they're invariant to local cues? So, I don't know. We also see it in the primates. There are very few regional signatures of expression difference within a conserved cell type, interneuron type in the primate.
Yeah, I just wanted to come back to the comment on the transient connections. So there's some, and I believe, no, you were on a paper last year where there's actually in the mouse brain, there's greater density of projections to some of the motor neurons in a young juvenile animal that then, or infant that goes away as an adult. And sometimes I think of this as it actually might be a bad thing to have too much plasticity or too much control from the cortex and more innate control of behavior to avoid some dangers. An example in the vocal learning field would be that if you produce a lot of varied vocalizations, it's hard for predators to habituate to the varied sounds that a species has, and therefore you're more likely to be eaten. And so therefore you want to get rid of those cortical projections and just make it a boring, simple, innate sound so you're not detected as easily. And that might be a selection against, those projections.
So I think some of the parts that we discussed from various speakers today, the part that I think I summarize is that human cortical superficial layer is somewhat, well, not human, primate, let me say primate. Primate superficial layer, pyramidal neurons are different than rodent. One of the unique features of the pyramidal neurons in superficial layer, they are the ones who are really responsible for heavy cortical connections. And then Stowe pointed out that it's not exclusive, but the superficial layer of the pyramidal neurons are somewhat more vulnerable in terms of the psychiatric disorder related to genes. So, and as multiple speakers point out, that compared to the GABAergic neurons, that these pyramidal neurons, there are greater differences depending on the regions. So putting all these things together, I can't stop thinking about that whether there are any specific cortical connections in the superficial layer, somewhat more vulnerable or more important in relation to these psychiatric disorders, or because in some sense that you have a data set that you can look at because you do know that what part of the brain, superficial layer pyramidal neurons have a specific sets of genes that are expressed, and if you lay on top of that to this a high risk genes, whether any particular connections, whether they give any hint that any particular connection is more important. It's easy to maybe guess maybe the associated cortical areas might be somewhat more important, but I don't know that actually be really detected in those large data sets.
You know, so there's really is a difference that people haven't remarked on that I've noticed about layer 5 versus supragranular layers in rodents versus primates, what are usually a rodent and two primates. But in the distinction that supragranular cells are the intercortical ones is not the case in rodents. So layer 5 has callosal and intercortical projections. So one of the things that you might be looking at in that group is a sort of, it might be a small to large brain, just segregation of function that you might develop over greater developmental time, so that one gets to have just that projection.
Yeah.
So I think.
No. Yeah. I didn't want to. But just to take off on that point, we were talking at lunch. I think from some of John Rubinstein's work, some of the work that was maybe not presented directly, but there was a beautiful knockout of the kind of layer 6 connectivity of TBR1 and the layer 5. When you knock out the layer 6, which is fugally projecting, versus the cortical layer 5, you get different phenotypes. And I think it's the social, you can correct me, social phenotypes that you get from layer 5 in that, which would fit with this issue. And in mouse and human primates are really quite different in that, I think when you look at Allen Institute data and other mapping data, a lot of genes that are kind of layer 5 in mouse are actually layer 2, 3, and in humans it's not unusual. I think Ed Lean once told me about 30%. We did a quick survey that would agree with that. So, the data that we're looking at is human and macaque, which may be different, but the point is that it's a development of cortical connectivity. That our prediction in 2013 from our data was that if you disrupt cortical connectivity, you're more likely to get the kind of core autism deficits than disrupting cortical fugal connectivity. Not that you couldn't do it, but that it's just, it's going to be more likely. And I think that this data from the TBR1 specific stuff actually fits with that. And so there isn't enough data yet. So some of the data that I showed partitioning the autism GWAS, so common variants in GWAS, into upper layers, deep layers, and all the cells based on fetal data. And we've seen Damon's work really with 40,000 cells now really recapitulates the earlier work from Kriegstein's lab with a smaller sample, though, but it really shows that what they observed and their conclusion was correct in that there really is very little regional and kind of diversity among the pyramidal neurons at those early fetal stages of development. I think you mentioned this as well. That looks really, really clear, and it's kind of developing later, either very, probably early postnatal. We don't know exactly when in humans, and so we don't really have the specificity to pull out. Is it a particular subclass that are projecting? But I would probably guess they're probably more widespread than we might think. That's a great question, though.
Hi. So I'm actually particularly interested in whether there is anything known about the molecular mechanisms that control the soma size of the neurons. So that it has been suggested that the bigger the soma, the more stable the connections between the cortical connections are. And it seems that supragranular layers, they seem to have a little bit more bigger soma size. Therefore, the connections with the other cortical region, it seems to be more stable and more informative in terms of signal processing. Is there anything known about the molecular mechanism that determines the soma size of the neurons?
Well, Andrew's work, I mean, he finished in the summer. He didn't have a chance to show the data, but if you knock out DNA JC30, you get smaller pyramidal cells. And if you use an antibody, it's probably one of the best markers for large pyramidal cells. I mean, that was also one other reason we picked that gene, because when he showed me staining, you work on that gene, because it labeled beautifully layer 3C pyramidal cells, bed cells, and minor cells. And they probably the reason because they have a lot of mitochondria. So, and if you look at humans, people have shown also, us and others, if the metabolic genes are also upregulated, I mean, including Dan's early work in humans. And one of the reasons is that humans have also, not just humans, trend in primates, sorry, we should be careful generalizing it that much. But there is a trend in primates, to the best of my knowledge, I know about cetaceans and other, that you have less corticofugal cells. So there are more cortical, okay? And the reason is that they are very expensive now. You have to understand that in mouse, to project from layer 5 to spinal cord is like what? 1 centimeter? But in some humans, it's probably a meter. And it's the same, it's one cell has to make 1 centimeter, 1 meter. I can run 50 yards or feet, but after that I'll drop, because I just never ran. And so it's a completely different game if it's a short versus long distance. So yes, and and if you look at humans, we have really humongous bed cells, minor cells that mites, rodents don't have. And one thing is, and his gene labels them nicely because I think it's one of the mechanisms, a mechanism to compensate for really having you have to have very functional mitochondria. And how do you increase it? First, controlling fusion and fission, which he didn't show you, but this gene does that. And secondly, even the machinery has to be closer to each other. Others have shown that. And really, so cells has to have a mechanism just for metabolic demands to, to change something. Just one second. And so studying that same homologous cell in human versus mouse will give you different phenotype or primate. That's the key. Because those adaptations for big size will...
Yeah. Well, I have a little bit different point of view from a whole lot of developmental size disparity experiments. So there's a whole lot of things have been done by, doubling the amount of input to a region or halving it or so forth, and then looking at what the effect is on cell size. So what we haven't really distinguished here is, is there a difference in cell type? Is that due to the connections? Is it due to the long axons? Or we set cell type in some genetic way prior to that? So what is it that is allowing frontal cortex neurons to get bigger? All developmental, in development, all input is always in excess. So you have many more exuberant axons and things are dying that the axons are retracting and so forth. So you can't, what exactly is setting the limit on how much and how many different kinds of inputs a single neuron can take? And I think we really should be really careful about cause and effect in this.
I completely agree.
Yes.
I mean, if you look at the cortical plate, I mean, even in the motor, you don't see big, gigantic cells early on. So, but also, I think it's probably what
Okay. Maybe that's a good place to wrap it up. Let me just thank the speakers once again. And they've been thanked a lot, and they're wonderful, and they gave amazing talks. I'm just so happy with all of that. I do want to thank also one group that hasn't yet been thanked, who are the heroes, who are the volunteers, who are the non-organizer generals that you've been corresponding with. Can you please stand up, those of you who are here right now?